Okay. A warm good morning to all of you, and also a warm welcome to everyone. It's a pleasure to see all of you here in Tartu, and I think we are going to have more people in this salon, but it is early in the morning, 9:15, hence it is taking time. But we have also seen that there are quite a lot of people that are joining us online in the Zoom. But my name is Ali Reza Fazeli. My name is not Gregory Lavio. We are not clone of each other. We are two different. There is a mistake here. Don't worry. And I am the ERA chair of the Combivet program here. And I thought maybe I have 5-10 minutes that I can somehow speak with you. I can tell you a bit of history of what we are doing here and how long ago. Amazingly, it's now nearly around 6 years that we are somehow running these sessions. And this meeting, this conference, this 2-day conference is nearly our 8 or 9 conference that we have had about different topics related to the extracellular vesicles. Everything somehow started in 2017 when we had our little conference about extracellular vesicles. And since then, the extracellular vesicle research has been supported in Tartu, in both of the universities, in Tartu, in University of Tartu, and also in the Estonian Life Sciences University, by successive grants that we have been able to secure from the European Union. And as a result of it, because it has been also something that I was very interested in it, we have supported it in the work that we have and we are doing. And one of the highlights of this work has been to have this 2-day meeting, and each year we have the 2-day meeting on a different topic. Sometimes, for example, it was 3-4 years ago, I remember it was about interspecies communication with extracellular vesicles. And this year we have tried to have, although it's not 100% that, but we try to have a bit of a team. And then at the same time, we look around what are the latest developments, who are the best speakers that somehow can come, and then we invite them to come here. Hence, you would have a mix of everything in addition to have a bit of a team also in it. And this year, our idea was, speaking with Greg, nearly even 6 months ago, we started this journey to speak about specificity, about how specific is the message of extracellular vesicles for the recipient cells and how that can somehow have an effect in the heterogeneity of the EVs that they are producing. I have to also mention that during the last 6 years, we have gone through a bit of a journey. One other thing that we have done in Tartu and has been very successful is having a kind of course, a crash course of how the basics of working with extracellular vesicles, that so far, I think over 130 different students from different countries in the world have been somehow from different continents. Even during COVID, we had online maybe over 70 participants that was coming to our course, and during the COVID we had to do it via videos and making videos, and our students are coming from all over the world. This year we had from the United States and even from India subcontinent that they were coming here, not mentioning all over Europe that are coming. And that is also, I think, another success of the work that we do. And as a result of that, we are spreading the messages of extracellular vesicles. Then one of the things that I think is also important to say is when we started this, the EV field was relatively young. And still I can say it is young. It's not somehow so much mature compared to the other fields. But sometimes always when I look at the work that has been done in the last few years, I want to compare EV field somehow like to the situation with the stem cells, particularly the embryonic stem cells. I was in the beginning of the embryonic stem cells when it started, and I saw also somehow how it did grow very fast, and it was a lot in the news everywhere. It was coming up. And then during the years, it slowly by slowly did go down, maybe partially. I don't want to say completely it has gone down, but especially the embryonic stem cells. Partially, it was due to the fact that a lot was somehow promise that would somehow come out of the embryonic stem cells. But at the end, that promise didn't materialize so much as it was somehow expected. All that therapeutic applications and so many other things, although it can be a debate. But I feel the EV field is also going through the same direction. However, and that is maybe another 4 or 5 years, because we are promising a lot that the extracellular vesicles are going to deliver. And it is on our shoulders somehow to make that delivery to happen. If that delivery doesn't happen, if we wouldn't somehow be able, whether in the diagnostics, whether in the therapeutic, to be able to deliver that, we would also, that probably would happen also to extracellular vesicles, that it wouldn't be as hot topic as it is now at the moment. Hence, that is also one of the other themes that we have during these meetings, to see how we can reach to that delivery and how can that delivery somehow go forward. Having said all of this, bringing you all here and coming here and having this meeting is, and you would see it also during the next 2 days, it's a substantial logistic issue, and I wouldn't be able to stand here so happily saying to you that is happening without having the backup of a number of people that behind the scenes from the last 6 months have been working very, very hard to do this, to accomplish this task. And particularly, I have to mention that each year we have a leader that we would follow, and this year our leader has been Subhashini, and I want to thank her already, and I'm sure by the end of the meeting we would be all very grateful for the meeting that she put together, for the work and the amount of the work that she has done with her team to make this happen. I think that is everything I wanted to say, and by this I would like to give the session to the chair of the session for inviting the next speakers. Thank you very much.
Good morning, everyone. Thank you very much, Alireza, for the introductory remarks. And so myself, Suranga, and Chanaka will be chairing this session. And so I'll be inviting our speakers to the podium. And I think we have one online session in this morning lecture series as well. So to start today's procession, I think we have a very nice start with Dr. Gregory Levu. He's from INSERM France, who's an investigator and head of the ERLU 1316 group, I would say. And he has been working on EVs for a long time, and he's known for his work on the membrane trafficking, I would say. So I'm sure today we are going to hear a lot more on the membrane trafficking. So please, Dr. Gregory, the floor is yours.
Good morning. So thank you for the nice introduction. Thanks for the invitation. Thanks to all the organizers. And Alireza, I would like to especially acknowledge Annika Hurt, who made it possible, because I had some issue yesterday and really she made it possible. So thanks for that. Okay. Is it possible to have a pointer, a laser pointer? Ah, so I have to. So, okay, but okay, okay. So I will manage. Okay. So today, what I would like to do is to spend some time with you and focus on one aspect of the trafficking and the transport of extracellular vesicle, which is their uptake and their content delivery within the acceptor cells. No, because apparently some people are in Zoom, so I have to. Okay, thank you. Thank you, sir. Okay, so as everybody knows, so extracellular vesicle can navigate and probably exchange material from a donor cell to an acceptor cells. And so since, many years now, we start to have a precise view and almost a comprehensive view of the mechanisms that regulate the secretion of the extracellular vesicles. The same is not true when we look on the side of the acceptor cells. Actually, the uptake parts, so what I call the uptake is when, the EV will touch and then get internalized and ultimately release their content inside the cells. So that is what I'm going to call uptake. And the final content delivery is not well established, at the cell, at the cellular level, but more importantly, at the molecular level. And it's actually highly debated, right? So we have two major questions that are not completely solved and that are still highly discussed in the community, which is, does EV content delivery truly occurs? And if yes, what is the site for the content delivery? So we have at least two options. So the first, that I will call the direct pathway, will consist of the EV touching the plasma membrane and bound the plasma membrane to fuse later on with the plasma membrane and release the contents. So far, when you look in the literature, there are only a few papers that propose that pathway. The other pathway that has been used by many viruses is the one that I call indirect pathway, in which the vesicle will be first internalized in the endosome, and then for the content delivery, if that occurs, so then the EV will fuse with the membrane of the endosome. And you will see that the endosome as an entry point is supported by more paper, which doesn't mean it's true. I mean, and because we don't do science by vote, right? So the first point is the first question is, what is the entry point? The second one is, does EV content, truly occurs in all type of cells? And again, here is still highly debated. So many years ago, everybody wanted the EV to deliver something inside cells in a specific or non-specific manner. But I would say that over the last 5 years, several groups try very hard to work on that. And what you can see here, it's a selection of several papers. In some cases, you can see that the group could not establish content delivery. And in other cases, they could establish content delivery. So we don't have a consensus yet. And what I'm going to show belongs to the second section, when we can detect the content delivery. But as you will see, it is not a high yield process. Okay, so in my group, what we wanted to, when we started to work on that on that question, we wanted to solve and answer a very simple question regarding the entry point. So the hypothesis is that if the endosome is the entry pathway, so usually the signature of the endosome is acidification. So we ask a very simple question: is acidification required and sufficient to trigger EV content release? And that's a mechanism that is used by many viruses. So we hypothesize that perhaps the EVs will follow the same pathway. And so that one has been published. So what we did first was to try to reconstitute the final step of the content delivery by recreating in a cell-free assay that final step. So it was a very simple approach. So what we wanted to do was to isolate EVs that contain the cargo and incubate in a buffer the donor EV with a putative target membrane extract from plasma membrane. And in the buffer, we added the proteases. So the rationale is that if the cargo is inside the EV because of the bilayer, so the cargo should be protected from the protease. So now, if a signal trigger fusion between the EVs and the target membrane, the cargo is expected to be released in the buffer and accessible to the protease that can now degrade the cargo. So that's exactly what we did. So we purify and isolated EVs on the one hand. On the second hand, we develop a new protocol to isolate plasma membrane sheet as putative target membrane in solution, and we mix them together. And then we look by a well-established protease protection assay if the cargo that was inside the vesicles was protected or not. And here you have the key results. So we look by Western blot, you know, for protection or not of the EV cargo that is represented here. And we use a chaperone HSP70 that is a well-established generic cargo present in many types of vesicles. So here you have the control, you know, and here you have the same cargo in presence of the proteinase K. And as you can see here, most of the cargo is protected. It's also quantified here, suggesting that, as expected, the cargo is truly inside the vesicle. Now, if we only acidify the vesicle without acceptor membrane, you can see that the cargo is also still protected. There is no effect of the acidification per se. Now, of course, the positive control, if we use detergent to disrupt the membrane, so now we break down the membrane and the protease can degrade the cargo. That was a positive control. So now, importantly, the key experiment is there. So we mix the EV with the plasma membrane sheets. And as you can see here, if we do nothing else than just putting together EV and plasma membrane sheets, nothing happens, the cargo is protected, there is no content release. But now if we trigger acidification, and we can do that because it's a cell-free assay, so we can control the environment. So if we acidify both EVs and plasma membrane, we can see that now the cargo is degraded by the protease, suggesting that the cargo has been released, probably because the EV fused with the plasma membrane. So that was the first, key result for us, suggesting that, content delivery could occur, and that was pH dependent, suggesting that if it happens, that would happen in the endosome. We then establish that protein are also important because if we now do a pre-treatment of the donor and target membrane with a proteinase K to literally shave and remove all the protein that are at the surface of the EVs or the acceptor membrane. So now we will block the content delivery. And that is shown here. So here is the result that I showed before when we acidify. So we can see that there is degradation of the cargo, but now if we do pre-treatment to remove the protein at the surface of EVs or plasma membrane, we inhibit the content release, suggesting that EV content delivery is protein dependent and pH dependent. So we are very happy with that first set of results, but we of course wanted to recapitulate that with living cells. So for that, we developed a new assay taking advantage of a very sensitive enzyme called nanoluciferase that we use as a marker to follow the fate of EV cargo inside acceptor cells. So we isolated our cargo expressing the nanoluciferase HSP70, the same generic cargo that is inside the vesicle. We use ultracentrifugation and flotation assay to isolate EVs. Again, we demonstrated that our cargo of interest was protected and truly inside the vesicle, and we characterize the vesicle by Western blots. And as you can see here, they are enriched with classical EV marker, such as CD63, ALIX, CD9, but they are completely depleted with calnexin, an endoplasmic reticulum marker. And so then what we did is we incubated our nanoluciferase tag EVs with the acceptor cell, and we measure the uptake efficiency within the acceptor cells. Unfortunately here, because of the computer, the curve has been deformatted, but you can still see here the line. So you have to appreciate here. Sorry for that. It's a format issue. So we did first a dose response. And the first thing that you can see here is that the process is not really efficient. Because even when we go at very high doses of EVs, we cannot saturate the uptake. Okay, to give you an element of comparison, if you compare with your, the dose response, if you do that with LDL, for instance, which is a well-established, ligand for the LDL receptor, you will reach saturation here. Okay, and it's known that LDL is using a specific receptor for that. So that was very surprising at the beginning because it suggested that the uptake was not really efficient. We also perform a kinetics. And again, as you can see, so then the uptake will grow over time. But again, it's not really efficient because if we look at the first hour, there is only 1% that can be uptaken. Okay. So again, that suggests that it's a poorly efficient mechanism. So then we wanted to test it if the EVs were able to first bind the membrane prior to being internalized. So for that, we did two kinds of experiments, one at 37, which is the one that I show, and one at 4 degrees here. So at 4 degrees, you inhibits the internalization of the EVs or any particles, but at 4 degrees, you keep the protein-protein interaction. So in other words, if you have specific interaction that is used to recruit your EV at the plasma membrane, you should detect some luciferase activity associated with the acceptor cells, which is not the case here. So again, if you do the very same experiment with the LDL particle that will be tagged, so you will see a strong binding. Okay, so altogether, that experiment suggests that the EV uptake is a low yield process, and probably at least in the cells that we try, and we try many cells, there is no specific bona fide receptor that would be specific to tether and capture the EVs, suggesting that probably the EV are internalized through micropinocytose that require dynamic membrane movement, but no bona fide receptor. As many other groups did, we wanted to ensure that the EV were internalized. So for that, we use a confocal microscopy and did colocalization study with RAB5, an early endosome marker, and LAMP1, a late endosome marker that also localized the lysosome. As you can appreciate here on the example, so you can see that we could detect the EV marker GFP-HSP70 colocalizing with early endosome or late endosome. And it is quantified here again because of the formats. You cannot see the graph, but you have the key number here. So basically, half of the population was localizing with the early endosome, while the second half was localizing with the late endosome, suggesting that the EV will follow all the lysosomal maturation pathway. Okay, so that was already interesting. We developed an assay to measure the global uptake, but what we really wanted to do was to measure acutely the EV content delivery. In other words, what is truly released inside the cell. So for that, we kept the same protocol, but we add an additional step in which we did cell fractionation after incubating the vesicle with the acceptor cell. And the goal here was to disrupt all the membrane and separate the cytosolic fraction that will remain on the supernatant and the pellet that will contain all the membrane fraction. So the rationale here and the reasoning here is that if the EV bind the membrane or if they stay associated with the endosome, they should go in the pellets. But if for some reason content delivery happen, and if the cargo, the HSP70 luciferase is released in the cytosol here or here, now we should be able to directly measure the activity in the cytosol. Okay, so that's what we did. We just separated both fractions. And as you can see in the following slide, so when we did that with the soluble cargo here, you can see that approximately 30% of the cargo was found on the cytosol. So to summarize, so we were able to have 1% in the first hour that will be captured and internalized. And from that 1%, 30% will be a candidate for content delivery. And it's specific because if we look for nanoluciferase tag tetraspanine that remain always associated with the EV, we do not reach the 30%. Okay, very satisfyingly. So we also established that acidification is important for that process, because if we block endosome maturation and endosome acidification with baphyllomycin, you can see that we don't change the uptake, but we dramatically decrease the content delivery. Very consistent with what we observe with the cell-free assay. Acidification is required for the content delivery. So we suspected that fusion would be required for such a content delivery. So to answer that question, we took advantage of a family of protein called IFITEM that have been shown several years ago to prevent fusion with the membrane and many viruses membrane. And again, because there are many similarities between viruses and EVs, we suspected that perhaps the IFITEM protein will do the same function, blocking a putative fusion. So what we did is that we forced the expression of the IFITEM protein, either IFITEM1 or 3, on the target cells. And you can see here that they label the plasma membrane, but also all the endosomal compartment. And again, now we tested the content delivery in those cells. And as you can see here, So when we force the overexpression of IFE10 in the acceptor cell, we strongly reduce the content delivery, suggesting that fusion is probably required, right? So we did that with the cell fusion assay, but also we recapitulate that, reviving the cell-free assay, as I showed before, except that now we put the IFE10 protein overexpressed in the PM sheet. And so as you can see here, in the mock condition, when there is no IFE10, that's what I showed before. So there is a strong degradation of the protein, so a strong content delivery. But if you overexpress IFE10 or 3 in the target membrane, you prevent the content delivery, strongly suggesting that the content delivery occurs from membrane fusion that can be inhibited by IFE10 protein, right? So both, the cell assay and the cell-free assay suggests that content delivery can occur. It's not a very efficient process, but we can measure it. And that the entry point is within the endosome, and that requires the acidification of the endosome. So now that we started to understand better at the cellular level, we wanted to test further at the molecular level. And for many years, several tetraspanine, including CD63, CD69, that are classical marker, has been suspected to play physical function in the EV-mediated transport, including perhaps the uptake and the content delivery. So with some collaborator, we took advantage of Cas9 editing to basically knock out either CD9 or CD63. And here you have a Western blot that validates, donor cells that are knocked out for CD63 or CD9 here. And first, so what we did is isolated EVs from knockout cells and tested their ability to do the uptake and the content delivery within acceptor cells, wild-type acceptor cells. And what you can see here is that we couldn't see any differences in the uptake and nor in the content delivery. Okay, here what you see again is a positive control when we block the acidification, suggesting that CD63 or CD9 do not play a role when they are on the donor membrane. We then did exactly the same, but this time we tested the effect of knocking out a CD63 or CD9 on the acceptor side. Okay, so this time we isolated wild-type EV donor, and we incubated the EV donor with the same luciferase assay on acceptor cells that were depleted or not with CD63 or CD9. And here you have the Western blot control, to ensure that CD63 was not on the EV nor CD9 here. And, again, we couldn't see any effect, neither on the uptake, neither on the content delivery. And that has been done in collaboration with our partner, in Utrecht University, with a group of Peter Wader, who confirmed all these results using another assay, another cell line, and other cargo. Right, so we did that independently, and the work has been published this year, and I put the first author here. So together, we concluded that CD63 and CD9 do not play a role in that process. Doesn't mean that they don't play a role at all, but at least not in this process. So with that, I just would like to pause a little bit and propose the model that we think is used for the EV to deliver some of their contents. So we believe that based on the results, we believe that the EVs will be first internalized inside the endosome, and that's acidification of the endosome will probably favor protein-protein interaction to enable content delivery, probably through fusion. And now my group is doing Cas9 screening to identify part of the fusion machinery. And hopefully in the upcoming year, I'm going to be able to show the screening results. Okay, but that's the model that is supported so far by all our work. And it's important to also highlight that we did some collaboration with a group of Carmen Buchreiser at Pasteur Institute, and they work with a Legionella-derived EV, so bacteria-derived EV. And it seems that they are still capable to deliver their content inside mammalian cells. So it's probably a conserved mechanism that can happen with mammalian cells, but also from bacteria-derived EV incubated on the mammalian cells. So it's probably conserved-free evolution, suggesting that if there is truly a familiar fusion protein, there are conserved-free evolution. Okay, so what we did in this first part was really, focusing on the fundamental aspect to understand better the mechanisms that enable the uptake and the content delivery. For several years, EVs have been, almost sold as, very promising vector for therapeutics delivery, assuming that they will do the content delivery efficiently, which is not the case. So we thought that, okay, if we really want to make some efficient, virus-free vector, we need to manipulate the natural EVs to enhance their property. Okay, so what we also did in my group was to basically bioengineer donor cells to enhance basic property of EVs. And our reasoning was that at least, 3 key steps that you want to control if you want to control the phase and the properties of the vesicle. One is the loading of the cargo or the therapeutics inside the EVs. The second one, so now what you want is to have your protein, loaded copiously inside the EVs. Then what the next step that you want is to control the targeting. So then your EVs can be targeted toward the specific target cells. And finally, what you want is truly to increase the content delivery property. And for that, what you want to do is to overexpress a fusion protein. That has been done, for many years, but most of the time using viral protein that can fuse. Okay, and eventually you want to get rid of, viral material. So what we did in my lab was to try to find a fusion protein that will not come from viruses, and that's what I'm going to show here. So the first method that we developed, and that is now patented and published, is what we call a loading system. So we took advantage of a well-established drug-inducible system that take advantage of interaction between the FKBP and the FRB domain that can be triggered by a small drug called dimerizer. So we basically fuse the FKBP domain to the tetraspanin, again, that is a classical marker of the EVs. And you can tag any cargo. In our case, to characterize everything, we again use a luciferase because we can track the cargo to the FRB. And, in addition of the drug, now you are expected to recruit the cargo that is normally cytosolic. So again, what we did is isolation of the vesicles, and to measure the efficiency of the method, we basically measure the luciferase activity in presence or in the absence of the dimerizer. And here you can see the key results just for the loading efficiency. In absence of the dimerizer, you only have a minor portion of the cargo that will be recruited, probably because it's abundant in the cytosol and you have like a bulk loading, not a specific one. But no, if you add your dimerizer, you dramatically increase the recruitment of your cargo. So in that case, we did that for luciferase, but you can do that for any protein of interest. And in our case, as you will see later, we did that for killer toxin. We did that for Cas9 for further editing. Any protein will work. So then what we wanted to do was to control the fusion step, as I mentioned earlier, but we didn't want to use a fusion protein from viruses. So what we did is we added syncytin-1 that comes from the placenta, and that is important for placenta formation. So it's a fusion protein, a human fusion protein, and we force the expression of this protein at the surface of the EVs. Okay, and now what you can see here, again, the work has been published, so I'm only showing the key results. If you look at the content delivery, so here is the control that I showed before, the low efficiency content delivery that you can still measure. So now if you put syncytin-1, you can see a dramatic increase of the content delivery. And is as efficient as VSVG, the viral protein that is mostly used for vectorization. So now we offer another solution that is virus-free for fusion. And the last step, that is a work that is still unpublished, or we'll spend a little bit more time here. What we want was to force the targeting and to address our EV to a specific cell, because as I mentioned before, when we look at the uptake, we couldn't find a receptor, a specific receptor. So we thought that, okay, we need to graft an adapter on the vesicles. So now they can be targeted to the cell of interest. So the idea was here to have what we call an adapter that will be a GPI anchor protein. So we put a cherry to follow with a confocal microscopy, and again the luciferase fused with streptavidin that can bind with biotin. And the idea is to recruit any biotilated antibody that will be bound to the streptavidin through the biotin, and that will recognize the target, in our case here CD8, because we are aiming at editing and targeting T cells. So the idea is to literally have a sandwich between the EVs and the plasma membrane with that complex. And so as you can see here, so now if we do all the combination and if we have the, first the adapter without the streptavidin, you can see that there is no increase in the uptake and the content delivery. So if we add an antibody that is not biotin-related, as here and here, again, there is no increase. But very interestingly, and as expected, now if we add in between, the EV and the acceptor cells, or biotin-related antibody, we can see a dramatic increase of the luciferase activity, suggesting that, yes, we can efficiently force the tethering of the EV that harbors the adapter to the, towards the cells that express CD8. And that can be recapitulated by morphology if you look here. So here you have the CD8 GFP positive cells, okay, in green, and you see a strong reinforcement at the plasma membrane where CD8 is preferentially localized. If you use no biotin, you don't see the EVs that you can track because remember they have the cherry, right? So you can look at them by confocal microscope. So you don't see them binding the EV at 4 degrees as I showed before. But now, if you put the biotin-related antibody, you can see a massive recruitment at the periphery of the cell. So all the red dots here are massive and copious amount of EVs that have been recruited even at 4 degrees at the surface of the target cells that have CD8. And it is specific because if we mix CD8 positive cells with non-positive cells, you can see that the EV will be exclusively recruited at the surface of the CD8 positive cell, but not at the surface of the cells that do not express CD8. You can appreciate the result here by confocal microscopy or again biochemical quantification when here we do a mix between wild type and CD8 GFP positive cells. So we slightly increase and progressively increase, and you can see a nice dose response. When we increase the number of target cells, we see a massive increase of the uptake. Okay, validating our targeting system. So now the most interesting part. So because now we have the 3 modules, right? The loading, targeting, and the fusion. So what we wanted to do is can they work synergetically, especially the fusion protein and the targeting system? And the answer is yes, because now if we only use the targeting system and now if we add on the top of the targeting system the fusion protein, you can see that we can jump from here to here, basically. Sorry, from here to here, all right? So what you will do with CD8, with the targeting, you will force to go to your cell of interest, but then because you have the fusion, you will release even more inside the cell. So it's synergistic, okay? So now with that, what we have in our hands, it's basically 3 modules that we can put together. That is versatile because here we show CD8, but we can, if you have a biotin-related antibodies against any protein of interest, a cell surface marker, you can use it. And so now what we are trying to do is to develop a new class of vector with specific application. And so I have no time to show the result, but some of the work has been published. So what we are basically developing is what we call killer EVs. So using the loading system, what we do is now we force the loading of the catalytic site of the diphtheria toxin that can inhibit protein synthesis and kill the cell. And we establish that donor EVs, containing DTA and equipped with syncytin-1 can efficiently kill cancer cells. We also develop editing cells for gene therapy. So we force the loading of Cas9 inside the donor EV. And again, when they are equipped with syncytin-1, you can have up to 30% of editing of the acceptor cells. And now we are trying to do that specifically with the CD8. So that work is not published, and I will probably show it in the upcoming year. So with that, I would like to acknowledge a member of the team who did the work, especially Emeline Bonsergent, who did all the cellular and the cell-free assay, Cheryl Brie and Julia Dancourt, who work on the bioengineering of the EVs, and our collaborator at the Institut Curie, INSERM, and Utrecht University. And I would like to thank you for your attention, and I will take any question.
Thank you, Greg, for very interesting talk. And so now the floor is open for any questions. I think Albert has the first question.
Thank you, Greg, for that beautiful work. I think it's the first time I actually hear your talk, so it's great. I made it in time. So question related to the uptake, right? So you're showing basically 1% can uptake, and only 30% of that 1% actually get released the content. So to what extent do you think that limited uptake at the membrane is generalizable, or whether it's just cell type specific? So that's the first question.
So perhaps I will answer that question. So we try many cells. So what I show here are mostly HeLa cells, but we also try HEK. We try, a model of dendritic cells. We try macrophages. We are supposed to be highly competent for, and it was slightly better, but none of them were, really better. Mm-hmm. So when I say 1%, for instance, with macrophagy, it would be like 3%, something like that.
Got it.
And we also try, so human to human and human to mouse, and it was relatively the same. So we don't believe in neurons. When we work with cell in culture, there is no species dependency.
Great. And I guess the other question that I had was related to the model where you think that you have a surface molecule on the EV that's acting sort of this ligand receptor cognate relationship to release the content. So in your experiment where you treated with proteases, you treated the EVs with proteases. So do you see all the upstream steps occur, but it's just the final step of release that is gone?
So we didn't do it that way actually because we work with the cell-free assay only. So when we do that, we only look at the final step. Mm-hmm. So when we do, so it's still obscure what's really happening in the cell-free assay. When we shave the protein, we don't know if there is no content delivery because there is no contact anymore. We can't visualize that yet. Basically, we don't know if there is no tethers, mm-hmm. no tethering, so then no fusion, or if there is just no fusion.
Yes. Okay.
Okay. So what we are doing now to answer that is we are adapting our cell-free assay. So now what we are doing is we are depositing plasma membrane sheet on a glass cover slip to basically monitor with turf microscopy, so fluorescence microscopy, fusion and tethering independently. So we will answer that question in a couple of months, I believe.
Great, thanks.
Greg, here. Thank you very much for your great presentation. Regarding your cell-free assay, I wonder about the contribution of lipids, because as you know, plasma membrane has an asymmetrical representation of phospholipids. And do you have an idea whether fusion is accomplished more efficiently on the inner or outer leaflet? And also regarding the lipid composition of the plasma membrane compared to the lipid composition of endosomal compartments, what can you say in this regard?
So to answer the question, so it's true that there is one main caveat in the cell-free assay that we, as it was shown here and published, is because in solution, it's possible that the EV access both sides, yeah. the one outside and the one inside, right? Yeah. So what we did is to test, because if it was not physiologically relevant and it was from the inside, then at least what we could do is to test is ATP was important for that, because then you would expect that the cytosol and the ATP would be, so we couldn't see any effect. Okay, so but that's not the ultimate proof, right? So that's why, again, what we are doing right now is we are depositing and we are controlling the orientation of the plasma membrane. Okay, so it's not published yet, but I can share with you what we are doing now. So we know that we have proper orientation of the plasma membrane sheet on the glass with a luminal phase, on the top. And so if we load a massive amount, a very high density of, EVs, now we will see them to start to tether and to move. We don't see fusion yet, but at least they attach at very high concentration. Okay, so we believe that when we see content delivery, it's probably because it's with a proper phase. Okay, now concerning the the lipid composition, if you compare plasma membrane and endosomes, so there are differences, but also many similarities.
Right.
Okay. So my guess is that perhaps, the composition of lipids, they will be, enriched in the endosome and more adequate to favor the fusion, but you will still find some of them in the plasma membrane, and that would be probably enough and explain why, the fusion occur.
So one of the reasons I brought this up is because, as you know, Grunberg's studies have shown that LBPA, also known as bismonoacyl glycerophosphate, and ALEX may be involved in back fusion of intraluminal vesicles.
Yep.
In a similar way that exosomes may be back fusing and releasing their content. Have you, in your cell in vivo assay, depleted ALEX and see any effect?
So we had a knockdown, not a knockout, but a knockdown. We didn't see a strong difference. And also with the cell-free assay, what we did is not published, but we did that extensively. We use an antibody against LBPA that has been always described as an inhibitory antibody.
That's right. Yeah.
We didn't see any effect.
Oh, that's interesting.
So for us, that was a suggestion that LBPA is probably not involved.
So you guys
yeah. But to be completely rigorous, and I choose my word carefully, the antibody has been proposed as an inhibitory, antibody. It's not validated like that, so we don't know if it's truly an inhibitory antibody. Okay. We didn't test any other functions, so
right.
So that's the limitation.
Thank you.
Just one point I want to also mention before that at the end of the day, we are going to have 1 hour of moderated discussions, and probably anything that you hear that is a bit controversial or so, we can put it as questions up there and somehow to discuss it during that 1 hour. And one of those controversial, actually, the first question was someone, in the way that you explain it, Greg, the system is very wasteful. You just 0.3% of it just gets delivered. And then after that, if there is any machinery inside the cell to read that 0.3%, maybe it would become even 1,000. In nature, somehow to have such a wasteful system, does it make sense?
Well, that's the big question, but you are working with fecundation, right? And so I think for me, one of the best examples to have a little input that can change a lot if you look for spermatozoid and sperm maturation, right? So again, we don't have the ultimate proof, but there is
very clever.
A series. You invited me, so I try to be nice. But so for everybody, what is very important and what has been published is, so if you look at the EVs that are secreted by the APDD where the maturation occurs. So you have different zones. I speak under your control, right? But basically, I don't know the proper term, but you have an upper zone and a lower zone. And so if you look at the EVs that are secreted, the RNA content is different. So now when they take the RNA from the lower region and they take the sperm from the upper one, and they inject the RNA that that have been described to be in the EVs, they can force the maturation of the sperm. So that suggests that you don't need that much from the donor, right? So if you work with, and for instance, we use that as a control, if you have a transmembrane protein, you cannot have such a delivery because it's always remain associated with the membrane, right?
Yes, but I guess those could be like remodeled inside the cell, and like and also the lipids are like transported from different organelles to another. So
Okay, so then if you have a transmembrane protein with a cleavable part, that will be in the cytosol. So perhaps you know that would be a model. We didn't try that, but other group had such a, a model in which they have something that was cleavable. And at least one group shows that if it's cleavable, then you can find the small part in the cytosol. It's in one of the paper in the presentation that I showed before.
yeah, interesting. Yeah, thank you.
Thank you.
Yeah, Greg, you have to wait. You have a souvenir from us. Yeah, thank you very much, Greg, for your talk, and also, yeah, we are glad to have you back again. Thank you very much. We have a certificate for you guys. Okay, so now we are going to move to the next talk of the day. So we have Dr. Albert Liu from University of Barcelona, from the Department of Biomedicine. So he has been working on membrane trafficking, but mostly on RNA biology and genomics with a special focus on membrane trafficking. So today he's going to talk to us about understanding exosomal microRNA sorting from genomics application to basic biology. Yeah, Dr. Liu, the floor is yours.
First of all, I would like to thank all the organizing committee for this very kind invitation. I was very happy to be able to come here and present our research. So Greg gave us a fantastic talk describing mechanisms of EV uptake and delivery, content delivery. But now I'm going to go some steps upstream on this process. And I'm going to be telling you about first the development of artificially barcoded microRNAs and how we use this tool to interrogate several aspects of the underlying biology of extracellular vesicles. And then in the second part of my talk, I will be telling you about a possible role between KRAS and hnRNPA2B1, which is an RNA binding protein, and how they interplay to regulate exosomal microRNA content. I mean, sorting, sorry, should I say. So there are about 10 to the power 16 extracellular vesicles circulating right now in each one of us. And it wasn't until very recently that these vesicles were considered just cellular waste, as you'll know. But in the past few years, there has been an increasing appreciation that these vesicles may play important roles in a myriad of physiological and pathological processes. For example, extracellular vesicles are thought to promote cancer growth and metastasis, but as well as they have been proposed to regulate, for example, immune regulation. And altogether, these vesicles then hold promise as diagnostic biomarkers and therapeutic vectors. Greg showed us some examples of how these vesicles can be engineered for these purposes. So I'm just going to tell you about some generalities that I'm sure you all were known about. There are two main types of extracellular vesicles, microvesicles and exosomes. Exosomes are generated by inward budding at the limiting endosomal membrane. And there are two main mechanisms of exosome formation, SCORT-mediated or ceramide-mediated. SMPD3 is the neutral sphingomyelinase gene that converts sphingomyelin into ceramide. And together, these two pathways generate these intraluminal vesicles that, upon release, become exosomes. The process of release can be regulated by several factors in the cell, including calcium sensor proteins such as synaptotactin 7, SNER proteins, YKT6, syntactin 1A, small RAP GTPases such as RAP27, RAP35, RAP11, RAP7, or other endosomal channels such as mucolipin 3. But the reality is that both exosomes and microvesicles can be generated by similar pathways. And again, ceramide and the SCORD pathway are also regulators of microvesicle generation. So given given the current isolation methods, we cannot clearly distinguish these two types of extracellular vesicles by typical isolation methods, as I said. Finally, remodeling of the subcortical actin cytoskeleton has been involved in both, or has been shown to regulate the release of both microvesicles and exosomes. So whatever is the case, we know that EVs carry a wide range of biological molecules, either associated to their membranes. So on the EV surface, we can find proteins, transmembrane proteins such as cerebrospinins, integrins, MHC class molecules, but also peripheral proteins. So proteins that bind to proteins that are transmembrane proteins of EVs can bind other peripheral proteins on its surface. There are also lipids that may play important roles on these vesicles. They are enriched, specifically enriched on exosomes and microvesicles, for example, phosphatidylserine. And like I said, there can be also other surface-associated protein complexes, such as LDL. Other cargoes are intraluminal or reside in the lumen, and these include proteins, such as chaperones, as Greg showed, HSP70, and also nucleic acids, mRNA, tRNAs, and microRNAs. So microRNAs are transcribed in the nucleus, exported into the cytosol, where they're processed, matured into single-stranded small RNAs that repress translation by multiple mechanisms. It is known that a subset of microRNAs can be targeted to extracellular vesicles, and a number of RNA binding proteins have been proposed to be involved in the process of sorting microRNAs, cellular microRNAs into these types of vesicles. Here, there are a number of these proteins, and one of these proteins is also HNRNPA2B1, which I'm going to be talking about throughout this talk. So, taking advantage of current knowledge of how microRNAs have been proposed to be exocytosed by extracellular vesicles, we developed artificially barcoded exosomal microRNAs, or as we call them, bexamirs. So bexamirs were designed to contain a 15 nucleotide random sequence, and this is already published work. And this 15 nucleotide random sequence, which is placed right at the base of the stem region of the microRNA, this is a typical structure of an unprocessed precursor microRNA. So this 15 nucleotide random sequence, which has complementarity with the two strands of the microRNA, is followed by an exosome targeting motif placed right at the 5' end of the 3' end of the microRNA. This exosome targeting motif was described by the group of Sanchez Madrid, and it was shown to bind HNRNPA2B1, which sorted microRNAs containing motifs in the fully mature microRNAs, and in doing so, HNRNPA2B1 sorted microRNAs that were to be exported in exosomes. So bexamirs are expected to be processed just like endogenous microRNAs into 22 nucleotide long matured bexamirs in this case. And the motivation of developing such tool was to assign a unique molecular identifier to each one of the extracellular vesicles released in a cell culture in pool format. So each one of these cells would express a unique bexamir, and the vesicles from each one of these cells could be identified by reading the 15 nucleotide random sequence of the bexamir residing in the lumen of these vesicles, if that makes sense. So the first thing we did was to show that bexomeres are indeed expressed and released in extracellular vesicles. And here I'm showing you in these two lanes, two EV fractions, isolated EV fractions. As you can see, they're enriched in LAMP1 and CD81, but they are devoid in ER, Golgi, or cytoskeleton markers. And as you can see, we were able to detect the presence of 3 different bexomeres in these fractions, and we did that by RT-PCR. And as you can appreciate, the size of these microRNAs, it's about 65 base pairs, and this is because the way we perform these RT-PCR experiments, the end-up product has 65 base pairs instead of 22. But as you can see, in these isolated EV fractions, we were able to detect at least 3 different bexomeres. Not only that, further quantitative RT-PCR experiments of EV fractions that had been previously detergent solubilized and treated with RNAs, we could see a decrease in the abundance of one of these bexomeres, indicating that these reporters are actually packaged in exosomes or extracellular vesicles, which indeed floated at the right density in a discontinuous sucrose gradient. So we took this density and this fraction, and we were able to detect also the presence of a bexomere in these floated membranes. Okay, so this further suggests that bexomeres are not only released but also packaged into vesicles, extracellular vesicles. Finally, we treated cells with sRNAs to deplete RAP27A, which was previously shown by the groups of Clotilde Terry and others to regulate exosome release. And what we found is that RAP27A depleted cells showed decreased abundance of bexomere level in isolated extracellular vesicle fractions. So all of these results were done with a limited number of bexomeres. And in order to validate the use of these reporters as high throughput, in order to validate these tools as high throughput reporters of exosome release, we synthesized a library of thousands of these microRNAs, which were then cloned into an lentivirus vector that was used to infect cells in pool format. So all of these cells here in this cell culture, this is a suspension cell culture. Each one of the cells was expressing a single bexomere, and we accomplished that by infecting a very low multiplicity of infection, such that cells are only infected 0 times or once. We then isolated vesicles from these cultures and extracted the RNA associated to these vesicles and did deep sequencing to measure bexomere abundance in this extracellular vesicle fraction. And as you can see here, this is the results from 2 replicate cultures showing the abundance of 32,000 bexomeres. And as you can see, it's very reproducible. And one thing that it's very apparent that I'm sure you can see is that while some bexomeres were highly enriched in the extracellular vesicle fractions, others were barely detected. And because all these bexomeres only differ in the barcode sequence, these results indicate that intrinsic differences in the sequence of the barcode is probably driving such heterogeneity. So we're currently trying to collaborate with bioinformaticians, trying to see if there are specific base pairs or sequences or motifs in this barcode sequence that may be driving enrichment or disenrichment of these artificial microRNAs. So with all these previous data, we gain enough confidence to now couple our bexomere reporters with CRISPR-Cas9 technology to develop a genetic screening approach that would enable us to interrogate in a systematic fashion which genes are involved in the generation and release of extracellular vesicles. So for this, what we did was to synthesize again libraries oligonucleotide libraries consisting of pairwise associations of guide RNAs and vexomeres. So these libraries contain each gene was targeted by 10 different guide RNAs, okay? And each guide RNA was associated with a unique vexomere. So with this, we synthesized 250,000 sgRNA vexomere pairs, oligonucleotide pairs, to cover the whole genome. And this number also includes negative controls. The oligos were cloned then into a lentivirus vector, such that the guide RNA is driven by a different promoter than the vexomere. Then the lentiviral library was used to infect cells, again at low multiplicity of infection. And we infected both wild-type cells and Cas9 expressing cells. So in this scenario, you can imagine that if, for example, guide RNA X is targeting a gene important for extracellular vesicle release, what's going to happen is that when we measure the abundance of vexomere A in extracellular vesicles isolated from Cas9 positive cells, we're going to see a reduction of vexomere A in these vesicles compared to the abundance of vexomere A isolated from vesicles coming from the wild-type scenario. Okay. So we grew these cultures. We isolated extracellular vesicles from liters of culture supernatant. And when I say liters, we actually had to isolate extracellular vesicles from over 100 liters of cell culture in order to cover the whole genome in this genetic screen. Finally, we extracted the RNA and compared vexomere abundance, measuring by deep sequencing. So this genome-wide screen allowed us to identify known players. For example, the neutral sphingomyelinase was validated. Also, ALEX, HRS, the SNR protein YKG6, the calcium sensor protein synaptotactin 7, or the ion channel mucolipin 3 residing in endolysosomes. But also with this approach, we identified many new genes with previously unrecognized roles. And these include both activators and suppressors. So an activator, a gene that activates or positively regulates exosome release, when we depleted it in our screen, in that case, the vexomere abundance was decreased. And on the other hand, a suppressor is a gene that when deleted in our screen, increased the abundance of the vexomere measured by deep sequencing. So an initial screen focused on membrane trafficking mitochondrial motility genes already revealed several genes that have been previously proposed to regulate extracellular vesicle release, shown here in green. We then performed further subsequent genomic screens using this system and further validated other factors. But of course, as I said, we uncovered other genes with previously unrecognized roles. And one of these genes that we decided to focus was RK18. And this work that was done during my postdoctoral studies in the lab of Suzanne Pfeffer at Stanford University. Her lab is actually focused on small RAP GTPases. And for that reason, we decided to focus on this protein because RGF18 is actually a one exchange factor for raw small GTPases. In other words, RGF18 promotes exchange of GDP to GTP for raw proteins, and that in turn, raw proteins, when activated, can remodel the subcortical actin cytoskeleton. And as I said before, the remodeling of the subcortical actin cytoskeleton has been shown to regulate both microvesicle and exosome release. So in order to validate by orthogonal assays this finding in our screen, we performed a nanostring profiling assay. In this assay, we can measure the abundance of endogenous microRNAs with fluorescent probes. Okay, so here what we used was as a control, we used the neutral sphingomyelinase knockout cell line because this gene has previously been shown to regulate exosome release. And then we generated by CRISPR-Cas9 technology, we generated also an RGF-18 knockout cell line. And we compared the abundance of microRNAs, both in cells and EV fractions. We compared these two knockout cell lines to the control. So each one of these white dots is a single microRNA, and the levels are here on the y-axis. They're shown as % of control. So as you can see, while most microRNAs did not change in the cell fraction, the overall microRNA signature in the EV associated fraction was decreased in both the positive control, the SMPD3 knockout cell line, and RGF-18 knockout cell line, suggesting that RGF-18 is also regulating exosome release. Now, if we look at these 74 microRNAs that we were able to detect in the extracellular vesicle fraction, if we look at these microRNAs in the cell fraction, we can see that these changes observed in this fraction do not occur in cells, further suggesting that, or confirming that our findings indicate that RGF-18 is actually promoting exosome release and also microRNA enclosed in these vesicles. But nevertheless, this last essay did not actually allow us to determine exactly where RGF-18 is acting, because RGF-18 could be perhaps upregulating microvesicle release, or on the other hand, regulating exosome release. So in order to differentiate these two pathways, we borrowed an assay originally developed by Frederick Verwey and in Michael Pechtel's lab, and also Guillaume Banil. And in this assay, we use a fusion protein, CD63 pH fluorine. This is a pH-sensitive GFP mutant that fluoresces when in the presence of neutral pH. So as you know, CD63 is enriched in exosomes. So under lysosomal pH conditions, the reporter is quenched. There's no fluorescence. But when there's fusion of this compartment with the plasma membrane, exosomes become in contact with the extracellular media with neutral pH. And at this point, we can detect an increase in the fluorescence of this reporter. There's a burst in the fluorescence detection, as you can see. And we can measure with this assay fusion events. In other words, we can monitor exosome release. So here in this video, you will see that in these different circles, different fusion events. I'm not sure why. Oh, yeah. Right. Yes, that's what I thought. So as you will see, one event, another event here. One more, and finally, one more and another one. So this assay allows us to monitor in vivo exosome release. And so with this assay, we were able to, again, confirm that RGF-18 is actually involved in the regulation of exosome release. You can see that control cells display this number of fusion events. When we knock out RAP27, there's a reduction in the fusion events measured in the cells, and also happens when RGF-18 is depleted. But we also validated other hits that we obtained in the screen, including ROV, which is another Rho small GTPase, BAIAP3, which is a MONG13 family protein, and a subunit of protein phosphatase 1. With this assay, we're able to show that at least these different hits may be doing something in this process of exosome release. Okay, but of course, we identified many other genes here shown in red or blue colors, and color intensity and reflects the effect size, so the phenotype, whether they affected more or less exosome release. And the size of the circle corresponds to how significant was the hit in the screen analysis. One of the things that piqued our attention was the finding that there were a lot of hits involved in wind-related signaling and trafficking, as you can see here. And these involved both positive and negative regulators of this process of wind-related signaling and trafficking. Wind is a lipid-modified morphogen that regulates different developmental processes, but also has been involved in a variety of cancers. So in the absence of wind, the so-called beta-catenin destruction complex targets beta-catenin to proteasome destruction. Upon wind stimulation, wind binds Frizzled receptor and LARP5/6 co-receptors. And this leads to inactivation of the beta-catenin destruction complex, which finally ultimately results in accumulation of beta-catenin in the cytoplasm, which then translocates into the nucleus, acting as a co-transcriptional activator of wind target genes. So in order to explore the impact of wind signaling in the regulation of exosome release, we treated cells with recombinant wind or with a GSK3 inhibitor. Let me remind you that GSK3 plays an important role in the destruction of beta-catenin, such that if we inhibit GSK3, we are actually also indirectly activating the wind pathway. So with this, either treating cells with wind or inhibiting GSK3, what we observed was a reduction in different exosome-associated markers, LAMP1, HSC70, CD81, indicating that wind actually is also regulating exosome release. In this case, would be a negative regulation. Okay, so in summary, for this part of the talk, I showed you that artificial varicoded microRNAs, or as we call them, veximers, allow high throughput monitoring of EV release. Analysis of vexamer abundance coupled to CRISPR-Cas9 screening technology enabled the discovery of many genes involved in EV biology regulation, including known players. But also, we were able to uncover new hits or genes involved in this process at RGV18, which is another regulator of exosome-mediated microRNA release. And finally, with this approach or strategy, we also uncovered a role for wind signaling in the regulation of EV release. Okay, so now I'm going to move to the second part of my talk, and I'm going to be talking about KRAS and an RNA binding protein, RNPA2B1. So I think we're all aware that cancer is a genetic disease, and we get mutations in our genes that confer selection for growth. And in fact, one of the, or perhaps the most frequently mutated oncogene in humans is KRAS. There are about 18 million new cancer cases every year. And from these 18 million of patients, 2.5 million of these patients exhibit KRAS mutations. So KRAS is this small GTPase anchored to the plasma membrane, but also to other endomembranes. And it cycles from an inactive GDP-bound state to a GTP-bound state. So under normal conditions, growth factors bind to receptor tyrosine kinases, which lead to activation of KRAS. And activation of KRAS leads to binding and activation of downstream effectors, such as RAF, and the activation of many signaling pathways, including the well-known MAP kinase signaling pathway, which promotes cell proliferation. But as I said, there are many other signaling pathways that are regulated by RAS proteins. Our previous work showed that a pool of KRAS can be endocytosed via clathrin-coated pits and reach the endolysosomal compartment, from which KRAS can also activate the MAP kinase signaling pathway. Now, in order to downregulate this pathway, what we showed previously is that KRAS is then sorted into intraluminal vesicles, and once sorted, this signal is abrogated or finalized. Okay. Now, before I move on, I would like to say that KRAS has different, and other RAS isoforms have different downstream effectors. Here I'm showing RAF, but one of these downstream effectors is also HNRMP-A2B1, as we will see. Previous work by Sanchez Madrid showed that HNRMP, as I was telling you before, is a protein that once sumoylated is able to bind the 3' N of a subset of microRNAs that display or exhibit an exosome targeting motif. And this protein was proposed to regulate exosomal microRNA sorting into extracellular vesicles. Later work by our collaborator Neuza Jail at the University of Barcelona showed that HNRMP-A2B1 is actually a downstream effector of KRAS, and this was shown in pancreatic cancer cells. Okay, and then later the groups of James Payton and Bob Coffey at Vanderbilt University showed that the mutational status of KRAS dictates the microRNA signature of extracellular vesicles. So all of this previous evidence led us to propose that perhaps there is a role between KRAS and HNRMP controlling exosomal microRNA sorting. Okay, so in order to test this model, we have performed a series of experiments that I'm going to show you. So the first thing that we did was to confirm that mutant KRAS preferentially binds to HNRMP-A2B1. HNRMP-A2B1, the gene encodes for 2 different isoforms, the B1 and E2 isoforms. They differ just by this little stretch at the N-terminal portion of the protein. And as you can see, when we co-expressed the A2 or B1 isoforms in the presence of the oncogenic form, we get more binding, here shown in the last lane or here, compared to the wild type KRAS. So confirming that the oncogenic form of KRAS binds preferentially to this RNA binding protein. Not only that, we also observed that upon expression of the oncogenic form, there are these high molecular species of HNRMP proteins, both the A2 isoform, but also the B1 isoform, if we overexpose this gel, indicating or suggesting that that mutant KRAS is inducing some sort of post-translational modification on this protein. And this suggests that perhaps could be sumoylation, as previously proposed by the group of Sanchez Madrid. Okay. So then we further and went ahead and did more immunoprecipitation experiments to show that also because we know that different KRAS mutations exhibit different affinities for downstream effectors. And we wanted to test this model in colon cancer. And in colon cancer, the typical mutation that we find is the G13D. So we also confirmed that, that the G13D mutation binds just as well as the G12V mutation to HNRMP-A2B1. And again, we could see these putative post-translational modifications in HNRMP-A2B1, which we suspect it's a simulation. Immunofluorescence experiments in cells treated with bafilmomycin A1. In our previous work, we showed that bafilmomycin A1, which is an inhibitor of lysosomal function, KRAS is accumulated in this compartment, LAMP1 compartment. And in fact, when we did this, also we could also find HNRMP present in GFP KRAS decorated endolysosomes, as you can see in these 2 different examples, in this cell co-expressing these 2 proteins. And then we performed fractionation experiments and showed that in isolated EV fractions, when we express the mutant form of KRAS, you can observe that there is an increased recruitment of HNRMP in the EV fraction, as opposed to the wild type or a GFP negative control. As you can see, the oncogenic KRAS is more enriched in the EV fraction, and that also leads to an enrichment or an increased enrichment of HNRMP in this fraction, suggesting that KRAS is driving sorting of this protein into extracellular vesicles. This was also confirmed again with the colorectal cancer mutation, the G13D. As you can see, expression of this mutant increases the abundance of HNRMP A2B1 even more so than the G12V mutation. Here, the G12V mutation, the oncogenic KRAS containing the G12V mutation did not show an increased enrichment as opposed as the previous experiment that I just showed you. We don't know why in this case, but we have performed several times this experiment. In most cases, we consistently observed an increased abundance of oncogenic KRAS compared to the wild type scenario. So the next thing and the latest thing that we have done is to confirm the impact of the presence of or absence of HNRMP A2B1 in colorectal cancer cell lines. We used a cell line that contains one wild type allele of KRAS and another cell line that contains both the wild type and an oncogenic form. In fact, this cell line was generated by deleting the wild type form of KRAS. So this the DKS8 cells serve as negative control as opposed to DLD1 cells. So in these two backgrounds, we use CRISPR to delete HNRMP A2B1 such that we have the two cell lines that I'm just showing expressing or not expressing HNRMP A2B1. We then isolated EVs from both cell lines, extracted RNA, and performed a small RNA sequencing to measure and to analyze the differential expression of the different microRNAs in both cells and EV fractions. We did this in 3 replicates, and here are the results. The first thing that you can notice is that the cellular microRNA signature is very different from the EV-associated microRNA signatures. Each one of these rows is a single microRNA here on the right. Not only that, the mutant and the wild type KRAS cell lines also show substantial differences, as you can appreciate in this heat map. And when we express or do not express HNRMP A2B1, in this case, it's a little bit harder to see differences. But you can believe me if I tell you that we have found several candidate microRNAs that we are currently validating. So principal component analysis, again, showed what I just told you, very well different clusters of the data from cells compared to EVs, from both wild type or mutant cell lines expressing or not expressing HNRMP A2B1. And as I said, we are currently validating candidate microRNA hits that I apologize, I cannot show you today. So together, what we think is happening is that active KRAS, perhaps from endolysosomes, is activating binding HNRMP, is perhaps inducing simulation of HNRMP A2B1, perhaps activating it. Then it recruits it by binding onto endolysosomal membranes. And HNRMP A2B1, which binds a subset of microRNAs, is then incorporated into exosomes that are finally released. And so this would be a signaling axis regulating exosomal cargo composition that could have important implications in colorectal cancer. So together, mutant KRAS binds HNRMP A2B1 and induces a post-translational modification, likely to be simulation. Oncogenic KRAS colocalizes with HNRMP A2B1 on endolysosomes and drives its sorting into exosomes. Differential microRNA expression analysis revealed differences in the presence or absence of HNRMP A2B1 in both wild type and mutant KRAS cell lines. So our current follow-up studies are assessing the role of KRAS mediated simulation of HNRMP A2B1 we're validating, as I said, candidate microRNAs and exploring the impact of these microRNAs in acceptor cells upon transfer functional studies. So with that, I'm not going to go much further, but because we're going to be discussing this topic later today. I just wanted to mention perhaps that there have been a number of factors involved in the uptake that as Greg described earlier today. Also, the role of lipids was brought in during his talk, and as well as I fit them proteins. Finally, recent work by Serial's group showed that RNA can be perhaps sorted out of endosomes via tubulation. So this could be another pathway that perhaps is regulating microRNA delivery in cells. And as well, who knows if there are microRNA dedicated channels for microRNA export from endolysosomes or directly from exosomes. But again, this is something that we can discuss later today, as Alireza pointed out. So with that, I would like to thank our collaborators at Stanford University, Susan Pfeffer, Mike Basic, who helped us with all the CRISPR screens, and our group or several collaborators at the University of Barcelona, Paul Selke is a PhD student, and other PIs that are also contributing to this work, the latest KRAS work, as well as Francisco Sanchez Madrid, and also funding sources. And with that, I'll be happy to answer some questions. Thank you.
Thank you, Albert, for the fantastic talk. I think there may be more questions. Like you suggested, maybe we have to keep some of the questions for the evening session as well.
Sure.
So are there any questions from the audience?
So what's the quota? What's the number of question we allowed? One question?
Yes, please.
Okay. So related to beautiful work, by the way. So related to your finding on the requirements, what is the role of wind in uptake of these EVs? I mean, it's a bit surprising, right? Because I mean, you need
Release, right?
Release. Sorry. So in cancer cells, you actually have an increase of wind signaling. So
Yes.
then
So-
in that case, you would actually inhibit this release of these extracellular vesicles. So how do you reconcile that?
Right. I didn't show all the results that we published, but what we found is that wind signaling actually downregulates RAP27A expression, and that possibly explains why there is this decreased release. Now, what is the significance of inhibiting the release from a cancer cell type scenario? I mean, both the release or inhibit or the upregulation or blocking the release of extracellular vesicles may be related to the cargo, right? There could be cargo that could be perhaps favoring growth, or perhaps communicating through other cells associated to cancer, such as CAFs, cancer-associated fibroblasts. So perhaps wind is unregulating the release of some vesicles that could be, instead of favoring cancer growth, inhibiting it, such that for that reason, perhaps the idea here is to block the release of vesicles, or the other way around. I mean, the release or not, or the inhibition of the or promoting or inhibiting the release may be related to the cargo that EVs are containing, and the cargo may be favoring or not favoring cancer growth or metastasis or invasion. So.
Yeah. So and related to that, so in the last model that you showed with KRAS and HNRP, right?
HNRP, yes.
So once you recruit or you simulate HNRP, then you have KRAS that gets internalized in these vesicles for subsequent release.
Yes.
So if that was the case, then you would think that first in cancer cells, you will actually get rid of oncogenic KRAS from that cell, and therefore you attenuate signaling in those cells.
Oh, right.
So then how?
Right. Yeah, that's a good point. I mean, it depends. Sure, that is true, but perhaps the export of these vesicles have something that some microRNAs that could be further favoring growth. Or, I mean, I see your point, but the number of molecules that are being exported, I mean, we don't really know whether it's a big number of KRAS molecules or it is not. And whether that could have an impact in the overall RAS signaling, that remains open. We haven't measured KRAS signaling upon, let's say, upregulation of exosome release, because what you're trying to bring up is that perhaps the reduction of KRAS molecules is downregulating the overall KRAS signaling, right?
Yeah, so I think that would be the right experiment, right? So
Yeah.
do the experiment in the presence or absence of your SHNRP, and then look at ERK signaling,
Right.
right? Because then you would expect then in that case that your ERK signaling would go dramatically down, actually. And if it goes down beyond a certain threshold,
What we don't know is whether HNRNP is actually enhancing KRAS release, or it's the other way around, KRAS enhancing.
because if you have HNRP, then what will happen? You will take oncogenic KRAS and clear out of the cell. You said yourself, you're signaling from endosomes.
Right. Yes, but yeah, that we could test that. Yeah.
The paxomers that you spoke about them at first, have you ever looked at where are they in the exosomes? Are they in the as a cargo or inside, or are they located at the surface of the EVs?
Paxomers, we believe they're packaged in the lumen because our RNase protection assay with detergent solubilized fractions suggests that they are in the lumen.
But have you looked or have you tried to see, for example, doing any RNAs or any kind of things to see if they are really inside the...
With like in situ hybridization or something like that, or...
Something that somehow show where they
we haven't formally visualized them inside, and we only perform this RNase protection assay in the presence or absence of detergent. So that's the only experiment that would suggest they are packaged in the lumen.
Why the sequence of these paxomers can somehow determine their amount in the amount that they are? Why should that sequence have to do anything with the release of them from the cells?
It's probably via RNA binding proteins. We believe that the random sequences that we place there may be specific, like I showed, there may be specific sequences that drive a tremendous export by binding some RNA binding protein, because microRNAs are always usually constantly bind to RNA binding proteins. They're not traveling freely. They usually bind to some protein. So we think that this is probably driving there are proteins driving their export by binding to some sort of factor that we don't know. Yeah,
thank you. Very interesting.
Thank you.
So in the absence of any questions, so we have the pleasure of handing over to Sonia, class certificate. Right, so we have come to the tea break or the coffee break, I think. So the coffee and tea is ready outside, and please enjoy, and then make sure that you return back to your seats by 11:15. Thank you very much.
So I haven't been
Okay, welcome back. Before starting the formalities, I just want to inform that our Zoom link has been hacked, unfortunately. So we apologize from all of you and also for the people on Zoom. Our technical people are trying to figure it out what happened actually. So our apologies for that. Yeah. So next in line is Dr. Sasha Rashke, who has been a dear friend for us for a long time, working with our group for, I don't know, for like 5-6 years of time.
Since 2016.
Okay, since 2016, even before.
Yes.
Yeah, so he's not a stranger for us, but of course, just as a formality, I would like to introduce him. So he's coming from the company that produces ZetaView instrument. So he'll be talking about the ZetaView technology. So the floor is open for you, Dr. Sasha.
Okay, thank you very much. Thank you for the kind introduction, Alireza. Thank you very much for the kind invitation. So I would like to spend a couple of minutes to present you a little bit the ZetaView technology, more specified this introduction into the ZetaView nanoparticle tracking analyzer, NTA, nanoparticle tracking, especially for characterizing extracellular vesicles. So first of all, I would like to give you a picture for those of you who don't know the instrument or who don't know NTA at all. So this is the instrument. And what you can see is it's a quite small instrument. What you can see here is it consists of an injection port where you put in your samples, your highly diluted EV samples. You have also an opening for sample recovery, which means if you have a sample which is quite valuable for you, you can also recover the sample. And we have connection for the water and buffer, and everything what comes in here goes out here. This is the waste. A brief word regarding the ZetaView setup, the optical setup. So the instrument contains of, in the upper part, the laser. And this laser illuminates the sample. Here we have the cell channel cross section, and we have here the particles in it. At the lower part, we have the objective, which is combined with the video camera. And with this setup, we can measure the size of the EVs or of the particles. We can also measure the concentration, and if we apply an electric field, we can also measure the zeta potential, which means this is the surface charge of the particles. Furthermore, we can also distinguish subpopulations, and this is an important thing here with the fluorescence. We can also do if we slide in a fluorescence filter. I will come to that in a moment. So what we see if we switch on the instrument and we put in the sample, we see something like this. So these are the particles in the field of view. They are moving according to the Brownian motion, and this is what we see. What the instrument sees is something different. So if we look at one single one single particle here, it might move like this. Yeah. So it moves regarding on the basis of the Brownian motion. And what the software does is now it calculates or it detects and localizes all the particles here in the field of view. So every single particle is localized and detected. And the software also quantifies the mean square displacement per a certain time interval or per a certain frame of the video what we record. And thanks to Stokes and Einstein, we have here the relationship. We can then calculate from the Brownian motion. We can then calculate the radius or the diameter of the particles. So this is in principle the basics or the technique behind nanoparticle tracking analysis. So what can we do with it? Nanoparticle tracking analysis is not only for EVs. Of course, I would say 90% of NTA measurements or NTA machines are quite important in the bio nanoparticle field, like extracellular vesicles, also viruses or virus-like particles, and so on. But it can also be used for inorganic particles like polystyrene or quantum dots, nanometals, and so on. Nanobubbles is also a quite important field, not here in Europe, but more in Japan, and in the technical field also polymers. So, but I would like to focus definitely on the EVs. So the range, the size range between the size range in which we can measure our nanoparticles is for this instrument is 20 nanometers to 1000 nanometer, and of course everything should be measured in liquid, otherwise we cannot measure the Brownian motion. A unique technology with the theta view is that we can measure multiple positions throughout the measuring cell. So we have here the microscope and the laser. The laser illuminates a certain volume inside the measuring cell, and it can scan automatically up to 11 different measurement positions, and that gives us very robust statistics because we are measuring also far more particles than only having one single position. What we get then is a particle size distribution or a histogram, and that describes how many particles do we have of a certain size, and related to this also which particle of a certain size is assigned to a specific bin class, which is defined by the software, or which can be defined by yourself in the software. So on the basis of what I told you, now the question arises: do you really know if you inject your particles, your EVs in the theta view, and you take or you get a size histogram? Do you really know what's in your sample? And the answer is no. And I'll tell you why. Because what what I told you now is just the scatter-based nanoparticle tracking analysis. And so again, let's look at this picture here. We see some particles here in the field of view, but you can not be sure if there's really a nanobubble, or if it's an exosome or extracellular vesicle, is it maybe a protein aggregate, or just a insoluble salt precipitate, or something like this. So you simply do not know, and this is why we need to be a little bit more specific. And the specificity is, for example, done with fluorescent labeling or fluorescence-based nanoparticle tracking. So we here an extracellular vesicle, and what we can do is, for example, we measure them labeled or in a labeled condition. So we have a fluorescence dye, for example, a membrane dye. And what we are using currently is the dyes from the cell mask series, cell mask green, orange, or cell mask deep red. These are membrane dyes or lipid dyes, and they integrate in the membrane. And then if you measure the particles first in the scatter mode and then in the fluorescence mode, then you can see here, this is our total particle count here in the scatter mode. So these are all the particles what you have here in the sample. But in this example here, you have only 9.2% of fluorescently labeled particles. So these are the membranous particles here. Yeah. And this is how it looks like inside the instrument. So there's a quick switch between the scatter mode and the fluorescence mode. Yeah. So the question here is, are we done? Or can we do further can we do a more specific measurement or experiment? And yes, we can do. And this is shown here. So this is an example of a previous experiment. What I did. So these are two single stainings of MSC EVs. And I stained it first one is stained with the anti-CD9 with the APC fluorophore, and the second one is CD41 Alexa fluorophorate 8. Again, you can see here, this is the total particle count here, and we have only 5 and around 10%, 9% of the really fluorescently labeled particles. And so the rest of the remaining particles here is something else. Yeah, for example, some protein aggregates or lipoproteins or whatever. So which means scatter-based nanoparticle tracking is definitely, I would not say it is outdated, but it is not enough to characterize your EV samples by using nanoparticle tracking analysis. So if we go one step further, the latest version of the ZetaView instrument can also do colocalization. And this is exactly the same technology what we can also do with the normal fluorescence. So and I think I would like to show you, yeah, again the video. So we have a quick switch between different fluorescence filters, and this is also indicated here by the software. So it switches from fluoride 8 to the 640 nanometer laser, and of course the appropriate filter. And what we can get, what we get then is something different. We get then a scatter plot with detection in the green fluorescence and in the detection in the red fluorescence channel. And all the colocalization events, what we measure so far then is then indicated by cross. So this is in principle how the colocalization NTA works. And of course, there are some precondition to do it. For example, if you have first you need to establish the single staining for example here the first fluorescence dye, then the single staining for the second fluorescence dye, or maybe also the antibodies. And once you establish this, then you can put them together. But you need to remember, or you need to maintain the ratios of the fluorescence dyes or the antibodies. And once you have done it, then you get something, yeah, what I already showed you here. And this is also one proof that colocalization NTA works, and it is quite comparable to, for example, the imaging flow cytometry. Yeah, so if here again the detections in the green and the red fluorescence channel, and also the colocalization, and if we compare it here, we have a good agreement by 58% of double positive in the imaging flow cytometry, and 58.5 with the colocalization NTA. If we go further with detergent control, which this is also quite important, then we have a similar picture here. Colocalization ratio here, we end up here at 4%, and with the imaging flow cytometry with the same experiment, we end up here with 5%. So colocalization NTA is, if you compare it with the imaging flow cytometer, is quite comparable, and this is for us, of course, a really good indication, because when we did the experiment the first time, we didn't know if it is really so reproducible and so comparable with another technique. We also did a triple labeling. This is at the moment, this is our let's say most comprehensive experiment. So we did a triple labeling of with CD41, CD9, and CMDR. CMDR is a membrane dye, and you also see here that we have a really good colocalization with CD9 Pacific Blue and CD41 Alexa Fluor and all the others. And if you look at the detergent control, this is also quite comparable, and also the results are quite expected. If we go to a triple labeling, we also need to consider that there are many staining sequences. So if we have more than 2 antibodies or 2 fluorescence dyes in general, we can do many sequences, like for example both antibodies together, and let them incubate, and then add the membrane dye or the other sequences. And the good thing here is that the sequence is, or the labeling sequence is not really affected, or in other words, the results are quite comparable with the staining sequences, which is quite good. So it doesn't matter which staining sequence you perform. The colocalization ratio is almost the same, and this is also a good indication. So as a summary, I would like to, or as a take-home message, I would like to tell you that NTA. I think you also know it. NTA characterization, or NTA is a characterization method on a single particle level. We have a really robust statistic due to the automatic 11 position scan. we can differentiate between scatter and fluorescence mode, where the scatter mode is, of course, quite unspecific. We have the total particle count in the scatter mode, and in the fluorescence mode, we can phenotype and characterize the EVs, also EV subpopulations. The good thing here is, of course, also we can use commercially available fluorophores or antibodies. The colocalization is also possible with NTA via the quick detection of surface markers, also in one single run. So this is what I wanted to inform you about quite briefly. Unfortunately, I cannot do it until the end of this session. I need to leave quite early. If you have any questions, I'm happy to answer them right now. Or if you have any, if any questions arises later, you can email me. This is my email address, rashke@particlematrix.de, or you can email at the company, info@particlematrix.de. Thank you very much for your attention.
Thank you, Sasha. Are there any questions? I think already there are two questions. Who's first? You?
Thank you so much for such a wonderful talk. I have a very small question. I don't quite understand what is the objective of colocalization.
What the objective is. Since we have several lasers and several filters, we can do colocalization, and it is if you would like to phenotype your EVs, and you would like to know, okay, if do the EVs have a certain or maybe 2 or 3 certain surface markers? You can do it in a single run, which means you are much faster than just having one experiment with one label, second experiment with a second label, and so on. Yeah, so it's just a quicker experiment. Yeah. Yeah, go ahead.
Yeah, it's much more than just a quick experiment. At the moment, our knowledge about EVs, heterogeneity of them, and so we are like a child in a meadow. So we don't know anything about how many different kind of proteins, molecules, and so are on the surface of these EVs, or how they are packaged, how the cells are ready. Albert was showing that 10 to the power of 16 different EVs in our bodies. God knows how they are arranged, the different molecules that they are in. Technologies like this would allow us to be able to see that what are the different antigens, proteins, different things that is going. And now it is, for example, too, I hope a day comes that, like another time Albert was showing, we would be able to see 1,000 different molecules on each EV and be able to somehow characterize them based on that. Sorry.
Yeah. So coming from a person working with horses, the antibodies you're using, do you know which species you can use them? In which species you can use this?
In which species?
Yes.
Yeah.
From the vesicle of what?
Yeah. Currently, so my personal experience with my experiments, it was only human. Yeah. So currently, I didn't try anything in plant EVs or animal EVs, but that's a good point, by the way. I will try it in the future.
So if there are no other questions, shall we thank Rashke again, and then we have a small Sonia for you as well, because of the organizers asked me to come over here to see them. And you'll be looking good in your
Yes, of course. Yeah.
Okay, then our next speaker is Dr. Saul Meiki. She's the group leader of Technical University Darmstadt, Germany. Today she is going to deliver the talk on the topic the difference functionalities of a small EV-derived MIR in PGE2 dependent tumor correlation with the tetraspanin composition of small EVs. So it's going to be a virtual discussion. So the floor is yours. Please start, Dr. Meiki.
Yeah, thank you very much for the kind introduction. I hope you see my slides. Unfortunately, I was not able to come in person, but I'm happy to have the honor to present our recent findings now via Zoom. Now, our group is interested in understanding how inflammatory processes in cancer are regulated by lipid mediators and microarrays. And one of our key interests is the regulation of prostaglandin E2, or in short PGE2, which is a lipid mediator, bioactive lipid mediator, which has a wide range of biological effects in cancer and in chronic inflammation. And here you see the biosynthesis of PGE2. In the first step, you have the release of the chorionic acid from a cellular membrane, and it's converted by cyclooxygenases to PGH2, and PGH2 is a pre-compound for all prostanoids. And specifically, the microsomal prostaglandin E synthase 1 is synthesizing PGE2, and PGE2 is released by cancer cells, for example, and it has an autocrine and a paracrine function, so it binds to its receptor, EP1 and to EP4, and then it's inducing proliferation, angiogenesis, metastasis, or even evasion. And when we started our projects, we ask ourselves how microRNAs can regulate prostaglandin E biosynthesis. Now, what is a microRNA? MicroRNA is a tiny little RNA molecules, which has a huge impact on gene regulation. They have a size between 20 to 23 nucleotides, and they were discovered 30 years ago in C. elegans. Now, over the last 10 to 15 years, comes up that microRNAs has different functionalities. The best characterized functionalities are the so-called canonical microRNA functions. So microRNAs can bind in a sequence-specific manner to their target mRNA, and then they can induce translational repression or mRNA degradation. But there are also non-canonical microRNA functions. Known, for example, microRNAs can directly interact with RNA binding proteins, which means that the RNA binding protein cannot bind anymore to the target mRNA. So these microRNA act as a direct antagonist for the proteins, and so the gene expression can be induced. Now, what we identified is that that microRNA 5745P regulates specifically MPGS1 expression in lung cancer. You remember MPGS1 was an enzyme which is responsible for the PGE2 biosynthesis. And this microRNA interacts with an RNA binding protein, and so-called COTP1, and so it's inducing MPGS1 expression and PGE2 biosynthesis, which leads to an increase in inflammation and pain. And we could show that very briefly that this microRNA 5745P is upregulated in lung tumor. And in vivo, we could show that the microRNA 5745P, if we enrich this, increase significantly the tumor growth, and this is due to PGE2 biosynthesis regulation. Now, over the last years, it comes up that there's also a second non-canonical function. You find microRNAs in exosomes or extracellular vesicles, and then they can bind to the immune receptor, toll-like receptor 7 and 8, and then they can induce cytokinases. Now, what we could show was that the same microRNA, the microRNA 5745P, when you find them in extracellular vesicles, then they can this microRNA can act as a Toll-like receptor 7 and 8 ligand, and induce, in this case, was in rheumatoid arthritis patients. You find this microRNA highly enriched in the synovium, and then it's leading or increasing osteoclast differentiation. So here you see the sequence of the microRNA, and you could see that it's highly homolog to the well-known Toll-like receptor ligand RNA-33. And we could see that this microRNA binds with a high affinity here to Toll-like receptor 8. And then we performed some functional analysis with this microRNA where we enrich the level of the microRNA within the vesicles, and then we could show that this microRNA induces osteoclast differentiation via Toll-like receptor 7 and 8. Now, we have two different non-canonical microRNA functions of the same microRNA. And when we started this project, which I'd like to present now, is does this extracellular microRNA 5745P has any impact on PGE2 biosynthesis in lung cancer? As you know, the tumor does not consist only of tumor cells, but you find also immune cells such as macrophages, but also fibroblasts you find. And the intercellular communication of the tumor cells and the stromal cells are very important for the tumor progression. In principle, this is actually my opinion, but I hope you agree with me that better understanding how tumor cells can communicate with their environment is really essential to discover new cancer therapies. Now, in general, lung cancer is the leading cause of cancer-related death worldwide, and prostaglandin E2 is very strongly upregulated in lung tumors and increase tumor progression. Now, you agree that small extracellular vesicles are a new part of the cellular communication, and as you're an expert, I will jump up over this introduction about extracellular vesicles, but these vesicles are very important in the tumor progression, so they can trigger diverse ranges of biological processes. And here's live cell microscopy pictures of our lab, where we have stained in green the vesicles, which we isolated, and then you see that here in the sex cells, that the uptake of the vesicles are very fast, between 15 to 30 minutes. Now, non-small cell lung cancer is a major form of lung cancer, and it has divided in different subpopulations. And the major subpopulations is adenocarcinoma and squamous cell carcinoma. In the first step in our study, we wanted to analyze whether the microRNA really co-localize with MPGS1 or with CUG1. And just very briefly, this microRNA really nicely co-localize within the cancer cell with MPGS1 and with CUG1, and this is in adenocarcinoma as well as in squamous cell carcinoma. Even in the necrotic area, we find a very nice co-localization. Now, the next question was, does PGE2 induce secretion of some microRNAs into the vesicles in human lung cancer? What we have done was very simple. We have chosen randomly lung cancer-related microRNAs. Of course, also we included the microRNA 5745P. And then we have chosen two different NLCSC cell lines, I509 and 2106T cell line, which represent the adenocarcinoma and the squamous cell carcinoma. And then we treated the cells with PGE2, and then we measured via qPCR the amount of the microRNAs within the vesicles. And what we could see is that only the microRNA 5745P was significantly upregulated in within the vesicles after PGE2 stimulation, and without altering the amount of the particles. When we treated the cells with the MPGS1 inhibitor, we were able to reduce significantly the secretion of the microRNA within the vesicles. And in total, we could say that we firstly identified that PGE2 is a stimulus for microRNA secretion, in particular the microRNA 574, into the vesicles. And this is mediated by its receptor EP1 and EP3. And in EV research, I think the characterization of the vesicle population is very important. And to that, currently we are using the ExoView R100 platform for characterization for the EV populations. In principle, we are able to bound the vesicles directly from the supernatant of the cells to chips via antibodies against surface markers such as tetraspannins. We are able to measure the vesicle size via light scattering, and then we are able to perform an additional staining, immunostaining, to visualize, analyze the surface markers or even the content of the vesicles. And then we are able to do colocalization staining of 3 different surface markers on one single SEV level. Now, when we, of course, we analyzed and characterized the A509 and 2106T derived SEV population. Firstly, we are the size, but we don't see any differences in the SEV vesicle populations, size populations. But when we take a look on the colocalization, we see similarities. When we take a look on CD81 and CD9 positive vesicles, we see very nice similarities. When we take a look on the CD63 positive vesicles, we see here differences in the tetraspanning composition. And this might have also an impact on the function of the vesicles. We will see. To that point, what we can say is and conclude is that the 2 different SEV populations differ in their tetraspanning composition. But what is the physiological function? Do we see differences between them? And what we have done was we used a certain plasmid, express plasmid, and we transfected the cancer cells with this plasmid, and then after 24 hours, we collected the vesicles with an engineered microRNA amount, or it's scrambled controlled SEV vesicles, and then we added those vesicles to the EV target cells. Of course, we characterized them. The vesicles, first, we were really able to enrich the amount of the microRNA within the vesicles significantly. And we performed also a treatment X and RNase treatment assay to show that really the majority of the microRNA is really within the vesicles and not outside attached. Next, we ask ourselves whether the cells are able to take up the engineered vesicles. For that, we perform live cell imaging assays. And first, we treated the 2106T cell with its engineered vesicles, and we really see that after 15 to 30 minutes, we see that the cells really take up the vesicles. Interestingly, we see only in 2106T cells an accumulation of the vesicles at the membrane. In the case of the A509 cells, we see that the A509 cells are also able to take up the engineered vesicle population, but we don't see the membrane accumulation as we have seen here. But what happened when we do the opposite round is that our 2106T cells are able to internalize the A509 populations. And in this case, we can say no, they're not able to take up the vesicles from the A509 cells, But when we take a look on the A509 cells, we see that actually those cells are able to take up to 106T vesicles on a lower uptake rate, but they are able to internalize those vesicles. So in total, we can say that there is a cell-specific SEV uptake by both cell lines. And of course, coming up to the major question for this project was that does the SEV-derived microRNA has really an impact on the PGE2 biosynthesis in lung cancer cells? And to make a long story short, yes, this microRNA has impact, but only in the A509 cells, not in the 2106T cells. You see here that in response to the extracellular microRNA, the MPGS1 expression was significantly decreased, and this was mediated by toll-like receptor 7 and 8 signaling. The same results we see when we measured the MPGS1 enzymatic product, the PGE2 level. So to summarize our findings from this project was that intracellular, this microRNA interacts with CO2-1, and this induces the PGE2 biosynthesis. And PGE2 in turn binds to its receptor and triggers the secretion of this microRNA into the vesicles, which in turn binds to toll-like receptor 7 and 8, leads to a decreasing of the MPGS1 and PGE2. We have here very nicely feedback regulation of PGE2 biosynthesis in lung cancer. And I would like to point out now that the intracellular function of one microRNA can be completely different compared to the extracellular function of the microRNA. And then we started another project in a neuroblastoma. The idea of this project was that, okay, we have now very nicely characterized the function of one particular microRNA on PGE2 biosynthesis in one tumor type. But PGE2 plays a major role in different tumor types, such as colorectal cancer, prostate cancer, breast cancer, but also in neuroblastoma. Now, neuroblastoma is a disease which cancer cells form from the neuroblast, for example, in neck, chest, adenoid glands, or spinal cord. Neuroblastoma mostly begins in infancy, so you find in by around children at 1 year or younger. And neuroblastoma is very interesting. It's unique among all other human cancers because it has the characteristic to undergo spontaneous regression or to differentiate to the beginning to ganglion neuroma. And as I've mentioned before, neuroblastoma is PGE2 dependent tumor type. And compared to lung cancer, where the cellular where PGE2 is formed by the cancer cells, here in neuroblastoma, we have the fibroblast as a cellular source for PGE2 biosynthesis. This makes it so interesting to understand the function or to compare the function of the microRNA 5745P in neuroblastoma compared to lung cancer. And at first, we performed co-localization studies in a subtype of neuroblastoma, in 11q deleted neuroblastoma. And we see here nicely that the MPGS1 is expressed in the fibroblast, but also interesting in starting to differentiate neuroblastoma cells. Here, as you see here in the blue dots here in the big cells here, for example. Now that was interesting. And one of the first steps was, okay, does PGE2 induce the secretion of the microRNA into the vesicles? And for that, we used an SKNS cells, which is 11q or in a neuroblastoma cell line representing harboring the 11q deletion. And what we could see is that, yes, PGE2 is also able to induce the microRNA secretion into the vesicles. When the particle count number was not altered by PGE2 biostimulation, and it's also like comparable to the lung cancer cells, this effect was mediated by EP1 and EP3 receptors. And of course, we started to characterize the SEV populations. In this case, now for the functional analysis, we compared it with another neuroblastoma cell line, SKNSH, which does not have this 11q deletion. Okay. And what we see here, the vesicle size are nearly the same, but when we take a look on the colocalization, we see here as well similarities, but also differences, as we see here in the CD81 positive SEVs or CD63. So as well as, yeah. Then we started to engineer the vesicles of the both neuroblastoma cell lines. And also here we were able to enrich the level of the microRNA significantly within the vesicles, as you see here. And also we performed tRNA and RNase treatment assay just to ensure that this microRNA is really within, or the majority of the microRNA is really within the vesicles, not outside attached. And then, of course, the first experiment was to see whether this microRNA has an impact on PGE2 biosynthesis in neuroblastoma. And what we can say is, here an example from SKNS cells, no, it does not have any impact on MPGS1 expression, as well as on PGE2 level. But when we take a look on the fibroblast differentiation, we could see that it has an impact and significantly increase the alpha SMA level of within fibroblast, and leading to an increase in the fibroblast differentiation. When we also used the other engineered other cell lines within engineered vesicles, for example, lung cancer cells or the SKNSH cells, and we don't see any effect, even a significantly reduction of the fibroblast differentiation, or here in this case, alpha SMH level. Interestingly, this effect was mediated by toll-like receptor 7 and 8. Okay, that is the same mode of action. And here you see a live cell microscopy picture from fibroblast, where we could see here very nicely on the active side, more or less, the fibroblast and the vesicles are taken up. Now, we see in our previous project and also in this that vesicles or the characteristics of the SEVs seems have an influence on the function of the microRNA, or in principle of the cargo. And what we have done was we inhibited the function of the tetraspan in CD9, CD63, and CD81 with antibodies. Then we wanted to see whether we are able to reduce the functional effect of the microRNA 5745P on neuroblastoma, here in the case of the alpha SMH level, A level, or in lung cancer when we take a look on the MPGS1 level. And what we can say is, yes, we're able to inhibit significantly the effect of the microRNA on alpha SMA when we block CD63 or CD81. And here we see here and the CD63 when we take a look on the effect of the microRNA 5745P. So the composition, we could show here that of the sulfate of the vesicles might influence the functionality of the SEV-derived microRNA. Then we wanted to elucidate whether the uptake rate might be different or changed. And in total, what you can see in two different methods, and lutein-based assay or with the microscopy, we're not able to see that the uptake rate differ and changed when we inhibited the function of the CD9, CD63, or CD81. And in total, I would like to point out that the blocking of tetraspinans and SEV envelope modulate the function of a cargo, and here in this case of the microRNA 5745P, but without altering the vesicle uptake rate. And finally, I would like to give an overview about what we identified here, is that the microRNA 5745P has multiple functions in neuroblastoma microenvironment. As in lung cancer, microRNA 5745P really interacts with CUT1 and induce PGE2 biosynthesis. And PGE2 in turn binds to EP1 and EP3 to induce the secretion of the microRNA of any neuroblastoma-derived vesicles. But here we have not an output function, but a paraffin function. So it's inducing pathoplasm differentiation. And with this, I would like to thank my whole group, especially here Anna, Julia Donzelli and Eva Fröstler. In this picture, we performed the experiments which I have presented today. All collaborators, and thanks to you for your kind attention. Thank you very much.
Thank you for the nice talk. Then let's turn to the audience if you have any question.
Thanks for the beautiful talk. So this is related to the concept that miR-574 is acting as a ligand for TLRs.
Mm-hmm.
So TLRs would be at the surface with the extracellular domain. I mean, the signaling bit, the intracellular domain would be in a cytosol. And then the ligand binding motif, obviously, extracellularly. And then, so these EVs that are delivering 574, I think conceptually I'm having a hard time to understand how these microRNAs encapsulated in an EV are going to activate TLR from, even if, unless you were talking about a membrane fusion event, and then these microRNAs are now in a cytosol. But even then it's confusing because the ligand binding domain is on the outside.
No, I just want to highlight that the toll-like receptor 7 and 8 is not on the cellular membrane, but in the endosome. You find them in the endosome. When you and it's known that it's recognized single-stranded RNA from virus. So if a virus or a vesicle, whatever, is internalized by the cell via endocytosis, it's entered and it can interact with toll-like receptor 7 and 8. This is special for toll-like receptor 7 and 8. I think 9 is also located in endosomes. I think other toll-like receptors, I agree, are on the cellular surface. But in this case, we have a lot of other cellular localization, which mediates, and it's possible, makes it possible that the microRNA interacts with the toll-like receptor 7 and 8. But it's specific for 7 and 8 and not for the other.
All right, great. Thank you.
You're welcome.
Thank you very much for this nice presentation. I think your experiment is a little more related to my PhD project. I'm also working the same project, but I'm working about STAT5, which is when it's activated, one of the target gene of STAT5 is prostaglandin or cyclooxygenase 2. I have one comment on your when you talked about the uptake of exosome by A59. According to my experience, I find that after 2 hours, A59 can uptake exosome and it reach peak after 4 hours. So I find in your pictures, you just look at after 30 minutes, I think, or have you ever tried after 2 hours or longer time to see the uptake of EVs?
Yes, we continued. I mean, the 30 minutes, we continue for 6 hours in total, but we recognize with the A509 cells that, or in other, that the majority of the vesicles are taken up in the earliest phase. So in our case, one hour, it's enough to take up all the vesicles in the majority. Yeah, that is what we found out.
Yes, thank you.
You're welcome.
Thank you very much. It was really interesting to see. And you're completely right. TLR 7, 8, 9 is endosomal. TLR 3 is also endosomal.
Yeah.
That is going to be also, yeah, it's inside the cell. All the ones that are dealing with the viruses and viral ones or RNA, DNA based. But continuing on the last question, have you also looked at the number of the EVs needed to initiate this or to do this? I mean, number of the EVs compared to the cell. How many EVs do you know, for example, Peter Wader is not here. He uses, God knows, 10 million, 100 million EVs for one cell to get the reaction. How is in your system? How many EVs do you need to get a reaction going or see a functionality?
Yeah, we never really counted the vesicles in because, as you may also know, is the analytics for vesicles, how many vesicles do you have or whatever. It's also a project we can discuss, which is the best one. Actually, we want to do that. One of our next projects is we are using super resolution microscopy to really analyze and to see how it's taken up. That is actually a currently planned project to do that in a good way. Yeah, to ensure a good quantification, whatever. Yeah, that's a good idea in what we want to do. Yeah.
Thank you.
You're welcome.
Okay, if no more question, let's move forward. Dr. Meiki, thank you so much for sharing your knowledge with us. Our next speaker is Dr. Julie Gavard. She's the CNRS Research Director of Cancer Research Center, France. She's going to deliver a talk on role of extracellular vesicles in the dialogue between endothelial cells and tumor cells in glioblastoma. So please, it's floor is yours now.
Introduction. Thank you very much for the invitation. It's a pleasure to be here in Tartu, and I'm going to present some of our data in my team, signaling in oncogenesis, angiogenesis, and permeability (SOAP) in the Cancer and Immunology Research Center in Nantes. And for those that might not know, Nantes is the 6th biggest city in France in the western part. I'm losing the pointer. Never mind. Okay, in the western part of France, which is a lovely place, let's say, for food, wine, also nice landscapes, and we even have a giant elephant. But more interestingly, maybe, this is also a very good place for an highly dynamic place to work on extracellular vesicles, because we have the chance to have a regional network, which is a sort of an open lab where we share tips, equipment, information on extracellular vesicles. It's almost more than 100 members of the community which is kind of half of a third of the national community on EV. And this is our institute downtown by the river and embedded in the hospital just behind. So let's go back to study extracellular vesicles in brain tumors, and hope there's some Harry Potter fans in the room, because I feel we need a bit of magic when we deal with brain tumors, and especially glioblastoma. So as you may know, glioblastoma are highly aggressive tumors of the central nervous system with a poor prognosis as the median survival range between 12 to 50 months. And the current treatments that didn't change since over 20 years include surgery when possible, followed by a concomitant chemotherapy with the alkylating agent temolizumab and radiotherapy. And despite these harsh treatments on patients, relapse is almost inevitable and fatal within a short window of time of 7 to 10 months. And like in many other cancers, there's evidence and growing evidence of the participation of a subpopulation of malignant cells with stem-like properties and called cancer stem cells, and in the case of glioblastoma stem-like cells or GSCs. And these cells are then suspected to be involved in the initiation, recurrence, and resistance of the tumor. They are similar to adult neural stem cells, able to self-renew, to differentiate in the CNS lineage, as well as in the perivascular lineage. But as malignant cells, they're resistant to most of the treatment, and they can induce tumors when implanted in immunocompromised animals in series and recapitulate the tumor ontogeny from the patient. But one interesting thing as well for GSC is that in the brain tumors, they encounter at least 3 different microenvironments. The central necrotic core, which is a highly challenging metabolic environment. The perivascular niche, where GSCs, as you can appreciate on this image, here in green, GSCs identified by the nestin markers are in close interaction with blood vessels. This is a more confined and protective habitat for GSCs. And the last environment is the so-called leading edge, where GSC will be in interaction with healthy brain cells. And it has been shown that they can actually use white mitotracts or blood vessels to invade the brain parenchyma. But in our lab, we are more interested in the zone where GSCs and are in functional interaction with the brain endothelial cells. And to tackle these questions, we set up first an ex vivo model by co-culturing patient-derived glioblastoma cells that retain some stem-like properties in vitro. They express some stemness markers. They can grow as 3D, although this is more a pancake than spheroids. And in the absence of serum, in a specific cocktail of mitogens. They're also able to differentiate in the CNS lineage, and as malignant cells, they also resist to treatment, especially to the temolizumab, and they can induce tumor formation in animals. On the other side, we culture normal human brain endothelial cells that recapitulate some features of the blood-brain barrier, such as junctions and expression of specific transporters. It's well known that glioblastoma vessels are highly permeable, and with this ex vivo system, earlier work from a lab showed that the conditioned media from GSC can induce permeability in vitro. And we also characterize some of the factors involved in this process, such as the islet chemokine, the growth factor VEGF-A, vascular endothelial growth factor, and the guidance molecule semaphorin 3A. But then we wanted to know more on the modus operandi deployed by this GSC to release these pro-permeability factors, and got interested in the extracellular vesicles. So the first thing that we did was to try to set up a good way to isolate EV and characterize EV from this condition media. This protocol has evolved over the years and will probably continue to be improved. But the first step is notably the expansion of patient cells, which is kind of tricky in our case because we have slow growing cells without serum. Culturing cells without serum is an advantage when we talk about EV, and we end up with 30 million of cells to be a good amount of cells to then isolate EV. And then we can culture them for 24 hours to 48 hours to get 100 ml. We're not in liters like Albert, but this is already a good way then to isolate EV by low-speed centrifugation and filtration to end up with a concentrated condition media of less than 1 ml. And you can imagine that in that case, we might expect to concentrate EV, but also proteins that were solubles in the medium. So the first step then to isolate EV enriched fraction was to use the size exclusion chromatography that will exclude most, not all, but most of the soluble proteins, followed by a sucrose gradient, and then end up with the high-speed pellet. And this is the typical images that you can get with CD9 and Alix fraction, enriched fraction, no GM13 cellular contamination, sorry. And then we can measure size and concentration of vesicles either by the TRPS or with the Q-nano or with interferometry with the Myriad. And time to time, we try to do electron microscopy, although we are not the best at that in our platforms. So with that, we then ask whether this GSC-derived extracellular vesicles can communicate with brain endothelial cells. And we label our EVs with lipid fluorescent probes, either the PKH or the membrane. And we saw that indeed brain endothelial cells can detect this fluorophore in a quite rapid manner and in a temperature-dependent effect. Interestingly, these EVs still have some biological activities that they can induce sprouting of brain endothelial cells in 3D, as you can appreciate here, as compared to positive control and negative control here. So they are still active in promoting change in the endothelial cells. This is the quantification of these images. So then what happened in terms of permeability? And we observed that, I lost again the pointer, I'm sorry, I'm pretty bad with that. So we observed that EV derived from different patient cells induced permeability in vitro on brain endothelial cells, and was true also in vivo in terms of vascular leakage. Then with an antibody array for angiogenic factors, we start to screen what kind of factors are present in this EV fraction from GSC. And beside metalloproteases, we found that VEGF here was present in this extracellular vesicles. This was confirmed with an ELISA on EV from patient cells or from plasma of newly diagnosed patient, although the concentration is quite variable between patients. Then we wanted to know whether this VEGF associated with EV can be the mediator of the permeability effect we saw right here. So what we did was to block the VEGF signaling with two tyrosine kinase inhibitors, semaxinib and sunitinib, that are used in clinics. And as a matter of fact, when we blocked the signaling, the permeability effect of EV is blunt. So to get a bit more specific on VEGF signaling, we silence VEGF-A in donor cells, the GSCs, and collect VEGF-depleted EV to feed them endothelial cells. This is the control for the ELISA, for the VEGF depletion. And now here on the permeability, you can see that this VEGF depleted EV are less able to induce permeability. And same was true when we look at the effect on the sprouting of endothelial cells. So this first piece of work showed that not only cytokines are really soluble, but are also probably associated with vesicles. So that was true for VEGF-A. So then what about the guidance molecule semaphorin 3A? And similar to VEGF, we found that semaphorin 3A was associated with EV fraction from patient cells and from the plasma of glioblastoma patient. We then revisit the silencing experiment that we did with the VEGF, but this time knocking down semaphorin 3A. And again, the permeability effect was reduced in vitro and in vivo. So suggesting that at least these two factors can work together for to convey permeability effect on endothelial cells. So then the next step was to see whether this was also true in vivo, in animal models. And for that, we prepare a protocol to isolate EV from xenograft animals. And we are preparing a STAR protocol that will be released soon. And what we did was to implant human patient cells in immunocompromised animals, either intracranially or ectopically, subcutaneously, and collect peripheral blood, which is the tricky and limiting step, the amount of blood that we can get from mice, and isolate EV with the SEC method, and then ultracentrifugation density gradient, to be honest, is quite difficult with this amount of blood in this condition. Nevertheless, we were able to isolate EV, and one interesting things with this model is that we can discriminate the EV from the host with mouse antigen for CD63, and this is what you can see here. There were no much or obvious change in the signal for mouse CD63, whether the mice were tumor-free or bearing tumors in the brain or subcutaneously. But interestingly, when we use a CD63 antibody recognizing the human form, so presumably coming from the graft and of tumor origin, we do see in significant level from 3 to 20% of circulating EV from human origin. And this was also the case when we measure semaphorin 3A by ELISA in the same fractions. So with that, we showed that GSC release cytokines, solubles, but also associated with extracellular vesicles that can convey message to the human brain endothelial cells. And we saw similarities between the effect of VEGF and semaphorin 3A. And one interesting point is that although they have different mechanisms of production, release, and signaling, they still share one co-receptor, which is neuropilin-1, and this is something that we are currently investigating. But as many others in the field, we try to understand better whether this EV can also somehow contribute to resistance of the tumor, or also can be used as disease markers. And this is something that we start first in vitro. As you may remember from the introduction, these GSC are mainly resistant to temolizumab, the standard of care, meaning that they are not killed by the chemotherapy drugs. And we isolate then EV from either DMSO, vehicle-treated GSCs, or temolizumab-treated GSCs. These are the control for the preparation of this EV. We didn't see much change in terms of size of the EV that we collect from these two conditions, but we did see a significant increase in the concentration of EV upon therapeutic challenge. We then went on to characterize at the molecular level by proteomic analysis the difference between the two groups. I didn't show here, but there was a significant amount of shared proteins belonging to the exosome extracellular vesicle coterms, which was expected. But we also identify 3 clusters related to proteasome and ribosome that are shared between the two conditions. And the third cluster here, highlighted in green, related to actin cytoskeleton and cell adhesion only in the TMZ-derived extracellular, TMZ-treated cells-derived extracellular vesicles. And we are currently trying to dig into these candidates that might play a role in further, let's say, invasion of the tumor cells. So our next step was to try to see what happened in real life with patients, and we have the chance to get our hands on longitudinal samples of patients over their care. So just as a quick reminder, patient upon diagnosis will undergo a surgery, then this will be followed by 2 break sessions with the radiochemotherapy treatment, the so-called STEP protocol. Then upon this adjuvant chemotherapy treatment with the temolizumab, and then follow up and eventually a second line of treatment. Relapse will occur most of the time during this period. So what we did was to isolate EV from this cohort and then measure the concentration of extracellular vesicles, either by interferometry or using CD63 ELISA. And what we observed, is that the number of vesicles was reduced upon the radiochemotherapy regime. No much change during this adjuvant period here. But when we put things together here for all patients, we see a small drop after the radiochemotherapy period, and this number of vesicles seems to bounce back during the second line of chemotherapy, the adjuvant chemotherapy treatment or the second line chemotherapy treatment. And if you look for one individual here, you can appreciate that first the concentration of vesicle seems to be a dynamic parameter, it's not steady over time. This is a period of almost 2 years here. And you can see this peak here during this time of the treatment, which is around the time of the relapse. So then we revisit our different patients and pair them. And as you can see, between that upon relapse, there's for most of them an increase in the number of vesicles. Of course, we need to increase the number of patients in this cohort and try to seek for some biomarkers associated with the number of vesicles. And this is something that we start to look into by label-free proteomic analysis of extracellular vesicles at the time of diagnosis for glioblastoma patient as compared to healthy donor from the general population. The proteomic analysis return a good enrichment in exosomes and extracellular vesicles proteins, which was again expected and entering on the methodology. The PCA analysis also nicely discriminate between our GB patient and the LC donor, with a significant amount of shared proteins between the two groups, and some differentially expressed proteins, again, between the different groups. That can be further appreciated on this Volcano plot, where, for instance, these two proteins here, Ficalin 3 and the von Willebrand factor, are highly upregulated in the GB group, in the EV from the glioblastoma patient. And when we did an ELISA on a larger cohort of patients at the time of diagnosis, although it's quite variable, we have an increase in the concentration of von Willebrand factor from EV fraction, while it was not the case when we look in the plasma. So with this, I will conclude on this first part by saying that we show that GSC can release extracellular vesicles that communicate with the human brain endothelial cells with at least 2 different factors that can be involved in the permeability effect. We are right now digging into the effect of these vesicles on other properties of or malignant properties of these cells, such as spreading, evasion, and resistance. And one question is what happened to these vesicles when they are circulating in periphery where the tumor is not going, basically, because these tumors are not metastasizing. So this is ongoing question in the lab. But now, for the time being, I would like to switch to a second aspect of the research we are doing in the lab, and that concern the mechanisms involved in the GC maintenance and whether vesicles can somehow intrinsically participate to the properties of these tumor cells. And why that? There's a significant amount of literature that showed that GSC somehow parade the cellular trafficking to their own benefit. And actually, it has been well shown that these cells have a low level of lysosome functioning and lysosome flux that helps them to maintain their fitness. And any change in the number or a function of lysosomes will be deleterious for this cell population. And actually, in our lab, we found that this protease MALT1 helps to maintain a low level of lysosome. And if you go to the TCGA, the Cancer Genome Atlas for GBM patient, it's quite striking that a low level of MALT1 expression is associated with a better prognosis for patient. And when we look in vitro, we knock down MALT1 in this GSC, and silencing MALT1 was associated with an increase in the number of lysosomes. Their abundance was higher, and cells will ultimately stop to proliferate and die. And we were kind of lucky because there's a fairly good inhibitor targeting MALT1. This compound is called mepazine, MPZ here. And when we treat our GCs with the mepazine compound, we observe a general increase in the number of lysosomes. Stain is here either by LAMP staining or by lysotracker, same results, an increase in the number of lysosomes. And this was associated with a strong reduction in their viability and ability to form spheres. When inject in vivo, mepazine reduced the tumor growth. So then we try to understand better the mechanisms behind this effect on the lysosome, and to make a long story short, we identified that in response to MALT1 inhibition, cholesterol was somehow sequestered within this lysosome, and this is what you can appreciate with this Philippine staining. We use 2 different pharmacological inhibitors and 2 different duplexes to silence MALT1. And in all cases, there was an increase in the number of lysosomes and a trapping of cholesterol with this lysosome. And this was somehow reminiscent of the effect of blocking NPC cholesterol transporter. And interestingly, when we feed back our GCs with cholesterol, we did see that the number of lysosomes was still increased upon mepazine. But the most interesting phenotype was that viability was restored, suggesting that upon MALT1 inhibition, the number of lysosomes is increased, trap cholesterol, and this is deleterious for the cells. If you feed back cells with cholesterol, now you make it more available for the cells, and they are rescued. We use as a proof of concept of this mechanism the U86A compound that blocks NPC transporter, and this recapitulates the effect of mepazine as it reduces tumor growth. So this is something that we are still investigating. But one striking question then after when you think about extracellular vesicles and all these trafficking pathway is that there's maybe multiple routes for them that might somehow compensate. If we block the lysosome route, what happened to then the extracellular vesicles route? And can we target this pathway as a balance for the glioblastoma cell fitness? So to start addressing this question, we do a bit of data mining of the TCGA for genes that were reported and important for EV release and biogenesis. And with all of this, I'm sorry I didn't use the database from Albert. I think it would have been interesting to include here. But nevertheless, we have 9 genes which expression higher or lower was associated with a change in the probability of survival of patients. They are recapitulated here. And we were happy to see that RAP27A was among them, which was shown by many others, but that's validating the process. And we were interested in this MRKL gene. MRKL is an interesting pseudokinase. It's well reported and known to be a mediator of programmed cell death by necroptosis, but is also associated with the escort machinery. So we look at the effect of MRKL in EV released by GSCs. So what we did was to knock down MRKL in the cells, and this largely reduced the number of vesicles released by these cells in vitro. We then look at how cells were organized. We didn't see much effect on lysosomes, but instead what we see is the relocalization of CD63 from a more, let's say, peripheral overall staining of CD63 within the cells to a more confined and perinuclear staining of CD63, which is reminiscent of the phenotype of RAP27 knockdown cells. So then our question was whether we can target MRKL. And again, there's a good inhibitor, a well-characterized inhibitor of MRKL, which is called NSA. I lose again the pointer, sorry. So NSA, we check in vitro, was able to recapitulate the effect of the silencing of MRKL in terms of number of vesicles. But additionally, it was able to reduce the ability of cells to form spheres and to proliferate. It was a more anti-proliferative effect that we see in this case with NSA. We then test this compound in vivo with the good opportunity that NSA is able to block human MRKL, but not mouse MRKL, suggesting that in that case, we can target only tumor cells in our model. So we start this therapeutic regime with NSA in mice. And as you can appreciate, there was a modest but still a significant effect on the survival of animals over time with this NSA compound, which was associated with a reduction in the tumor burden, as visualized here with the bioluminescence, or at the endpoint here with this staining. You can appreciate the reduction in the tumor size. It's not completely gone, never happens, but a significant reduction in the tumor burden with this treatment. So basically, this is where we are right now with this project. And still, I think it's quite important to notice that there's many ways to control the way GSC will communicate with the environment. And there's maybe some intrinsic cues coming from the vesicular trafficking, either via the endolysosome route or the extracellular vesicle route. And this is something that we pursue to study. And so, of course, there's still many questions open concerning the specific communication between GSC and endothelial cells and the impact and maintenance that I already mentioned in the different conclusions. And with that, I'd like to conclude and thank the people in the lab that did the job from past and present member, or were involved either in the signaling GSCs and EV project. I'd like also to thank the core facilities in the Institute, our collaborators worldwide, and our funding agency. And thank you very much for your attention.
Thank you so much. So is there any question or comment from the audience?
So beautiful talk, thank you. So a couple years ago, I think I saw a study on relationship between nanotubes and resistance to TMZ and irradiating radiation. So in one of your pictures, you show that when you blocked the EVs, you reduce the length or the numbers of where those processes. I mean,
So-
you made allusion to angiogenesis, right? So you
Yeah,
skip
yeah.
right? So
the images I was showing are images of brain endothelial cells grown as 3D. So it's brain endothelial cells, and we showed the sprouting of these endothelial cells. So EV can induce the process of angiogenesis. Now, we never check on the effect on nanotube formation from the GSCs themselves. What I can tell is that we have ongoing study on the role of EV on adhesion of the cells and the spreading, and for sure they have a role, and we didn't dig yet into the nanotube. It's a completely new world for us, but this would be certainly interesting in the system.
Mhm. Thanks.
Thank you. Very interesting talk. I was wondering how you would rate the importance of using three-dimensional cell models also in studying EVs, and if you see big differences to two-dimensional cell models in the EV expression.
Okay, I didn't hear properly the questions. Sorry, can you?
So how important do you think is it to use three-dimensional cell culture models to study EVs?
So
Do you see big differences in the EV composition compared to two-dimensional?
This is something that we start to analyze right now. This is a very good point because, okay, so the cells, as they grow right now, they are in 3D, floating, non-adherent, without serum. As soon as you want them to be adherent, so 2D, then you add serum. So it completely changed also their the culture conditions are completely changed. And what we are trying to do right now is to set up a model where we can really compare the two, 2D versus 3D. And early results suggest that in 2D, there might be less of vesicles released, but maybe because they're trapped on the matrix. So right now we're trying to get vesicles, let's say, from the conditional media, flooding, and also the one that are trapped with the matrix. Because maybe the difference we early see are linked to the fact that some are trapped. But this is a very good point, yeah. Especially because there's a lot of adhesion molecules.
Thank you for the nice talk. So do you have any idea if your 3D cells can recapture the EVs that they are releasing?
Oh, yes, they do. They do. This is something that we check because one thing that we were wondering, especially with the MLKL project, that when we knock down or we block MLKL, there were less EV release, and this was associated with low proliferation. So one thing was that maybe they recapture less. So
that's not the case.
That's not the case.
In the basal condition, but for instance, when you play with when you block the cholesterol, for instance.
So
do you know if that
we never check that. We never checked that, but surely we need to check. This is something that we didn't have time yet to do.
Yeah,
Yeah.
because you have a huge effect, but I mean, it's possible that now you affect the reuptake, if you wish, and perhaps in such condition they cannot do that efficiently. So basically, what you do is perhaps some of
I totally agree with you because in steady state, they capture the same way, and adding EV won't boost them. But I agree with you. We need to challenge the lysosomes from inside, for instance, to see what happened with the EV. Yeah, that's totally something that we need to do. Yeah, for sure.
I have a question. Wonderful talk. The effect that you observed with U18, 668, there is a reduction in tumor.
Yes.
Right. Is there a reduction as well in EV release from this?
This is something that we didn't check yet. Yeah, unfortunately, we need to look at that. For now,
to the model of.
I know. That's also going along with the question of Greg. We really need to try to link the two processes. Yeah, that's surely something that we need to do.
Thank you.
Okay, Dr. Julie Coward, thank you so much for the
Thank you
nice
very
talk.
much.
And we have a small gift for you. Okay, this is the end of the morning session of our conference. Then it's time for the lunch. Then let's start the evening session at 13:50.
Hello, everyone. Welcome to the evening session of the conference. We have 4 wonderful speakers lined up for you, and there will be a moderated discussion at the end. And of course, we will have coffee and then take a group photo as well in this session. So to introduce the first lecturer, I think Omid will start the proceedings.
So hello again. The next lecture will be online by Dr. Kate Lee. Dr. Kate Lee is a research officer at the University of Hong Kong's Department of Obstetrics and Gynecology. He received a joint PhD in chemistry and medicine from the same university. During 2017 and 18, he was a visiting research fellow at the University of Cambridge Center for Turf Loss Research. His research interests include placenta development, extracellular vesicle biology, and reproductive glycobiology. Dr. Lee, if you can hear us, please go ahead and start your lecture. You cannot hear? We cannot hear you. It seems you are mute. just a moment. Yes.
Okay. Thank you. Can you hear me now?
Yeah, please.
Okay, so let me share the screen first. Okay, thank you for, first of all, really thank you for giving me a chance to talk about my research. And two sorry first. First of all, I can't make to come to join this great conference, but I really learned a lot during the previous sections because, well, it is the start of the semester in Hong Kong. So, well, everyone is busy with teaching. And the second thing is that, well, today Hong Kong is experiencing the strongest rain. They heard that it is the heaviest rain in the recent 200 years. So actually everywhere is flooding. And I heard that some of the communication facility is broken. So in case if the communication is not very good, I'm sorry about that first. So I would like to talk about the picture here. Well, first of all, well, it is the icon of our department right now. Well, we are requested to show this icon every time we present our work. But well, actually, I don't like this icon very much as a placenta biologist because, well, I found that, well, the placenta is actually missing. Well, the last time the placenta is missing from the physiology is around 500 years ago. So it is quite famous for Da Vinci drawing that missing the placenta. But anyway, so I would like to start my talk a little bit about the maternal-fetal interface, especially the placenta, and then I will talk about the placenta-derived extracellular vesicles on the macrophage, on the monocyte, and a little bit of what we are doing right now. So it is the maternal-fetal interface. Well, people would like to say placenta, but it is not really correct because, well, the placenta need to work together with the decidua. So that's why that makes the maternal-fetal interface is a very specific organ. It is the only organ that in our body contains two allogenic genotype that is coexistence, the gene from the mother and the gene from the fetus. So that means some of the immunomodulatory mechanism should exist. And the second thing is the fetal-maternal interface, the placenta, it is the only temporary organ in our body. It grows tremendously in the first 3 months of the pregnancy, and then they totally expel when the fetus is delivered. Well, the functions of the placenta, of course, include exchange of the materials, hormone secretion, but today we would like to focus a bit on the immune tolerance. So just a little bit of summary. Well, the blastocyte attached to the endometrium, and then the trophectoderm grows into a trophoblastic shield, and then the shield would further develop into the placenta. So in the placenta, the fetal placental villi is actually interacting with the endometrium, while the cells are intercommunicating with each other. So just a little bit of the summary that the cytotrophoblasts would differentiate into either the extravillous trophoblasts to mediate the artery remodeling, or the cytotrophoblasts differentiate, they fuse together with becoming the syncytial trophoblasts. Well, the syncytial trophoblast is especially important in our topic. It is because, well, most of the extracellular vesicle is coming out from the syncytial trophoblast. And then actually there are quite a high abundance of the placenta-derived vesicle can be found in the maternal blood during the pregnancy. And the number is increasing during the pregnancy in the proportional to the size of the placenta. Well, as everyone here is a EV biologist, I think I no need to talk about what is inside the vesicle and what's their function. But I would like to draw your attention to the something that is specific to the placenta extracellular vesicle. That is, other than the exosomes and the microvesicles, so nowadays we all group them together as extracellular vesicles. Actually, the placenta also produce quite a number of syncytial nuclear aggregate, which is very large in size, and then they are nucleated. But well, I think there's not so many research in this SNA. So there are quite a number of methods that can isolate the placenta-derived extracellular vesicle. So from the very basic, from the primary culture, from the explant, and if you are very confident with the surgical procedure, you can take the term placenta and attach to a pump so that you can have a perfusion of a very good quality placenta. Or just you can take the blood and then you can use the magnetic beads to isolate the vesicles. And of course, later on, we will have ultracentrifugations or gel filtration to further purify the vesicles. So in this study, we are using the placenta explant from the first trimester, placenta villi from the termination of pregnancy. Well, it is a precious sample because nowadays most of the termination of pregnancy is conducted by medical abortion, but not surgical abortion. So why we would like to use this kind of sample? It is because, well, the first trimester placental villi, it is the placental villi that is the placenta is actively developing. And it is actually the time that the fetal maternal tolerance take place. I mean, the most important time for that. So the explant will culture in a culture insert and then culture in a medium. And then because at that time, the placenta is in a hypoxic condition, so we will also put the culture in a hypoxic condition, and then we find that the H&E staining show that, well, the placenta villi is actually good in shape. So again, at the very beginning, we just take all of the vesicles and then to conduct microscopy, and then preliminary find that, well, there are microvesicle, there is exosome, and then we try to focus on whether if they are different or they are more or less the same. We find that, well, most of them we do a microRNA sequencing and find that, well, most of them are containing microRNA, and actually the microRNA are quite overlapping to each other, no matter their secretory mechanism is different. And then further on, we are using a quite classic protocol to isolate the exosome to further continue our study. Well, the vesicles are good in shape, and then, well, they express the common vesicle markers as well as a negative for the Golgi marker. Well, again, we conduct the microRNA sequencing for the exosome, and then using the software to predict the potential binding target of the microRNA, and then we can have a geoanalysis. And of all these geoanalysis terms, there's one of the terms that makes us interesting. It seems like, well, it seems like they are correlating with the myeloid cell differentiation. So that at that time, we would like to step a little bit further to the immunology side rather than the placenta side. So as a very basic study, I think, as again, most of you may already know that, well, we label the vesicles with the fluorescent label, and then we put the vesicle to different kinds of cells. And then at the vesicle, we find that as short as around 20 to 30 minutes, they start to bind the cell. And then the strongest time is about 18 hours. And then the signal would a little bit depreciate. But then the different thing is because we put the exosomes to the peripheral blood mononuclear cells, that means all kinds of the immune cells are present. But then it seems like the vesicles are not bind to all kinds of cells. well, it seems like they are selectively uptake to bind and or uptake to the monocyte, and some of them to the dendritic cell, and a little bit of them to the B cells. So we would like to, because of this result, we would like to continue the work on the decidual macrophage. So in the human decidualized endometrium, the decidual macrophage is the second largest population. It's approximately 20% of the leukocyte. And it is generally believed that the monocyte were migrated from the bloodstream, and then they go to the endometrium. And then when they expose to the microenvironment in the decidua, they differentiate into the macrophage. So previous study have already shown that, well, they do have a background of M2, that means alternative type, and plus some of the decidual specific markers. And if you see the pictures here, the macrophage, it is the spiral arteries, and these are the macrophage that next to the arteries. And here are the extracellular trophoblasts that invade the artery, that they try to reform the artery. So the macrophage are very adjacent to the macrophage, and people believe that they would have some functions in mediating the spiral artery conversion. And again, the change in decidual macrophage numbers and activities is associated with the placenta insufficiency and quite a number of pregnancy complications. So just a little bit of more about the artery remodeling, because when the villi that attach to the endometrium, the extracellular trophoblasts will go to the artery and remodel the artery to the wide opening. Well, why is that important? It is because when the fetus is growing in size, they need more and more perfusion. But then if the to have these kinds of perfusion, if the opening of the artery remains small, that means they need a very high blood flow. So the high blood flow will induce a very high blood pressure that could damage the fetus and the placenta. So that's why the opening, I mean the widen of the opening of the artery, is one of the key process of the placenta development. So we further start our study by a quite simple methodology. We take the human peripheral blood monocyte and then differentiate by using a factor MCSF. It is a M2 base factor and also abundantly found in the decidualized endometrium. And then at the last 2 days, we add the placenta-derived exosome into it, and then we check out, at first we check the different kinds of the markers of the macrophage, and with the M1, M2, M3, and some of the specific decidual macrophage markers. And our results show that, well, the placenta-derived exosome, but not the microvesicles, do affect, increase most of the M2 macrophage markers. And I'm sorry that I'm missing one of the results, that it also induced the ideal one as a decidual macrophage marker as well. So with this preliminary study, we further put the cell for the sequencing analysis, and the results also show that, well, the placenta vesicles do promote the cell differentiate into the M2 macrophage. Here are some of the macrophage, M2 macrophage surface markers. And we also do a GSEA analysis to match up the gene expression profile with the previous M1 and M2 macrophage, and it found that they are matched up with the M2 macrophage. And from the GO result, of course, there are important M2 cytokine signaling like 4, 13, and 10. But we also find out the extra matrix organization, which match up with our previous saying that the placenta development, the most important part is the remodeling of the arteries. So further study, we have identified one of the HDAC family, the HDAC9, is associated with the change of the macrophage, M2 macrophage profile. We suppress the HDAC9 with an siRNA on the human monocyte, and then they divide into macrophage, and then they find that, well, actually, they have a similar effect with the placenta-derived exosomes. So we further go back to the microRNA library of the placenta exosomes and find that, well, there are 3 microRNA that associate with the expression of the HDAC9. And then we take the mean mix of the microRNA and put it, and we find that one of the microRNA is associated with the functions, and as shown here. So as I mentioned, the macrophages are involved in the trophoblast migration, in the spiral artery remodeling functions. Well, in the spiral artery remodeling, it is a series of steps. Well, for example, start with the migration and invasion of the trophoblast. And then we also find out that, well, the macrophage that treated with the placenta-derived exosomes, and then they will release medium, and then that can induce the trophoblast invasion and migration as well. And then for the endothelial cells, an opposite result was found. That is, the endothelial cell will reduce in the migration, and then the angiogenesis of the endothelial cell will reduce. Well, it is because, well, actually the number of the endothelium during the spiral artery remodeling is unchanged, and then they are actually killed by the trophoblast and then transformed into a fibrinogen, wider opening arteries. But, well, our result unexpectedly showed that, because our previous thought is that the macrophage would further enhance, the immunotolerance profile, but we didn't find any further effects on the immune system on the decidue. So it is what we have right now. The placenta-derived exosome macrophage, they have a stronger M2 and M2 profile. But unexpectedly, one of the students that when he differentiate the macrophage, he forget to add the MCSF. So after 6 days of culture, and then he add the placenta-derived exosome. So quite unexpectedly, he find that, well, the vesicle on the monocyte, they have a much stronger effect than the derived macrophage. So we further confirm this effect with the mRNA sequencing, and again, the effect, we proved that is even stronger than the macrophage. So at that time, we formed our hypothesis a little bit. Well, actually, in the human circulation is about 10% of the monocyte. Well, it is the major phagocytic cells population in the systematic circulations. Well, again, it is mediators of the innate adaptive response. And then actually some of the functional change of the monocyte really do exist during pregnancy. But the mechanism is quite unclear. So that's why we follow up to look at the microRNA profile. Well, of course, the treatment of the exosome would suppress the general immune response to the monocyte. But quite unexpectedly, that it is found that most of the MHC1 and MHC2 markers of the placenta exosome treated monocyte were dramatically reduced. And then when we look at the literature, we identify monocytic myeloid-derived suppressor cells. And then we further shown that, well, the placenta-derived exosome would affect the differentiations of the MDSC. And then when we take back the blood from the normal and normal pregnant women and normal non-pregnant women, and then to try to isolate whether they do have the same change. And then we find that, well, actually, yes, the pregnant women, they also have an increase of the MDSC cells. That means, well, the placenta exosome may involve in the these kinds of phenomenon. Further on, because the MDSC cells is regulating the T cell response, we try to co-culture the T cells with the monocyte. But we find that if we co-culture the monocyte and T cell only with the monocyte-derived medium, there is no change of the T cell, CD4 and CD8 cell proliferation. But when we culture with the monocyte together with the T cell, I mean with the cell-cell interaction, it seems like there is a downregulation of the T cell and CD4 and CD8 cell proliferations. And we further show that there's slightly inductions of the regulatory T cells and reductions of the Th1 inflammatory cytokines. So a bit furthermore, we identified that, well, the placenta-derived exosome would trigger the PD-L1 expression. PD-L1 is one of the modulators of the quite important modulators of the tolerance and the Treg differentiation. It is inductions of PD-L1. And then using just as what I previously mentioned before, we check the microRNA, and then we find some of the microRNA do affecting the change in the PD-L1 level. And then of this microRNA, we identify a common target of P10. And then we further confirm the pathway is through the microRNA P10 and PD-L1 axis. And this change of P10 were also identified in non-pregnant and pregnant women. So to sum up, well, the placenta-derived exosomes, and some of the exosomes may affect the macrophage differentiation, and then they will becoming an M2 and decidual-like macrophage phenotype. And of course, I can just say it is a decidual-like macrophage phenotype because there are some other markers, I mean, is missing, and then there are some of the mediator of differentiation in the decidual that can also help the macrophage differentiation. Well, the macrophage could help the trophoblast functions. And then I think what is something new is that the vesicle is actually affecting the monocyte of the systematic immune system, which have total, I mean, overall effects on the T cell suppressive effect and induction of Treg. So lastly, I would like to talk a little bit about what we are doing right now. It is to study of the microRNA targetome of the placenta extracellular vesicle on a placenta complication called preeclampsia. So, well, actually, this slide shows, I mean, for me, it is the most important research question in the obstetric field. It is because for the obstetric complication, most of the obstetric complication will happen at the third trimester of pregnancy, like the fetal growth restriction, stillbirth, preterm birth, and preeclampsia. Well, preeclampsia is a disease that when the spinal artery remodeling is incomplete. Well, but then, well, actually, the placenta development is in the first trimester. Well, it is a time lag of the problem, the pathology, and then the complication. It is because when even though the problem of the placenta development, known as a placenta insufficiency, well, even though there is a placenta insufficiency, at the very first beginning, the fetus size is still small, so that even though the arteries cannot convert very well, the blood flow is still important enough to make the fetus survive. But then, well, when the fetus grows in size, they need more and more perfusion, and then the problems is usually occurred at the end of the pregnancy. So now on, the holy grail in the field is that, is there anything that we can use to predict the pregnancy complication at the third trimester during early pregnancy as early detection methods? So in this study, we have already obtained the maternal blood samples because there is no marker at that time, so that all the maternal blood sample will store until the end of pregnancy. just like the preeclampsia is about 2 to 4%. So every 100 maternal blood we're collecting, well, maybe at the end, about 2 of them will happen preeclampsia, and then we trace back retrospectively the blood, and then to take the microvesicles. We use quite a traditional method because the placenta extracellular vesicle have a specific marker of PLAP. We can just use the PLAP bits to isolate the vesicles, and then to compare the mRNA profile of the normal women and the preeclampsia pregnant women. We have by microRNA sequencing. So here is the profile. Well, it seems like it looks like the samples are separating quite well. There are quite a number of difference. And then we are using software to predict the mRNA binding target to see the GO and the KGG analysis. Again, we also find some of the markers. Let's like here, the myeloid cell differentiation comes out again. Well, but personally, well, I don't like the microRNA prediction software. I don't know whether you have experience with it or not. Well, of course they can predict you with give you some result, but I find that actually most of the time the result is not very accurate. Just like the HDAC story, they predict 10 HDAC, but totally find out just HDAC 9 is working. And then also for the other exosome, they predict 3 exosome, but finally one of them is working. So we designed to use AGO2 chipset method, which is a method that actually looking for the real binding microRNA on the target RNA. It is when the vesicles were treated with the cells, and then at some point we use UV to cross-link the microRNA and the target RNA, and then we use the AGO2 bits to pull out the target, and then we do an intermolecular ligation, and then we further purify it on a gel, and then we cut it out, and then to do a deep sequencing. So by doing some sort of bioinformatic analysis, we can identify the microRNA, RNA chimera. Well, these two are really the binding partner. Well, I'm sorry, the project is still ongoing, so I can't tell you too much at the moment, but of the cells we identify nowadays, some GO terms again related to the placenta development, and these are some of the positive and negative marker that affects when the placenta exosome treated with the cells and associated with the cell differentiation as well. So in summary, well, the placenta-derived vesicles to polarize the macrophage to help the spiral artery remodeling or just the trophoblast function. And the placenta exosomes are also affecting the immune tolerance of the circulating monocytes. And of course, the microRNA of the placenta-derived vesicle is possibly to have pregnancy-associated as a biomarker of pregnancy-associated complications, but we need more samples to construct the ROC curve for further calculations. And now on, we are going up to investigate the real binding microRNA targetome of the PEV on the cell differentiations. So lastly, I would like to thank all my collaborators and colleagues in Hong Kong, and a special thanks to our PhD student, Dr. Bai Kunfeng. Well, actually, the work that I present today is mostly work by him. So that's all of my presentation, and thank you, and questions are welcome.
Thank you, Dr. Lee, for your nice and interesting and informative lecture. And now the floor is open for any question. Thank you for the nice talk. So, Keith, so you show that you have an increase in M2 macrophages following treatment of those PBMCs with EVs derived from the placental tissue. So do you know if that increase in expression of macrophage marker is accompanied by increased M2 function?
Well,
Because that's just a frequency assay. You're just assaying the frequency, right? The number of cells that are M2 like characteristic, but functionally it does not necessarily translate into function.
Yes. Well, actually, the question you have a good question. Well, we didn't find the M2-like function of the monocyte-derived macrophage. As I mentioned, they did not affect the T cell functions unexpectedly. But as I mentioned in the later study, we found that well, the exosome regulate the monocyte, and then it seems like the monocyte have the M2 function rather than the macrophage. So that we try to claim that well, they have M2-like phenotype, but they don't have the function. But it seems like the decidual, I mean these kinds of exosome regulated macrophage do have a function on the trophoblast regulation of the tissue remodeling rather than the immunological functions. It is quite unexpected even for me.
So hi. So I have a question regarding the specificity of the placenta-derived EVs. So in most of your experiments, you use as a control PBS, which is fine. But so then from all your results, you know, what is really specific from the origin of the EVs, meaning placenta? So did you try to compare and to do other experiment with EVs that derive from other tissue, for instance?
Oh, well, I'm sorry, I didn't show that. At the very beginning, we have compared with the EV from the epithelial cell, I mean, from the endometrium epithelial cell, because in this sense, it is the fetal-maternal interface, it is the environment that I think is mixed up of mostly mixed up of the placenta EV and the endometrial epithelial EV. And the endometrial epithelial EV doesn't have these kinds of M2 or mediatory effects. But I'm sorry that I didn't show it.
No, so that perhaps I missed that part. So it means that everything you show is only specific and all the phenotype you observe are exclusively observed when you use EV from placenta, right?
Yes.
Thank you. It seems there is no more question, although it's very informative and interesting, and we are looking forward to hearing more from you, maybe during next year, and looking forward for more collaboration.
Thank you.
So by that, we'll continue with the next speaker. Thank you.
Thank you. Let's stop the share. Okay. Thank you.
Thank you very much. Our next speaker is Dr. Ilaria Prada from, she's a unit manager, cell biology group at Axam Limited in Milan, Italy. Dr. Prada got her PhD in Milan, Italy, and after working as a in postdoctoral and permanent principal investigator levels in Switzerland, Italy, and Sweden, she has started her current career at Axam, designing and developing cell-based assays for the identification of new therapeutical models, also involving the use of EVs. Her lecture title today is Selective Interaction of Glia-Derived Extracellular Vesicles with recipient cells in the CNS. Dr. Prada, the floor is yours. Thank you.
Thank you so much for the nice introduction, quite long as my age, and I'm really grateful to the organizers for inviting me, since I'm really excited to be here. As you heard, I'm no longer actively working in the EV field, but I switched from academia. The CNR in Milan, Italy was my main environment where I worked with EVs. And now working in a drug discovery company, I'm starting to bring also some interest in EVs. So I'm here also today. I would like to put together the piece of evidence that we built during my postdoc in Claudia Verdero's lab, to give a name, and then also to have some inputs from the community, the EV community, about which are the needs that early drug discovery companies, so early stage of development of big systems, could help you in to figure out together more specific mechanism, and in the end use vesicles and target vesicles to do what we want to do. So my view is to, of course, cure disease, as a kid would say, but in the end, this is it. So as said, I'm going to show you put together results of a number of papers and research, that I have performed together with mainly with the group in Milan, and trying to figure out, maybe here the pointer is better, how extracellular vesicles in the CNS, which was our main interest, can interact in different ways with the different cells present in the CNS, means neurons and glial cells, mainly our focus. So as you can see here, as all the other kind of cells, also glial cells can release vesicles. And as you can see here, quite old pictures, but very nice in my opinion. You can see here, I have to say, macrophages, not microglial cells, but really amazing to me and fascinating this videos in which we can see, maybe I can, well, no problem. The real release of vesicles, since I'm someone that wants really to see things, so I'm in love with imaging, and you'll see further. So our model, the vesicles that I've used mainly are microglial derived vesicles upon ATP stimulation. So this is something kind of specific already, since ATP triggers the P2X7 receptor, then causing the translocation of the acidic sphingomyelinase to the plasma membrane, where it hydrolyzes sphingomyelin lipids into ceramides, promoting the curvature of the membrane and promoting the release of large vesicles. I'm old-fashioned, so I will talk to you about large and small vesicles, since we have separated them by a differential centrifugation protocol, and then characterize them mainly by dimension, but not only. I will not show you all the markers, but believe me, and in the papers you'll find all the characterization about how pure are those vesicles. So as said, and also, of course, ATP trigger, ATP signal triggers the exocytosis of multiple multivesicular bodies and the release of small vesicles, or so-called exosomes. So here you are, the model. We have isolated mainly rat primary cells, so rat primary glia from rat brains, pups, P2 rats, and use also astrocytes and hippocampal neurons from the same kind of animals. So after 30 minutes or 1 hour incubation with ATP, then we have the mixed population. By, as said, differential centrifugation protocol, I got the two different populations. And I'll show you today mainly experiments performed with the big fraction of vesicles. So first thing first, of course, ATP is something that is changing the donor cell state. So in the years, we've also questioned which are the difference in terms of proteins, mainly this is a proteomic analysis, after an ATP stimulus given to the macroglial cells. And here we have the answer. They're quite different in proteomic profile compared to the control, let's say, untreated microglial cells, since we do know that microglial cells put in a petri dish, they react or are already reactive cells. So as you can see here, we have performed this study. We just isolate the vesicles, a lot, of course, of supernatants. And you can see here, first thing, that ATP, as expected, increased the number of vesicles yields, and mainly switched the profile in increased markers of proteins involved in autophagy and phagocytosis and endocytosis processes first, then also energy metabolism, cell adhesion, and extracellular matrix. So this is first hint about, again, specific a specific bunch of proteins, specific markers you can find in differentially stimulated donor cells. And the same is true for astrocytic vesicles, which we have used a little bit less than microglial cells. This is a paper not from our group, but indeed it has already been published. that also triggering associates with different stimuli can lead you to have enriched vesicles in different kind of proteins, like for example, increasing neural outgrowth signals, or dendritic branching, or IL-1 beta, a pro-inflammatory stimulus, can of course trigger the peripheral immune response. So first thing, donor cells exposed to different stimuli release EVs that carries different sets of proteins, something which is already well known, but that we have also added this piece of information. So what about microRNAs that we have already heard a lot today about? Also, in our case, we working with immuno type of cells, microglial cells. We wondered, well, we have always in culture resting or untreated microglial cells, then we can trigger them with different kind of stimuli, pro-inflammatory or pro-regenerative or protective stimuli. So we decided to prepare again bunches of vesicles, and in this case, we analyzed both the population of small and large vesicles. And in collaboration with Francesca Peruzzi at the Skull Cancer Center in New Orleans, we performed a profiling with the exicon chips of the set of microRNA present in the vesicles. And as already seen and well known, also as we found a really interesting fact that microRNAs, this was 2018. So it was quite interesting that we were able to find enriched microRNAs in the vesicles compared to the donor cells. That means that, as all we well know now, there are active mechanisms that enrich microRNAs inside the vesicles. And these are the profile of untreated cells, but you can see here that we have indeed microRNAs enriched in the particles, which are not that high expressed in the cells. That means that they should transfer some kind of signal, as we have seen. And among the microRNAs present in these vesicles, we have focused our attention on a particular triplet of microRNAs, and one in particular that are able to target neuronal genes. Our interest is the CNS. So our idea was, well, glial cells can communicate something to neurons using vesicles. That was our question, and that was we saw. So we decided to follow the fate of this microRNA 136, which is highly enriched in the large vesicles of microglial cells, and the same is for astrocytic vesicles, while it's very low expressed in neurons. This microRNA is increased in levels during inflammation. So when you have inflammation, then you have this that can possibly do something to neurons. So first question, okay, we have this species. We want to see whether the idea of transfer is true. Now we know that microRNAs can be transferred. So this is a very simple assay at the time in which we could show with a reporter system that we transfected hippocampal neurons with a renal luciferase sensor for the presence of the microRNA that switch off the light. And we exposed the neurons to microvesicles obtained from microglia triggered with pro-inflammatory stimuli. What we saw is that we had a 40% decrease in the light emission, meaning that something switched off the system. So this something should be the microRNA. And you can see here the control of direct transfection of the microRNA inside the neurons, and you have an 80%, of course, of darkness. And when we pretreated the donor cells with anti-microRNA, so a sequence that blocks, recognize the microRNA and blocks the possibility to align with a gene, with a sequence in the gene, then we have a rescue in the luminescence. So microvesicles, in this case, can transfer microRNA to the neuron cells. And this is only occurring when the vesicles are free to in some way interact with the neurons. So when we perform this experiment in which cloaked the phosphatidylserine residues exposed at the outer leaflet of the vesicle and masking those residues, so avoiding the possibility for the vesicles to interact with the neurons, then we can see here that there's no the light is still at control levels. That means that the system has not met the microRNA. So another point, large EVs can transfer microRNA. I would like to open a point this evening about this, since we all know that there's also debate in the field on how many copies of microRNA you need to really switch off a system, to really modulate a system. I don't have the answer, I have to tell you. I have just evidence. So, and I'm one of the many people having those kind of evidences. So I believe that we really need to investigate deeper and more in detail, which is the mechanism by which this is working. So, sorry, among the genes, so we wanted to see the functional, if there's any functional effect of this microRNA translated in the cells. So the target genes are neuroligin 1, namely, and which is a constitutive and scaffold protein in the excitatory postsynaptic compartment, and synaptotactin 1, a calcium sensor. So as you can see here, we just measured by classical Western blot in the neurons the effect, and you can see here that we have downregulation of both the proteins. So those microRNAs can actively do something in the cells, not only on the reporter system. Next question. Well, neuroligin 1 is, as said, an important protein present in the excitatory dendritic spines of neurons. So in a situation of inflammation, of chronic inflammation, what does it happen if we really have a downregulation of the protein to the neurons, to the dendritic spines? So we transfected hippocampal neurons with RFP just to have this kind of images, really detailed images. And then we started analyzing the dendritic spines. First, from a morphological point of view, so you can see here, control neurons, neurons exposed for 2 days to M1, so pro-inflammatory microglia-derived vesicles, or as a control, let me say, M2 macroglial. So you can see that we, with pro-inflammatory microvesicles, so a mere 136 containing vesicles, we have a decrease in the total number of dendritic spines. Mainly, the thin spines, so the immature spines, are affected. So going on, the other question was, okay, what about synapses then? So we performed immunofluorescence by double staining pre- and postsynaptic compartments with Bassoon and Shank2 markers, here in red Bassoon and in green Shank2. And we measured the juxtaposed compartments. That means that we have some formed synapses, not functional, but still present. And you can see here again that the number and also the density along the dendrites of the synapses is reduced after exposure of the inflammatory vesicles. And also from a functional point of view, this is so functionally the vesicles can reduce the spontaneous electrical activity of those neurons. So this is something, but more interesting, I showed you in vitro. Now here you can see what happened in vivo. So some colleagues of mine were able to prepare and place mini osmotic pumps filled with, of course, control and all the different kind of vesicles that we needed for the experiment. And pro-inflammatory vesicles were able to reduce the number of spines, so the spine density also in the hippocampus of living mice in this case. So to us, it was really something. So different kind of donor cells, different effects downstream of the vesicles. And here I want to point out that we do not have any, I mean, of course, mechanical and surgical signature of inflammation were there, but you have a kind of baseline, so we can really compare the effect of M1 vesicles to the other kind of vesicles. So this is really something linked to the presence of the inflammatory signs. Then As I said to you, I really love to see things with my eyes by imaging. So thanks to a collaboration with Dan Kozak in Trieste in Italy, we were able, as first group to our knowledge, to handle microvesicles, and that's why we needed to use the large ones, one of the many reasons, since we need enough resolution to be able to see them. And how we handle them? With optical tweezers. Optical tweezers is a system in which you couple confocal microscope, spinning disk microscope, any kind of microscope you like, to an infrared laser that allows you to trap nanoparticles. So it has been developed and used by physicists to move nanoparticles, nanorods, nano things. And we used it for biological derived nanoparticles. And as you can see here, hopefully you can see it. Yes, this is how the laser looks like. And when you put the proper filter in the in the light path, sorry, then you can see properly the image by contrast, and you can see here a vesicle. Of course, for the sake of time, I'm not going to show you all the controls, but believe me, they're all published again. We performed all the controls to be pretty sure that we were working with cell-derived vesicles, of course. And what we decided to do, so you have here a system in which you have the vesicle, which is strapped by a laser, and then underneath it, you have the cover slip where you have all the cells plate, and then you can spatially decide where to put the vesicle. So you move your cover slip, you have here the vesicle, and then you change the focus, and then you can control spatially the interaction between the membranes. So you can also force it, of course. So you have to deal and to set things in a standardized way, let's say. So what we discovered by performing this kind of experiment, again, using microglia-derived vesicles, I'm going to show you only these results, but we also tested other kind of vesicles like associated ones. We were able, it will take some time to load, yeah. Microglia-IV's interaction, we investigated microglia-IV plus astrocyte interaction. So here, maybe I can go back and then let you see it again. I don't know how to, okay. You can see that we have placed the vesicle trap off. That means that the laser is off and the vesicle is still there for quite some time. On the other way, on microglial cells, I mean, this was completely blind experiment. We in the beginning didn't know what to expect. So we did the same thing on microglial cells, different microglial cells. And as you maybe can see here, let me try to get back once more. Yeah, we placed here the vesicle or here, and then it's adhered, which was something, since my question was, well, this transfer should be physical. So we need to have contact between the vesicle and the donors and the recipient cell. To my surprise, they also move, and I have quantified the movement of the vesicles. So many of them adhered on both microglial cells and astrocyte cells, but only few, I mean 1 in 7, moved on the surface of astrocytes, while the majority of the vesicles moved along the surface, so surf on the plasma membrane of the microglial cells. And to add another piece of information, we again clocked the phosphatidylserine residues with an Nexin 2, and as you can see here, this avoided the interaction with the plasma membrane of the microglial cells, meaning that when you switch off the laser, then the vesicle will fly away. So the trap is on here, and I tried to really stuck the vesicle to the membrane, like pushing it. But when I switched off the trap, you can see it moving away instantly. That means that we didn't have any interaction that could keep the vesicle on the surface of the cell. So, well, this is what I just said to you. Okay, so now to the point. This is really interesting how the glial cells can communicate one each other, but our point is how glial cells communicate to neurons. So I repeated the same experiment onto hippocampal neurons. And here you can see frames of a movie in which I have placed the vesicle on the body of the neuron. And I can say that it adhered and resides for a long time, while again, after clocking the phosphatidylserine residues with annexin 5, then again, the interaction was disrupted. And you can see here in controlled vesicles, we have 72% of adhering vesicles, while it decreases down to 33% after the clocking. So not all the vesicles were discarded, but the majority. Still something was there that allows in some cases to have an interaction. Okay, so, and this has been recorded, as you can see, for up to 1 hour of imaging, so quite a long time. And the vesicle was still there, not disappearing, not moving, not doing anything different. So, okay, again, a question. If the vesicle is still there, how is it possible that it could transfer something inside the neurons, since microRNAs should be internalized by the target cells? So again, transfected hippocampal neurons, in this case with SNAP25 RFP, exposed for 3 hours in this case to microvesicles that comes from, in this case, astrocytes transfected with farnesyl GFP. Why astrocytes? Since I think you know, or if you don't know, I'll tell you, microglial cells, primary microglial cells are really, really tough to transfect. So it's easier to transfect astrocytes. And this farnesyl GFP, it's nothing but a marker, a fluorescent label that I put inside the membrane I put inside the lumen of the vesicles, trying to follow it by fluorescence. And as you can see here, I have reconstructed the surface of these neurons and located the vesicles that still were residing on the surface of the neurons. And you can see here also some projections on different axes. So this is maximal projection on the Z-axis of the stack images that I took. It's 500 nanometer resolution here. And then X and Y projections. And you can see really nicely that you have no overlap between, for example, here you see between the two fluorescent signals. That means that the members are possibly near one each other, but not internalized. And here, just for our knowledge, I've reported a number of papers in which, on the other hand, has been described the internalization of small vesicles inside neurons. So this is another point of differentiation. Large vesicles have never been observed inside the neurons, while small vesicles, yes, of course. And another experiment. So what's going on during this hour of interaction between the cell and the vesicle? Well, one possibility could be the complete fusion, but after 1 hour it's still there, we can see it. So I went back to very old style literature and I found out a self-quenching dye, which is this octadecyl rhodamine B, which is self-quenching, as said, at high concentration, and it can emit a red signal after the dilution of the dye in the membranes. So I prepared vesicles from microglial cells loaded with calcine, which is a living dye, green dye, to again have something to follow and to visualize the green vesicles, hopefully, falling on the neurons. In this case, I just incubated the cells with the preparation of vesicles. I didn't perform optical tweezer experiments. And I pray to God to see any kind of sign of fusion, since that was my hypothesis. And I have to tell you that it was not that frequent, but I was so lucky in a few occasions to be able to be in the right field where the vesicles were coming on the neurons. And you can see here some frames of the movies recorded, showing you that the green fluorescence, so the calcine, is still present from the first second up to, in this case, 30 seconds, while the red signal, because of the time resolution was not that fast of the instrument I used, I'm sure I missed some event of lighting of the red signal, but still you can see some kind of kinetics in the red signal that suggests at least a partial fusion, so dilution of the dye in the unlabeled membrane of the recipient cells. These traces are very short, since after some time I had the bleaching of the dyes. Okay, so this is about vesicles on the body of the neuron bodies. More interesting, what happens on the processes of the neurons? So some colleague of mine, Dr. Dariga, and other people in the lab published in the end a very nice paper, in my opinion, in which they showed that the vesicles not only interact with the processes of neurons, both dendrites, but most interesting neurites. And again, we're using the optical tweezer techniques. But also another interesting fact that they showed is that some kind of vesicles are filled with ATP. And this ATP is the energy source for their movement, while some others are not filled with ATP, and they can move binding receptors, like, for example, PRP, so prionic protein receptors on the surface of neurons. So we again have differences in the population of vesicles that behave also in possibly different ways. They were not able to discriminate between the anterograde and retrograde movement according to ATP loaded or receptor-based movement. But in the end, this is something that we have to unravel, in my opinion, to understand better these events. So that's mainly the summary. So what we have hypothesized, and also in this paper shown, is that small vesicles can be internalized in the neurons, as people has already published, so based on the literature. And they can also move inside the processes, but large vesicles cannot be internalized, but they are able to move, and the hypothesis is to spread something across the neural networks. So last point that I would like to address with you today is another paper in which we have focused on Alzheimer's disease. So spreading things, let me quote this, through microvesicles along the neuronal network is, of course, something that could be of large interest in degenerative diseases in the brain. So starting from the knowledge that Alzheimer's patients' CSF is filled with microglial vesicles. So you can see here, healthy control patients, mild cognitive impairment patients, AD patients. This is the amount of IB4 positive vesicles, so microglial derived vesicles. It's also now known that microglial cells in culture exposed to synthetic peptide, but not only synthetic, since we also have some controls experiments in which we have exposed microglial cells to the peptide produced by CHO cells, so more physiologically prepared and packed, let me say it like this. So microglial cells can internalize A-beta and can also associate it to the microvesicles that they release. Those microvesicles, in this case, again, derived from the CSF of patients, have also really strong neurotoxic effects. So again, some signal, in this case neurotoxic, is transferred by microvesicles to the target cells. And you can see here, this is just the percentage of neuronal death that we could measure with a control, normalized, and then adding a Alzheimer's disease-derived microvesicles, or we can see also a little bit of reduction in the toxicity when we treat the vesicles with anti-A-beta antibodies, so masking a bit the peptide. Well, again, the optical tweezers. So we wanted to observe in last year paper the behavior of the vesicles. We wonder, well, the cargo or the associated proteins can influence also the property of movement and the behavior of interaction of the vesicle between vesicles and donor cells. So again, you can see here RFP transfected hippocampal neurons and vesicles control microglial vesicles or A beta exposed microglia-derived vesicles placed on the neurites this time. And well, maybe you can, oh, okay, you can see the movie here. So you can see that the vesicle is really walking on the process. And the properties of this movement, of this interaction, change really a lot when the vesicles are, in the vesicle is present A beta. So the adhesion properties are not changing that much. But the movement abilities of A beta vesicles are really higher compared to the control vesicles. And also they are more fast, they move more, and they can cover longer distances. The really, really interesting fact is the directionality of these vesicles, since when we have control vesicles, we mainly have retrograde movement. That means from the periphery to the body of the neurons itself. But when we have this toxic signal of A beta, then we have the majority of the vesicles that move towards the end of the axon, the synapse in the end, and possibly taking contact with the subsequent neurons. So some kind of spreading of the message, not only by neurochemical modulators or other things, but also by the vesicles. I know that parenchyma is really, really packed. So another question to address is in 3D models, how this could happen. But at least in vitro, we have seen this, and not only in vitro. So skipping this, just to say that also in this case, A beta vesicles are able to modify the density of the dendritic spines, and also the number of synapses, not showing here. The last experiment I want to show you, which is really, really exciting to me, is an in vivo experiment in which Alzheimer's disease is a neurodegenerative disease. Early stages are now a little bit, I mean, we are trying to clarify also this point, which were not so clear, and still are, we need to have more knowledge in this phase of the disease. But we know that almost everything starts from the entorhinal cortex here, which is through the perforant path is then signaling to, I mean, the other way around, sorry, the entorhinal cortex to the dentate gyrus in the hippocampus. So this is a clear path connected in the brain. And the experiment was like this: to inject either A beta EVs, control EVs, then I will talk about the oligomers themselves, into the entorhinal cortex, and then wait for different time of observation and register the electrical activity locally in the entorhinal cortex or distantly in the dentate gyrus. So you can see here, maybe better, that when we inject in the entorhinal cortex and we just, sorry, I don't know here, we can see here control, you have a long-term potentiation. So this is an electrophysiological protocol in which you just trigger the signal of the cells and register it and record it, sorry. So you can see that with control vesicles, nothing really affects the long-term potentiation, while after 1 hour or 24 hours of residency of the A beta vesicles, you have here no longer activity, neural activity. And the most interesting thing is that peripherally, distantly, in the dentate gyrus, you can see that with the control vesicles, you have this still long-term potentiation. After 1 hour of vesicles, you do not yet have impairment of LTP, while after 24 hours, you have the impairment of the LTP, meaning that in some ways, the signals has been transferred and could be diffused from one side to the brain to another, which is clearly wired. So possibly through the surfing on the surface of the processes. This is not something that happens just with A beta alone, so we need to have it on the vesicles. And lastly, again, the annexin 5 cloaking of the phosphatidylate residues abolished the recognition between the cells with the target cells. So we again did not have any impairment of LTPs, also at longer times, 24 hours. So just want to show you here the big things that I try to recapitulate here and that we are hopefully going to discuss in the whole 2 days that we have to meet together. Trying to point out that, of course, things that now we already know, but we have added our own pieces and we can place some novelty also, mainly to me, in the optical tweezer manipulation. So we got some really nice insights that the interaction is something physically happening and the vesicles are behaving like they're not just in the liquid, they're not just floating around, but they, in some way, to me, know what to do and know where to go. I don't know, but they know. And this depends on the donor cell state, on the cargoes, on all the different things that we have seen here. So as I said, I would really like to put together all the pieces to get the knowledge to be able one day to engineer the vesicles or to be able to modulate as we need their interaction with the target cells to find solutions. So a few seconds to really, really thank you for having me here today. And of course, to Claudio Verderio, my former PI, on all our collaborators in New Orleans, in Italy, in New York City, all our fundings, and also Axum that allowed me to be here today. So thank you. Happy to have any questions.
Already.
Someone has to ask question, right? So I have a question and related to the slide that you interpret as fusion when you decorate your EV.
Oops. Yeah, partial fusion, possibly.
Or that you call partial fusion.
Possibly.
So could you comment? So what I noticed is that when you look at the time scale, right, it's very long. And to me, when I look at that, it resembles more like a duct.
Possibility. Yeah.
EVs than a fuse EV, especially when you look at the green signal, because you would expect the green signal to be released, right, very quickly.
Yeah.
Because the calcine is soluble, right?
This could also be diffusion of the dye. Yeah. This was just a test to see what was going on.
Yeah, but because no, I mean, your signal to me is very stable. I would more interpret that.
Yeah, in the end, it is
as an immobile vesicle.
I was expecting for more diffusion of the signal and then switching off.
And then the appearance, especially of the red one, right?
Yeah, yeah. But
because even if it was incomplete fusion, so you would expect that the calcine will at least, gradually disappear.
Well, I have talked with
perhaps in
the physicists.
steps,
Yeah, it could depend. We have speculated this could depend on the size of the pore formation. Sure. So calcine is quite big molecule. So yeah, that what you're saying I was expecting first, sure. But this was just a claim of possible partial fusion. This is not final demonstration, of course.
Okay. So yeah, if it's fusion, so it's definitely not like an opening channel that will expand.
Yeah.
It's not a classical one, at least.
Yeah. Maybe kiss and run mechanism. That was.
Or perhaps, but again, you would expect some.
Yeah, some diffuse. Yeah.
And the time scale, even for the red one, would be faster. It would be 100 milliseconds. Here we are already, several seconds.
Yeah, I had a drop here, and to me, it was just bleaching.
I think so.
Honest.
Yeah, because it's too long for fusion. I think.
Yeah. Thank you.
Hi. That's a nice presentation. Thank you so much. So based on your explanation and your presentation, it seems that the EVs are selectively packaging their cargo, but a singular cell is sending different messages. So why would it be selectively packaging, but different EVs from the same donor has different information?
So you mean, sorry, I'm not sure I get your question.
So Can I ask a question? Do you think the EVs are selectively packaged or they are randomly packaged?
No, they're selectively. I mean, the cargo is selectively enriched in the vesicle. Talking, for example, about microRNAs.
Yes.
I'm not saying that all the vesicles released at the same time from the same cell should be exactly the same.
Okay.
I have not investigated this, but from knowledge of the community, it's pretty likely that you can have different kind of vesicles from the same cell. So
the packaging is selective.
could be. So we have the evidence, but I've investigated this. So what I'm saying is that the enrichment of microRNAs in the vesicles is selective. I mean, it's kind of selective since you do not have the same amount in the cell and in the vesicle as it just bunches out. So this is what I'm saying here.
I agree. So then, so my question is, maybe you could speculate a bit. Why is it that different EVs produced by that same cell are being packaged differently?
Oh, that could be a very good question. So I have not really experiments to answer to it.
Any other questions? Yeah.
Very nice, Hilary.
Thank you.
So if you think about, I guess I'm thinking about the image where you show a EV traveling through the process, and that process itself will be comparatively slow. No, that's outside. I'm talking inside.
Yeah. Okay.
Yes. So that process itself will be relatively slow, right? So an EV in the
that
is that an image?
Yeah, but this is about
that one.
Other studies, not ours.
Yes.
Yeah.
So can you? I guess I'm trying to get to the bigger picture, right? So can you speculate what type of sort of cross-network signals that will require such a slow process, right? So when you think about these things, like in morphogen signaling, right? So animals do these type of things. Well, with HOG, they'll actually package HOG on vesicles in these processes, right? And that's to do specificity and timing and scaling and matching the developmental the developmental clock with tissue development. So in this context of brain, so can you speculate on what signal, what strategy really? Why are we doing this?
Why we are differentiating the small ones and the big ones and the different. Yeah, well, strategy could be, I mean, many. So inside ones, so small ones, to me could signal inside the neuron. So it's something that the neuron itself, it's some self-autonomous system in which they just have to produce something. We know that also the RNA factories are found along the neurites and along the processes. So it's such a fine and how to say, complicated morphology, the one of the cells also in the brain, that you have to have factories along the processes. So you cannot always have new proteins, for example, coming from the cell body and then going down to the processes. And you really need quick answers. So when you have spine plasticity, for example, it's better if you already have something locally present that can help you in getting this plasticity, which needs to be really quick. While when you have to send messages far away from the site where you are for any kind of purposes, also toxic messages as a beta-loaded vesicles, then you don't want them to travel inside the neurons, slow down, and then maybe be mixed with other membranes, and then get exocytosed outside, and then recapture and reuptaken from other neurons. So to me, it's easier if you can stay yourself and then travel where you have to go and then deliver your message and then spread some kind of voice. This is an idea.
If there are no more questions, we would like to thank you very much.
Thank you very much.
It was a very amazing lecture. Okay, so The lectures for this part of the session is done. So we will go down now, take the group photo, and then it will be coffee time until 3:40.
Who's going to say that? Just take them.
Okay, good evening again. We are starting the next session with the lecture by Dr. Claire Crew. Dr. Claire Crew received her PhD in 2015 at the University of Oklahoma Health Sciences Center in biochemistry and molecular biology in the lab of Luke Schueder. Her doctoral work defined the initiating events in the development of cardiac metabolic inflexibility during high-fat feeding in mice. Dr. Crew is currently an assistant professor at Washington University School of Medicine in St. Louis. Dr. Claire, please, we are waiting to listen to your lecture.
Wonderful. Can everyone hear me all right and see my slides?
Yeah, everything okay.
Okay, perfect. So it's really great to be able to have this opportunity to share our work with you guys today. I'm really sorry that I couldn't be there in person. It would be really great to do so, but I had other commitments that precluded my travel. But thank you to the organizers, and I hope we can have some discussion. Please feel free to contact me after this if you have further discussion about anything extracellular vesicles. So, our lab is really interested in adipocyte-derived or adipose tissue-derived extracellular vesicles and how they signal locally and systemically to regulate metabolism. So we are particularly interested in this in the context of obesity and its comorbidities. So as many of you know, I'm not really sure what the stats are in Estonia, but here in the US and a lot of Western countries, obesity is a huge problem. And the reason it's a problem is because excess body weight predisposes you to a number of different comorbidities. Some of these comorbidities are actually lethal, like cardiovascular disease, which actually is the leading cause of death in obese individuals. And so you also have type 2 diabetes, which predisposes you to a number of different comorbidities as well. So we have a very adipose tissue centric view of the pathogenesis of obesity and these comorbidities. The adipocyte, which is the functional cell of adipose tissue, has a really important role to play in whole body metabolism. So this cell can sense the nutrients or the calories that we take in the diet and integrate those signals with hormones from other organs, and then it makes its decision whether to store the excess calorie in as triglycerides in its big lipid droplet, or to release that fatty acids as energetic substrates for the rest of the body. And that happens during fasting. But adipocytes are also endocrine cells. So they release a lot of what we call adipokines or adipocyte-specific hormones. And this has beneficial effects on systemic metabolism, insulin sensitivity, and reduces inflammation in different organs. So when it comes to obesity, the adipocytes specifically and adipose tissue as a whole become very dysfunctional. And so the adipocytes are no longer able to kind of sequester all that excess calories and lipids because they become so engorged and hypertrophic. And so these lipids end up building up in other organs. And this leads to what we call lipotoxicity. This is characterized by a lot of oxidative stress, a lot of inflammation, and is in general considered to promote tissue damage. And so this is the reason why many have suggested that dysfunctional adipose tissue plays a large role in the pathogenesis of these different comorbidities. So we know now that adipocytes don't only release soluble hormones, they do release a lot of extracellular vesicles. And I don't need to belabor this to this crowd, but we know that adipocyte EVs contain tons of macromolecules like other cell type EVs that can have really powerful signaling effects on the receiving cells. So really the focus of our research has been on the small EV population, which of course you know is mainly exosomes, but also small and other small EVs that come from the endolysosomal system and some small microvesicles that bud off the plasma membrane. So I recently had the opportunity to kind of do a literature survey and think about the functions of EVs. And this was mainly in the context of metabolism, but I think it applies to very broad functions of EVs. And so I could find these functions sort of characterized into 4 different categories. And so the first category, EVs can act as a warning signal. And so this is when a cell is stressed. And so obviously EVs are kind of in many cells a stress-induced response. And so these stress-induced EVs can be taken up by surrounding cells and induces that same stress in the cells. But this is not pathogenic. It actually increases the defenses of those cells and permits resilience. The second is collateral damage, which is similar, but instead of releasing stress-induced EVs, these cells that are releasing EVs are very sick. And so they're releasing EVs that actually propagate disease or spread disease. And we see this in Alzheimer's. Adipocyte-derived EVs actually mainly are in this category. They actually spread insulin resistance and lipotoxicity and inflammation to other organs in obesity. The next one is protection. So this normally comes from EVs from stem cells. These EVs have inherently protective effects. For example, they might contain antioxidants that can protect other organs from oxidative stress. And the last one is recruitment. And I think there are very few examples of this in the literature, but there are examples of exercising myocytes that can recruit lipids from adipocytes as an energy source. Or cancer cells can release EVs that promote recruitment of different support cells. And so today I'm going to try and give you a broad view of adipose tissue-derived EVs. And I'm going to give you one story that focuses on this warning signal and another that focuses on the recruitment function. So this warning signal story started in my postdoc a few years ago when I was a postdoc. And we were really interested in determining why adipocytes become so dysfunctional over the progression of obesity. One of the major determinants of this we found was a loss of mitochondrial mass and function. So during chronic obesity, the adipocyte loses mitochondria for whatever reason, we don't know, reduced electron transport chain activity, high free radical production, so a lot of oxidative stress, and this all results in low ATP production. So of course, the adipocyte can't function without enough ATP. So one of the colleagues in my lab was really interested in generating a mouse model where she could mimic this sort of mitochondrial dysfunction in a cell type specific way. And so she did this by overexpressing mitochondrial ferritin. So this is an iron chaperone protein that we found when you overexpress in adipocytes results in iron overload in the matrix of the mitochondria. And this creates a lot of ROS generation, a lot of oxidative damage, and ultimately mitochondrial dysfunction. She made this mouse to only express this transgene in the adipocyte. And so she's doing this using an adiponectin-driven RTTA transgene. So in the presence of doxycycline, which we provide in the mouse's diet, the RTTA can then bind the TRE promoter, driving expression of our transgene. So we can induce this transgene at any point in the mouse's life with doxycycline, and it is specific to adipocytes. So as you can imagine, the adipocytes in this model have a lot of mitochondrial dysfunction and overall are very sick, producing a lot of stress hormones and so and a lot of oxidative stress. And so my colleague was basically surveying other tissues as negative controls to say, okay, we don't see oxidative stress in other tissues. But we found something very interesting, that actually the cardiac tissue among all the other tissues did show a significant increase in cardiac oxidative stress in our transgenic mice. And this is, you know, a adipocyte specific model. And so at first we thought, well, there's maybe leakiness of the transgene in the heart. So we did a lot of control experiments to show that that's just not the case, that there really seems to be a pro-oxidant signal being sent from adipocytes to the cardiac tissue in this mouse model. And so through many different sort of rational or like educated guess, I guess, we were really interested in pursuing EVs as the modulator of this pro-oxidant signal between these organs. We can purify EVs from the blood of these mice and show that the total particle count was much higher in our transgenic mice compared to the control mice. But of course, this is the plasma, right? So we assume these are coming from the adipocytes, but we don't actually know that. And so we went in vitro to determine if adipocytes do release more EVs in response to mitochondrial stress. And we can do this for both human and mouse adipocytes. We can take the stromal cells from the adipose tissue, which are enriched for adipocyte progenitors. You can differentiate these adipocytes in vitro. And then we can induce mitochondrial dysfunction by overexpressing mitochondrial ferritin, treating the cells with different electron transport chain inhibitors at a non-toxic dose, or high levels of palmitate, which is the more physiological way to induce mitochondrial dysfunction. And in all cases, we found that adipocytes respond to this mitochondrial damage by increasing the release of EVs. And if we purify these EVs from the media, we can treat mature cardiomyocytes and show that the EVs that came from stressed adipocytes do actually induce a mild pro-oxidant stress in cultured cardiomyocytes. And so we were on the right track, but we wanted to actually show this in vivo as well. And so we isolated small EVs again from our adipocyte culture, this time treated with a large dose of palmitate to mimic the obese state in vitro. And then we purified these EVs and labeled them as small EVs with a superscript PA for palmitic acid. And then we injected these EVs and collected heart tissue over time and to determine the oxidative stress. And we measure oxidative stress with protein carbonylation. As you can see here, an hour after injection of these stress-induced EVs, we do see a significant increase in cardiac oxidative stress. It was very mild, as you can tell, and very transient. So within 2 hours, there was no effect significantly. But at 16 hours, we actually saw an antioxidant effect. And you can see this as well earlier with our mass spec-based method of cardiac hydrogen peroxide production. 4 hours post-injection, you do see this robust antioxidant effect in the heart. And so this looks very much like an adaptive response. So in response to this mild pro-oxidant stress, the heart is adapting to this, which we can give you an example here of how that's true. Catalase protein is increased in the heart at this 2-hour time point, which is one of the major antioxidant proteins in the cardiac tissue. So I just wanted to mention that we are injecting a dose of these small EVs that are about calculated to be about 0.03% of what is endogenously circulating in mouse blood. We don't really know if this is physiological for adipocyte-specific EVs, but we think that it is not super physiological or pharmacological. So the big question is what is actually in these EVs that is promoting this pro-oxidant signal in cardiac tissue, and that transient burst of oxidative stress and the adaptive response of the heart. So to begin to understand this, we purified EVs from our control and our diplomidoferritin mice from the plasma and did proteomics. And we were surprised at the time to find that there was a significant increase in mitochondrial proteins in our EVs from our transgenic mice, and that's denoted here with an asterisk. We can confirm this by Western blot to show that EVs that come from our transgenic mouse plasma have higher levels of VDAC, HSP60, COX-4, all mitochondrial proteins. But they also carried more oxidatively damaged proteins, which makes sense because we think they're coming from adipocytes that are under a lot of energetic stress. This is not an artifact of our mouse model because in obese mice that are wild type, we can still see an increase in circulating small EV-associated mitochondrial proteins. And to save time, I won't show you the data, but we can actually purify these small EVs from the blood and show that they respire. They actually produce ATP, but they are oxidatively damaged. So we collaborated with physician scientists on campus, Sam Klein, who gave us plasma from humans. He gave us plasma from lean, healthy individuals, obese individuals that were metabolically healthy, so they don't have signs of diabetes, and then obese individuals that were unhealthy, so they were more pre-diabetic or diabetic. And so what we found is the total small EV concentration in the blood was only significantly highest if the humans were metabolically unhealthy and obese. But in all obese conditions, we saw high levels of small EV-associated mitochondrial proteins and oxidatively damaged proteins. And so this looks like it is a potential mechanism that might also be active in humans, but we don't think it's restricted to obesity. There have been a couple of reports now, including from the Lotville lab, that showed in cancer patients, you also see an increase in mitochondrial proteins in EVs in the blood. And so this is likely a response to disease, some sort of diseased cell that is releasing its mitochondria into the blood. So again, we were working with plasma. Our adipocytes actually releasing their mitochondria. So we went back into our in vitro system. We have our differentiated adipocytes. We can treat them with these electron transport chain inhibitors. We see higher levels of mitochondrial proteins and purified EVs in the media. And we also see the same thing with palmitate. And so what are these mitochondria? And so we found that these mitochondria were basically what has been described in the literature as mitochondrial derived vesicles. This is a process that is a housekeeping process in a lot of different cells where the damaged pieces of the mitochondria are sequestered into these budding structures, which pinch off, and this is what they look like, and they're actually sent to the lysosome for degradation. But as we know, the multivesicular body is the birthplace for exosomes, and so we thought, well, maybe the cell is deciding to release its damaged mitochondria instead of degrading it. One of the main factors that promote the production of these mitochondrial-derived vesicles is parkin. So we can show that if we have adipocytes that are a knockout for parkin, we get less mitochondrial proteins associated with EVs in the media. And if we inject mice with EVs that were from adipocyte stress with palmitate, but that do not have parkin, we actually don't see that mild pro-oxidant stress at the 1 hour time point as we do with wild type mice or wild type cells, suggesting that this is potentially this pro-oxidant signal is caused by these mitochondria packaged into EVs. So again, summarizing data, we can actually show that endogenous adipocyte mitochondria can traffic into the blood of mice and accumulate in cardiac tissue. We did this by making a mouse model that overexpressed a flag tag localized to mitochondria, but expressed only in adipocytes. And after about a 12-week high-fat diet, we could detect the flag tag and its associated mitochondria in the blood, as well as accumulating in the cardiac tissue. So of course, the big question here is this pro-oxidant signal actually detrimental to cardiac function and promote cardiovascular disease? To understand this, we teamed up with the cardiology department at UT Southwestern when I was there to do this in vivo ischemia reperfusion experiment. And so this is sort of mimicking a heart attack in mice, basically. So the surgery is ligation of this left anterior descending coronary artery. This creates a large ischemic zone at the apex of the heart, which causes a lot of cell death. 45 minutes after this, we release the ligature to reperfuse that area. And this reperfusion as well causes a lot of oxidative stress and cell death. And so we can actually quantify how much injury the heart sustained by measuring the size of that infarct zone or that dead or fibrotic zone. And so what we did is we injected the mice with PBS or the palmitate stress-induced UVs from adipocyte. 2 hours later, performed the surgery, and then we did readouts for cardiac injury. And the first readout we have was plasma cardiac troponin, which is a surrogate measure for infarct size. And this is 24 hours after the ischemia reperfusion surgery. You can see that there is a significant reduction in circulating troponin in those mice that received the small EVs. So we were surprised because this suggests that there is actually protection from these EVs. We can actually quantify the infarct size to show that there is, in fact, about a 40-50% reduction in infarct size in the mice that were pre-treated with these stress-induced EVs. But if the EVs came from parkin knockout adipocytes, you don't get that same protective effect. Again, suggesting that it might be these damaged pieces of mitochondria in the EVs that are promoting this resilience. We can also show 7 days after injury that the scarred zone is actually also less as a further measure of reduced initial injury. And so at first we were confused because we thought, pro-oxidant signal in the heart is very important or in the etiology of cardiovascular disease. So we thought these EVs would actually exacerbate this, but it doesn't. And so what we were thinking is that this is actually an example of mitohormesis. And so for those of you who are unfamiliar, this is generally a cell autonomous process where a cell is exposed to a mitochondrial stress. It releases signals to the nucleus to say, we need to take care of this. The nucleus will signal to increase the adaptive responses, stress defenses, as chaperone proteins, antioxidants, and this will actually make a very resilient cell. So if that same cell is subjected to a lethal stress, it will be better protected from death than if it hadn't seen that original stress. And so that's what we think is happening here, that we are seeing the first example of what we call interorgan mitohormesis, where adipocytes are under extreme energetic stress in obesity. They respond by packaging damaged pieces of mitochondria into EVs and sending them out into circulation. They target multiple organs, but they tend to have a specific predilection for the heart. And this results in a metabolic preconditioning. So it induces an oxidative stress and adaptation in the heart that makes the heart more resilient. So if you go in with an ischemic injury, the heart is better protected from death or from this lethal stress. And so we think this is a way for adipocytes to warn other tissues to say, we are becoming very dysfunctional. We will soon not be able to, protect you from this lipotoxic stress. So here's a signal to sort of get ready for that. But of course, obese individuals do succumb to cardiovascular disease. So we're really interested in determining if this mechanism might be maladaptive over time and actually promote cardiovascular disease. And so I just want to make a quick note that really this study, as well as several others that were published within a couple of years, really opened up this new field of cell-free mitochondria. The Brestoff group here at WashU showed that actually macrophages in the tissue are a major determinant of if adipocyte-derived mitochondria end up in circulation. So long-chain fatty acids in obesity actually prevent the uptake of mitochondria by macrophages, kind of favoring their entry into the blood and accumulation in the cardiac tissue. It was also shown that brown adipocytes can release damaged pieces of mitochondria that they identified were also mitochondrial-derived vesicles. And this is actually really important for the mitochondrial function in brown adipose tissue during thermogenesis. The Levy lab showed that there are mitochondria in EVs in the brain, and more so in Down syndrome and neurodegeneration. And of course, Hidalgo group showed that cardiomyocytes can release their own mitochondria in these large exophores that are important for cardiac function as well, and that macrophages can take these up and degrade them. And so I'm excited to see where this field is sort of going to go as sort of a subpopulation of extracellular vesicles that carry mitochondria. All right, so that was sort of the first example of how adipocyte EVs can act as this warning signal. The second one is recruitment. And so this story started when we decided to make a floxed mouse for SMPD3. And most of you probably know this enzyme. It is also called nutrasphingomyelinase 2. And the reason we wanted to knock this out, of course, is because we know this is a ceramide generating enzyme that promotes the production, or is a positive regulator of exosome biogenesis. And so the hope was that we knock this protein out specifically in adipocytes, and we can see how this affects, the EV-mediated signaling within the tissue and system-wide. So again, we're using a very similar system that I've described, but instead of inducing a transgene protein, we're inducing CRE here, and we have floxed SMPD3 gene, which is knocked out when we express the CRE. Again, specifically in adipocytes. As you can see here, we have PCR primers that can recognize when the floxed alleles have been cut and recombined. You can see that mice that have the CRE in the adipose tissue and have the flox, we do get recombination. Without the CRE, you don't get recombination. So it looks like so far this mouse is working. And so we can actually digest the adipose tissue. We can purify the adipocytes from the tissue because they float. And then the stromal cells all pellet. And then we can measure SMPD3 mRNA in those two fractions. This is to confirm the knockdown. What we found was very confusing. So the adipocytes, instead of having a decrease in SMPD3 mRNA, there was actually a significant increase by over 100-fold. These are the same adipocytes that are expressing CRE. You can see nice CRE expression. And we can actually, well, first of all, we actually have two independent founders for this floxed allele. Both of them show this exact same phenotype. We can, instead of crossing it to an inducible CRE, we can cross it to this adiponectin CRE, which is a much stronger CRE, and it is constitutively active. So it's active throughout the development of the mouse. And we still don't get a knockout. We get an increase in SMPD3 mRNA in adipocytes. So is this mouse actually working? So we crossed it to a whole body CRE, and we can show that in this case, we do get a nice reduction in SMPD3 mRNA in the liver, but also the adipose tissue. We can also do this in a hepatocyte or liver-specific way. So if we use a liver-specific CRE, we can show reduction in SMPD3 mRNA with no change in the subcutaneous adipose tissue. And so what is happening? Well, we actually had a hypothesis right away, and this is because we've seen something very similar before. So many years ago in my postdoc, we wanted to knock out caveolin-1 from the adipocyte, And we did the same thing. We fractionated the tissue and looked at mRNA. In this case, we did see that the adipocytes were completely depleted of the CAV1 mRNA. But as you can see, the protein was still there very robustly. And so this is the inducible CRE, and this is the whole body knockout to show that our antibody is working. And we also cross this to many different CREs. This is the constitutive adiponectin CRE. And again, in adipocytes, we see really maybe about a 40% reduction compared to the whole body knockout. And so further investigation showed that the reason we couldn't knock CAV1 out from the adipocyte is because endothelial cells, which also highly produce this protein, actually package it into extracellular vesicles that are sent back to the adipocyte to replenish the CAV1 pool. So if we do a simultaneous knockout of CAV1 from endothelial cells and adipocytes, you actually get a true knockout in the adipocyte fraction. And so we suspected this is what is happening with the SMPD3 model. In this study, we actually showed that there was really a massive exchange of cellular components between cells within the adipose tissue. And this is happening through EVs. And so it is very likely that there is a cell type that is sending SMPD3 mRNA back to the adipocyte. So if this was true, we would expect that the incoming mRNA that are coming in from uptake of EVs would be in the cytoplasm, right? So in the nucleus of the adipocyte, you shouldn't see any de novo production or transcription of this mRNA. So we should be able to detect the knockout there. And we do. So in purified nuclei from adipocytes, you do see a significant loss of SMPD3 mRNA, again, suggesting that the cell is actually not producing this mRNA. We can purify tissue directly, directly from adipose tissue and show that particularly the small EV fraction has a significantly higher levels of SMPD3 mRNA, again, supportive of our hypothesis. To further study this, we went in vitro and we differentiated adipocytes from control mice or from our adipocyte-specific knockout. We took that conditioned media and treated just regular stromal cells from the adipose tissue and then measured SMPD3 mRNA in those cells. And you can see that the conditioned media from the knockout adipocytes actually induced SMPD3 mRNA in the stromal cells. We think that this is selective for SMPD3 because if we quantify the mRNA from other SMPDs or other ceramide metabolizing enzymes, we don't see any effect, although we do see CRS3 increasing, but this is not very consistent with other data because in vivo, when we purify adipocytes in these mice, we don't see any changes in these different ceramide metabolizing mRNAs, where, as I've showed you, we do see a significant increase in SMPD3 mRNA. So this data was really interesting to us because it demonstrated that adipocytes, in response to the knockout, are actually inducing a signal to the stromal cells to induce SMPD3 mRNA. And we suspect that this mRNA is actually being transferred back to the adipocyte based on the data that we have so far. So to actually show that, we are using here a molecular beacon approach. This is an RNA that is a short oligo that forms the stem loop structure, which brings a fluorescent dye in close proximity with the quencher. So only when it is base paired with its target mRNA, in this case SMPD3, will the fluorescent dye be brought away from the quencher and be able to fluoresce. And so we can show that stromal vascular cells that are transfected with this molecular beacon show a very nice signal for SMPD3 mRNA. And compared to the mock transfected cells, and our probe is specific because we can use SMPD3 from the whole body, stromal cells from the whole body SMPD3 knockout and show that there is much less signal for SMPD3 mRNA than the wild type cells. So we did the actual experiment. We have the stromal vascular cells on the top of a transwell insert, and these have been transfected with the molecular beacon. And at the bottom, we have adipocytes that are either wild type or knockout for SMPD3. And what we found is that if adipocytes were cultured with the mock transfected SVF, you don't really see any transfer of this molecular beacon or the SMPD3 mRNA, the fluorescence above background. but if wild type adipocytes are co-cultured with SVF that does have the molecular beacon, you begin to see these little puncti of fluorescence forming, suggesting some transfer of mRNA. But this is highly accelerated if these adipocytes were knockout. And so we can show here, if we blow up the picture, that there is fluorescence that is localizing to these adipocytes. And so it does look like this SMPD3 mRNA is being transferred. But this was really fascinating data to us because it showed that somehow the adipocytes were communicating to the stromal cells this deficiency in mRNA, and it was recruiting back this mRNA from these stromal cells. We can also show that this transfer of SMPD3 mRNA happens in vivo. So we have our whole body or global SMPD3 knockout mouse. We can take out 2 subcutaneous fat pads from the mouse. One of those we kept for the before transplant tissue. So this is to confirm the SMPD3 knockout. And then we transplanted the second fat pad onto the subcutaneous fat pad of a wild type mouse. And then after 2 weeks, these fat pads will fuse, and we can see, the interface and actually dissect away the transplanted tissue. And then this will be our after transplant sample to see if any SMPD3 mRNA has been transferred. And we did this using RNAscope, which can use specific probes to look for SMPD3 specific mRNA. You can see the interface between the endogenous and transplanted tissue pretty well. So SMPD3 is highly expressed in wild type, which is in red, and not so much in the initial transplant, which should be a whole body knockout. And we're using a adiponectin mRNA here in green to depict, what is the adipocytes specifically. And so this is the before transplant sample. You can see here that there are a couple of puncti that look like there is some SMPD3 left, which you might expect. But the tissue that was transplanted with a wild type for 2 weeks, you see this massive increase in SMPD3 mRNA. It was very consistent over the whole fat pad that was transplanted. And so we do think this is a very robust response, and it is something that also happens in vivo. All right, so what cell type is actually donating SMPD3 mRNA? Well, it's not the endothelial cells. We did experiments to show that, like it was for the CAV1 story. And so we did single cell RNA sequencing to try and identify what cells in the adipose tissue highly express SMPD3. And as you can see right off the bat, that there really only one population here. This population is positive for PDGF receptor alpha. So this is a pre-adipocyte population, and it can be further defined by positivity for DPP4. And so this is a marker that signifies a less committed pre-adipocyte. And so we actually back sorted the DPP4 positive and negative population of these cells, repeated the co-culture experiment with the molecular beacon, and we saw absolutely no transfer. So this is definitely not the cell type, which was surprising, that donates SMPD3 mRNA. But interestingly, this being a distraction, we actually see a couple of cell populations here that are actually B cells that very lowly express SMPD3, but in response to the knockout, you do see more cells that are positive for SMPD3. And so we're pursuing that to determine if B cells are actually the donating cell for this SMPD3 mRNA. And so we think that this is really the first data that suggests that a cell that is depleted in a specific mRNA can actually signal that to other cells in the tissue, upregulating that mRNA and allowing that mRNA to kind of come back in extracellular vesicles. The initial signal, we have no idea what the mechanism actually is, but we have some evidence that it is some sort of extracellular vesicle, because if you deplete conditioned media with an ultracentrifugation step of EVs, we don't see the same response. So you don't get induction of mRNA and transfer to adipocytes. And so we are looking forward to kind of pursuing this mechanism and figuring out what's actually happening here. so overall, you all know, and I don't need to, I'm preaching to the choir, that EVs can have very diverse functions. I've given you an example here of how adipocytes can send out damaged mitochondria to other cells that act as a warning signal, making particularly cardiac tissue more resilient. We know adipocytes, I didn't touch on this today, but we know adipocytes are very heavily involved in collateral damage when it comes to obesity and diabetes, but also adipose tissue EVs can actually work for recruitment. And so not as you might expect for energy or for support, but in this case, it seems to be for selective mRNAs. And so I would like to thank my lab, which right now is pretty small, but we are still looking for people to grow it. My postdoc mentor lab, Philip Shearer, and the members of the lab that were important in some of these studies, cardiology, UT Southwestern for the ischemia reperfusion experiments, Sam Klein and his group here at WashU for the human samples. Chen Liu helped us make the SMP3 floxed mouse. Laurent helped us with the RNA scope experiments. Ruth Gradidio is part of our the metabolic core who participated in these experiments. John Brestoff here at WashU is our strong collaborator for us because he's also interested in cell-free mitochondria. And so with that, I will take any questions and comments and look forward to some discussion.
Thank you, Dr. Crew. Thank you very much for the very nice and interesting talk. Now the floor is open for any question.
So congratulations, Claire, for the outstanding talk. So Greg Lavieux speaking. So I have actually two questions. The first one is related to the first part with the cell-free mitochondria.
Okay.
So how sure are you that the mitochondria are truly within some kind of EVs, right? Are you 100% sure that they are protected or not? And could you comment on that if you're not 100%?
Yeah, so that's a really excellent question and a nuance that, you know, is easily discussed in this group. So the Livy lab is convinced that these mitochondria are not enclosed in EVs, that they are naked mitochondria. We see evidence actually for both. So we can do, you know, density gradients, and we find that these mitochondria do co-purify or equilibrate with EV markers. More recently, John Brestoff has shown in the blood adipocyte-specific mitochondria with small particle flow cytometry are associated with EV markers like the tetraspanins, but there's also a large population that is not. So they are likely naked, right? So I think there's both. I think, and we didn't really distinguish that in this study, but we're really interested in that now. We have some mouse models where we can label the outer membrane genetically, and specifically in adipocytes, we can label the inner and outer membrane of mitochondria, and so we can assess that now.
Okay, and so did you try in your setup to extract manually the damaged mitochondria from the cell, and then incubate your isolated mitochondria on your target cells to see if you mimic the phenotype that you observe with your more naturally secreted, let's call that cell-free mitochondria? Did you try that?
We did not do that because the mitochondria that are in the cell, I mean, there's going to be some damaged mitochondria and there's going to be some healthy mitochondria. So that's part of it. But also we don't believe that the mitochondria that are released are always the same as the mitochondria in the cell. So if they truly are mitochondrial derived vesicles, as we think that they are, or at least most of them, some of those pieces of mitochondria are not going to have the inner mitochondrial membrane. Some are just going to be outer. Some are just going to be inner mitochondrial membrane, and some are going to have both. And various forms of damage, various components on a single mitochondrial level. And so we don't believe that, you know, lysing the cell and taking whole mitochondria is actually reflective of what is actually being released from the cell.
Okay, fair enough. So second quick question related to the second part. And actually the previous paper with the caveolin. So it seems that, you know, in your system, you are not only a very efficient system because you can replenish the cells that have been depleted for a protein. So that strongly suggests that it's a very efficient, you know, content delivery process, which is not very often and I would say rarely observed, right? And initially you proposed that it was between endothelial cells and adipocytes. But
Right.
if I understood the rest of the talk now with the new mRNA,
Yep.
so now you also have a specificity for the cargo, right? Because the route from endothelial cells and adipocytes is true for caveolin, if I understood correctly,
Yes,
but not
yes.
for the mRNA, right? So
Right.
do you believe that you have cargo specificity and cell, like pairing specificity? And how could you
Yeah.
explain that in your setup, the exchange is so efficient?
So to answer the last question first, the adipose tissue, for some reason, this mechanism or these mechanisms to replenish adipocytes is extremely active. So for maybe 20 years since the Shearer lab has been trying to make mouse models of adipocyte-specific knockouts, particularly, there are some models you just can't make. You cannot knock out this protein. And it turns out that a lot of these proteins, when we do mass spec, are actually in EVs. And so we're learning now that even we have 2 other mouse models right now that you can't reduce the mRNA that we think are coming from a cell type in a knockout model. This doesn't happen for all tissues because we use hepatocyte-specific and whole body knockouts in order to show that these floxed alleles work. And so it seems to be something that is very robust in adipose tissue, doesn't seem to happen that much, at least in other tissues.
And can you replicate that specificity with cell in culture?
So yes, so the cell pairs we can, at least for the endothelial cell adipocyte crosstalk, which if you label adipocyte derived EVs or endothelial cell EVs and you do mass spec to see where they go in flow cytometry to see where they go in the tissue, the endothelial cell to adipocyte signaling through EV seems to be extremely robust compared to other partners. But I think that there are specific partners for EV signaling, and I don't understand why that is, or because obviously now we're looking at B cells or a different cell type for this. So I can't really give you an answer to that. I think the cargo is specific, but we are doing RNA-seq on EVs from adipose tissue to confirm that SMPD3 is really the only one or one of very few that is increased in our knockout mice to show specificity, but you never know. It might not be as specific as we think it is.
Okay, thank you. Congratulations again. I'm looking forward to seeing your further development.
Thank you.
So I have a question.
Sorry, you're really cutting out very. Try again.
Can you hear me now?
Yes, yes. Okay, good. Thank you.
I have 2 questions. One related to the mitochondria derived EVs.
Okay.
So you mentioned that you guys think that it's mitochondria derived vesicles.
Mhm.
Where would these vesicles go, or how would they be released or transferred
Released from the cell?
into exosomes? Yes.
Yeah, so the cell biology that's been done with these mitochondrial derived vesicles has shown that they do enter the multivesicular endosome, and then they fuse to the lysosome. So we think that is the route, which there's other ways like, autophagy mediated, maybe like an exofer type release.
Right.
But we haven't pursued that, and we've only been looking at the smaller EVs for this. So we think that is the route.
Okay, so that's in relation to Heidi McBride's work.
Yes, Heidi McBride's work.
And so in relation to the second part of your talk, I was just wondering if you mentioned that ultracentrifugation did not media that was ultracentrifuged before adding it into that assay did not cause mRNA transfer. But I was wondering, of course, there could also be a non-vesicular component. Apart from extracellular vesicles, there may be other protein complexes that could be mediating this.
Yes, you are absolutely correct. And I said it seems to be EV mediated, but I actually don't believe that. I have a feeling that it is something like a stress granule that is being released. It is speculative because it is just based on one proteomics experiment. But we're working on that. But yes, you are absolutely correct.
Okay.
It might not be an actual vesicle.
Okay, thank you so much.
Yeah.
Yes, Claire, beautiful work. Congrats. I want to join my voice to the previous two questions. So related to the first part of your talk, where you showed that these EVs have a protective effect on the heart, right? So the EVs from the adipocyte, and you postulate that they are priming the system to increase resilience to further injuries. So, so do you think that this is only on the chronic versus acute? In other words, have you repeated that experiment using your GEM model, your genetic model, where you do sort of an acute, because you showed that data with the high fat diet, right?
Yeah.
So first question is if you've done that, and two, should this be simply regarded as, should we distinguish between acute versus chronic? So that's one.
I think absolutely distinguish between acute and chronic. I would suspect that chronic, we have not done the experiments, but we're planning on it. The hard thing with our transgenic mice is that, yes, adipocytes are releasing more mitochondria, but they're also releasing these stress hormones and ceramides like GDF-15 and, loss of adiponectin, which is also cardioprotective. So, so we don't want to continue experiments in that model because it's really hard to say, okay, this is EVs and not everything else that's happening. So we are trying to make a mouse model now where we can specifically manipulate cell-derived EVs, specifically like from adipocytes or mitochondrial release from adipocytes. We're far from accomplishing it, but it is one of our goals. But the way that we can do this is long-term treatment of these wild-type mice over the progression of obesity with adipocyte-derived mitochondrial vesicles, right? And so we're working on that, but we suspect that there will be exacerbation of disease over chronic obesity.
Yeah, so I guess in that regard, you also be interested to look at different sources, right? So if you look at patients that have kidney injuries after ischemia reperfusion, right, and look at the heart, if really, I mean, it seems to run counterintuitive, right? So in that case, you get a really acute injury, you have to resolve it within minutes, right? So within minutes, you already have a defect to the kidney and potentially to the heart. So I think it'd be interesting to look at it in that setting.
Yeah,
and then finally, I think the broader translational question. So are you suggesting then, should we then be collecting extracellular vesicles from our obese patients and in a way of protection against cardiac injury?
Yeah, I mean,
we've done that in cancer, right? We've done that in cancer with microbiome and response immunotherapy.
Yeah.
So
yeah, I mean, it's really hard for me to speak to translation at the moment. Like, we know that adipocytes, that in human obesity, there is more circulating mitochondria. Again, we can't show exactly that they're coming from adipocytes, right? And so I think what we need to do is actually have a time course where we understand in humans as well as in mice what those levels are over time, and if that changes the outcome over time. And so it's just really hard to say, right? Because if we believe that these mitochondria are exacerbating disease long-term, then it's probably not the best to sort of mess with them unless we have very specifically defined populations of people that we know will be protected. So it's tough, but we are always thinking about translation and working towards that.
Thank you. Thank you again. It was very interesting talk and discussion, but due to shortage of time, we will continue with the next speaker. Thank you again.
Great. Thank you all.
Thank you very much. Now for the last speech of the day, it's our own Subhashini Mohandiram, who is a third-year PhD student from the Institute of Veterinary Medicine and Animal Science in the Estonian University of Life Sciences. She has a bachelor's degree in medical laboratory science and master of philosophy degree in molecular and cancer biology from the University of Peradeniya, Sri Lanka. For PhD, she is working on understanding the proteins that mediate signal transduction in extracellular vesicles, mediated extracellular vesicle mediated embryo maternal crosstalk during pre-implantation. So Subhashini, go ahead.
Thank you very much, Kasun. Good afternoon, everyone. Today, I'm going to talk to you about how the trophoblast-derived extracellular vesicles modulate the endometrial epithelial cell secretome to contain critical players of embryo implantation. So I'm Subhashini and a third-year PhD student from Estonian University of Life Sciences. Infertility is emerging as a significant social and medical problem affecting the couples worldwide, with average incidence of infertility around 17% globally. So main events necessary for pregnancy to occur are ovulation, fertilization, and implantation. And interestingly, implantation failure accounts for up to 2/3 of the unsuccessful pregnancies in human, therefore plays a critical role in pregnancy establishment. Embryo implantation process is marked by three main steps. One is the apposition, where the embryo come in close proximity to the endometrium, and then it get attached, and then it finally invade the endometrium to initiate placentation. So the coordination between the maternal endometrium and the embryo before the implantation is critical for pregnancy establishment. Different proteins, hormones, and some of the bioactive molecules are known to mediate this communication between the embryo and the endometrium. But we hypothesize that these extracellular vesicles, which is a novel form of intercellular communication, is actually playing a role in this communication between the embryo and the endometrium just before implantation. So about extracellular vesicles, I won't go into details. We have been talking from the morning, but of course they are nano-sized vesicles secreted by almost all cells and have a bioactive cargo with different proteins, DNA and RNAs. So one of the very first studies published by our lab was able to show that extracellular vesicles that are secreted by the outermost layer of the embryo, which we also called as trophoblast cells, can carry RNA molecules that can get transferred into the endometrial epithelial cells. And also these EVs has the ability to induce specific transcriptomic changes in the endometrial epithelial cells, and these transcriptomic changes are actually related to the signaling pathways, which are specifically relevant in the context of embryo implantation. So in the further research from our group was also studying the dynamics of these transcriptomic changes over as a function of dose and time, were able to show that these transcriptomic changes are actually very swift and drastic, and these changes can be actually seen just after 30 minutes of EV treatment. But some of the other questions that remain unanswered when I just started my PhD about 2 years ago was that whether these extracellular vesicles has some proteomic level effects on the endometrial epithelial cells. And during very recent time, and there were reports that actually these trophoblast-derived extracellular vesicles can induce proteomic level changes also in the endometrial epithelial cells, but we didn't know whether these EVs also have the ability to change the secretome of the endometrial epithelial cells. So in one of my first studies, I was interested in determining the secretory proteomic changes of the endometrial epithelial cells in response to trophoblast-derived EVs. So in the studies that I was described in the previous slide, we were using an in vitro model to simulate the embryo-maternal crosstalk during the pre-implantation period. It has these trophoblast analog JAR cells and also endometrial epithelial cell analog RL95-2 cells. What we simply did was we isolate EVs from these trophoblast cells and we introduce into the endometrial epithelial cells to simulate the communication happening between the embryo and the endometrium during the pre-implantation period via EVs. So my experiment plan was very simple, that we had these RL95-2 cells, which is the endometrial epithelial cells. We introduced JAR cell-derived EVs into this, and cell culture supernatants were collected both at 0 hour and 24 hours. Cell culture supernatants were collected at 0 hour just to eliminate any proteomic protein contribution from the EVs itself. So then we can purely see like what is the actual proteomic secretome level changes that is happening in the endometrial epithelial cells. And I also use a control group where I introduce Excel derived EVs. This is like a non-trophoblastic origin, EVs of non-trophoblastic origin. So had the same cell culture supernatant collected in the same time periods, and the sequential centrifugation was done to eliminate the cell culture, the contaminants and everything, and the label P mass spectrometry analysis was done on the samples. So yeah, interestingly, we could see that the trophoblast-derived EVs could induce specific secretome secretory protein changes in the endometrial epithelial cells, and these specific secretory proteins were actually, some of them were well-known players of embryo implantation, including MFG8, or we call it lactadderine, CCT8, T8, LDHA, galactine-3, like that. So also these secretory proteins that we identified were associated with biological pathways such as glutathione metabolism, cellular redox homeostasis, gluconeogenesis. All these pathways are very important in the process of embryo implantation. So in the next step, we further wanted to study the dynamics of the secretory protein changes in the endometrial epithelial cells in response to trophoblast-derived EVs. So we use this milk fat globule EGF factor 8 protein as an indicator, because we identified this protein as changing in the receptive endometrial epithelial cells in response to trophoblast-derived EVs in the previous study. Of course, lactadderine or MFG8 is a cysteine-rich secretory glycoprotein, and the intracellular lactadderine is also known to enhance embryo attachment during embryo implantation, and it has also shown cyclic changes during the menstrual cycle. So somehow is related to the process of embryo implantation. However, not much is known about MFG8 secretion dynamics in the embryo implantation process. So we started studying the MFG8 secretion dynamics by first investigating the MFG8 secretion from receptive endometrial epithelial cells and non-receptive endometrial epithelial cells. We could see that actually receptive endometrial epithelial secrete more lactadderine MFG8 protein compared to non-receptive endometrial epithelial cells. And actually endometrial receptivity is partly established by two important hormones called estrogen and progesterone. And so then we decided to check the MFG8 secretion dynamics in response to these two proteins, two hormones, estrogen and progesterone hormone combination that mimic the luteal and proliferative phase of the menstrual cycle. And we could see, yes, this MFG8 protein secretion dynamics changes in response to these two hormones. Specifically, the secretion is suppressed, but intracellularly, the effect was different. Intracellular, this protein was getting increased in response to these two hormones, but we could not see any significant differences between the luteal and the proliferative phase of the menstrual cycle. And very interestingly, the effect of trophoblast-derived EVs on the MFG8 secretion in the receptive and non-receptive endometrial epithelial cells were the opposite. Actually, the trophoblast-derived EVs increase the MFG8 secretion in both receptive and non-receptive endometrial epithelial cells, and the intracellular levels were actually depleted, saying that some kind of protein secretion is happening that depletes the functional reservoir of the MFG8 protein within the cells. Next, to better understand what kind of a mechanism is involved in this EV-induced MFG8 secretion in endometrial epithelial cells, we did dose and time response studies where we introduce trophoblast-derived EVs into endometrial epithelial cells in different concentrations. However, the response was only seen at a very high concentration, such as 10 to the power 9 EVs per milliliter. This is still a very low cell to EV ratio, which was around 250 nanoparticles per cell. And very interestingly, we could see that in the time response study, that the trophoblast-derived EVs actually induce MFGH secretion within a very short time frame, such as 30 minutes of EV treatment, which persisted up to 24 hours. And interestingly, when we detect the gene expression changes of the MFGH protein in the endometrial epithelial cells in response to trophoblast-derived EVs, we could not see any gene expression level changes. So it is unlikely that MFGH secretion is somehow happening in the endometrial epithelial cells as a transcription activation and then translation and then release of the protein. But it seems it's a rather very swift response of the endometrial epithelial cells. And we also know that this MFGH protein is a very common protein in the EVs itself. So we were interested to see whether these MFGH secretion from the endometrial epithelial cells are actually happening at least partly by release of EVs. So what we could see is when we isolated EVs from the endometrial epithelial cells and detected the MFGH protein, actually it is highly enriched with this protein. So it seems that trophoblast-derived EVs, when it interact with the endometrial epithelial cells, somehow it increases the MFGH secretion from the cell, but partly in the form of another form of vesicles or extracellular vesicles. So in summary, we could see that estrogen and progesterone hormones are increasing the functional reservoir of the MFGH within the endometrial epithelial cells, but somehow it suppresses its secretion. Once the embryonic signals are received, only endometrial epithelial cells start to release the MFGH, which also has known to play a role in embryo implantation. And the effects of the trophoblast-derived EVs on MFGH protein secretion were dose and time dependent, and it happens like within a very short time frame of 30 minutes after addition of EVs. And we believe that maybe it suggests that there's a rapid fusion happening between the EVs and the recipient cell membrane, leading to release of MFGH protein in the form of vesicles. So we are thinking right now that this in vitro model and the MFGH readout hold some promise as a unique platform to further study the mechanism of EV interaction at the recipient cell level. In terms of specificity, we were also thinking how specific this response to this EV and recipient cell pair. But unfortunately, with the experiments I was doing, my feeling is that like many different types of EVs actually can increase the MFGH secretion from the recipient cells. Maybe there are quantitative differences that I don't have much evidence, but I'm really interested to see how specific this response in terms of different EV and recipient cell type pairs. Yeah, that's all. This is the lab group I'm working on, and some of the co-authors of the data I presented here are present here. Thank you very much for listening.
You, Subhashini. In the interest of time, since we are way past time, we will have only one question. But after this, there comes a session. It is all questions, right? So we will have Rosalita's question. Sorry, we are 20 minutes past time already.
Mine is quite small, and I think I'm not sure, like, why, do you know why, like, some EVs are released and they affect the success of pregnancy or implantation? So do you know, like, what is making the difference between the success and in some people and the failure of implantation?
Sorry, I didn't get your question. Like,
so the EVs, you said the EVs are affecting the implantation due to the
yes, EVs are mediating part of this communication between the embryo and the endometrium. If the communication is happening very efficiently and all, it can affect how efficient the implantation is. But then the question is
now, why is it that in some cases some people have they are successful in the implantation, and then others then it results to the failure? What is causing this?
Yeah, the implantation failure actually is a multifactorial case, like not only the EVs, but I think if we understand more about this EV communication and how it helps the embryo to attach to the endometrium, we can improve the cases where the implantation is not very efficient. So this is why we are doing it. Yes, sure, please.
Mother and we have the baby. If you don't have a pregnancy, either the mother is not good or the baby is not good, or mother and baby cannot help each other to somehow communicate. And so as Subhashini is working on that part, that's the reason that the EV comes, where they are somehow speaking with each other. It is multifactorial, but in essence, that is the reason that the EV is important, because EVs help the communication between the baby and mother to pregnancy to happen.
All right, so this is not really a question, but first I want to congratulate you on giving your talk, and then second, I think that Subhashini deserves a round of applause. She was really involved with the organization and all that stuff. I was emailing her back and forth. She was very responsive, so thank you. So thank you for making this also possible. So great.
Thank you very much. It's not just me, like there was a whole organizing team behind.
So thank you very much. We'll do the souvenirs. So that brings the end of the lectures of this evening session. Thank you very much for listening. Now, Professor Aridesa Fasile will take you in a moderated discussion titled, Do Extracellular Vesicles Have Specific Target Cells? So, up to you, Aridesa.
Okay. I put my thing there, but I cannot see. I don't know where I put it on the desk.
Yes.
Is it okay? We had something around 15 years of coast actions, and a number of people that are here are the results of the coast actions that we had before. As a result of that, we created a kind of community for ourselves. And one of the things that we were doing during our meetings was every day we had a kind of moderated discussion that people were giving opinion about the things that was the topic of that day. And that particular sessions were very useful because it would somehow, because different people come, give different lectures and go on. And then you are thinking at the end, you have one opinion, the other one has an opinion, and so, but that is not aired, and that kind of conclusions that can be made by those kind of opinions are not somehow reflected in the work that we do. in that respect, we thought, okay, we can maybe do the same tradition here, have a little moderated discussion, and we can speak about the different things that we heard today. The topic today particularly was about the fact that do extracellular vesicles have a specific target cells? That was, we heard about different models and different. I have to say, if I was asking this question maybe 1 year ago, 2 years ago, 3 years ago, 4 years ago, maybe it was a different answer. But at the moment, what is the general feeling that you have? Do you think that extracellular vesicles are specific, or do you think that they are non-specific? Is this, and before maybe, yeah, I can just, yeah, go ahead. And we would have two people that would somehow go here, and maybe Chanaka can be in the other side, and you can, yeah, Matusha can be in that side, please. No.
There we go. There we go. Yeah, so I was going to say that I think the answer to your question is in your question, in that you're asking this community a question that is actually existential. And I frankly believe, and this is just by looking at the literature and our own work, that it's actually a guided process. It is a regulated process. I don't think that it will be just haphazard. But we can also argue that maybe some cargo will signal via some very specific receptor ligand or whatever you want, but some other cargo is a little more general, where you don't need that specificity. So I think in the end, it comes down to also the cargo that you're actually referring to.
Then you are thinking there is a heterogeneity. There is some of the extracellular vesicles that are produced, they are just produced for general use, and they would be completely there. And some of them are specific for some kind of particular response or a. Do we have anyone that says no, they are all complete? Because I think there are quite a lot of converted people here. Let's see. Yeah, please go ahead.
Yeah, I mean, similar to Dr. Lee's presentation before, we've done similar work with PBMCs, and if there's a mixed population, the monocytes always win. So a phagocytic cell, I mean, obviously other cells do take EVs up, we see that, but if you put them in a mixed population, it'll always be the phagocytic cell that takes up, 95%. So I think that overrules any of the specificity. And it depends obviously what cells you've got when you do your uptake. And again, all the in vivo stuff we've done, it's in the liver, and after about 10 seconds, I mean, it seems to be all, cleared in the spleen, cleared in the liver. So you're thinking macrophages, Kupffer cells. So I'm not sure.
Okay, that's the whole idea. Somehow to Matusha is, then we have one, maybe let's go one round between everyone to somehow come out. Then we have one pro that there is probably maybe two, it's yes and no. The other one is no, I think it is general, it is no specificity. We would have get, and then we have the lady here from NUNS. Yeah, go ahead.
Okay. So I think it's important. I mean, we are discussing specificity, but it's important to replace the specificity within the context. And I'm going to try to explain myself, right? So physiologically, of course, you have to maintain a barrier between tissue because you want to keep the integrity of certain tissue. So it wouldn't make sense if you only focus on physiology to have a continuous exchange, non-specific between the 5 different tissues. They have their proper identity, then it keeps the identity, right? So then you would expect to have specificity and some kind of regulation within the physiological context. But now if you simplify and you try to develop, cell culture to measure the exchange, you may lose the specificity because what you are looking for now is just the expression of the machinery. Again, I will do analogy, and I like to do analogy and comparison with other systems. If you take the well-established, transport mediated by LDL, for cholesterol regulation. So we know very well about, the physiological function, what tissue is going to do the job to uptake, the LDL and regulate the cholesterol. But actually, the way it was discovered by Brown and Goldstein was using fibroblasts from patients who are depleted of the receptor, right? And so if you take a fibroblast, it will express the receptor, and you can use that as a model in culture. So physiologically, the fibroblast has no role in the uptake of LDL, but in cell culture, it's useful. You can use it because the receptor is expressed. So that's why I think it's very important to replace specificity of the mechanism and then specificity in the physiological context.
Okay. Then in the context of the system, it can be a specific or it can be general, depending on which system.
Yeah, it's really the level, right? Speciality at the molecular level and speciality at the physiological level. And perhaps it's both.
Okay. We will have Julie.
Okay, thank you. I'll try to make my ideas clear. Not sure it's that clear up there. So one thing that is striking is that because of the conversation with the monocyte, the idea is that the specificities come from the target cells themselves. So not provided by the EV, but by the target cells. And we can also think about the same from the endothelial cells. I'm coming from the endothelial field, as there will be the first encounter of circulating EV. So one can argue that it's more a matter of chance that cells will target endothelial cells when it comes from circulating AV or monocyte in some clearing organs with resident monocyte. So it's coming from the specificity of the target cells, but also a sort of a chance of EV meeting the right target cells. I don't know if it's clear in that sense. And also, we all agree and we all show like super heterogeneity of the EV in terms of cargo, in terms of size, in terms of numbers, so and lipids. So at the end of the day, it's finding this specificity of targeting with one single EV, a specific cells like virus can do. I'm not quite sure anymore.
Okay. Then there is a specificity, but that the specificity is explained by the target cells, by the recipient cells. And so hence a cell, donor cell, is just producing thousands or millions of different EVs. And then if lucky, the target cell would reach it and has the specificity to interact with it. Let's see, like,
You sum it
something.
quite well.
Yeah. Is there any other, okay. Now we go for the second round, or okay, let's that two, and then we would go to second round getting back there.
In my opinion, I am not sure about all kind of cell or healthy cell in the body, but in the case of cancer cell, I'm totally sure that they have specific targeted cell size. When we have, for example, lung cells, they metastase to some specific organ, it means that exosome also isolated from this. Lung cells, they have only specific target cell. This is just my opinion. And we have to do more research about which specific receptor we have in different cell line of cancer. And we look for some ligand in the ligand is a cancer cell line and cancer cell-derived exosome, and we look for some specific receptor in different cell line that this cancer metastase to these organs.
And the molecular basis of it, we have to understand to be able to know what's going on.
Yeah.
Okay. I just try to simplify. Please, Rohit.
Professor Alireza, since I'm very new to this field of EVs, and thanks to your group, I'm very excited to know about them. I might offer a naive comment here. So since Dr. Claire told us like there is no established research towards that there are EVs that can show some function in spread of the disease, that can show some function that it's been used in communication. Maybe now that if we'll look for how this specific mechanism that in the cells happening, that in the case of stress, like she said, like in the case of disease, led to the specific mechanism that led to release of these EVs, then we can solve the question regarding are they specific, and if they are specific, how is this communication between two cells? So maybe we can look for mechanisms now in EVs, and that can offer some.
Thank you very much, Roish. That's very, very kind of you. We have to look at the mechanism. Okay, we go now to the second round. Please.
No, I think.
Oh, sorry, Lalbert.
No problem. Thank you. I actually, in my opinion, there could be both specific and non-specific incorporation of EVs. And of course, all these in vivo models and microscopy-based assays that Jackie Getz and Frederick Berbe are currently developing, I think that these are going to be very important, trying to visualize where some type of cells are generating some type of EVs and where do these vesicles go, and we can visualize this. And so once we know if there's a specific cell type that can generate a specific type of EV that goes to a specific type of cells that internalize these EVs, then at that point, maybe we'll be able to dissect what are the molecules, perhaps in the donor and acceptor cells, that are enabling this specific intercommunication, as opposed to other cells that do not incorporate these EVs. But again, there are about 10 to the power 16 EVs in the bloodstream or biofluids. And so we also need to remind ourselves that a lot of these are going to be cleared in the urine. So there may be also non-specific mechanisms by which EVs are detected and cleared from the bloodstream and other biofluids by macrophages and other type of cells by non-specific mechanisms. So there could be both.
Then we have three advocates of three people here are saying that it can be a specific and unspecific. We have one that thinks that it is unspecific. Then nobody thinks that it is specific. This was round one. Okay, we go for the round two, please.
All right, so let's see how round two goes. But so I want to push back against those 2 points. One, on the interpretation of these experiments where you do co-culturing in heterogeneous population, and it's always a phagocytic cell that actually wins. I mean, to use the term that you use. But I'm not convinced that in itself is sufficient evidence to say that the process is non-specific. You can have a macrophage that has a receptor, and that one will still do specific EV uptake, but then you can also have a macrophage that can just do normal phagocytic processes. In both cases, you're still going to take the EVs, right? Maybe more with the phagocytosis process, right?
Yeah. Maybe the Chanaka, maybe you can bring the thing from that side.
Oh, sorry.
When I say not specific, I don't mean like fusion as in that non-specific. I mean that I don't particularly believe at the minute that there's different EVs that could 100% target different cell types. I mean, a lot of it probably is receptor mediated, scavenger receptors, things like this. It just does seem that if there's a phagocytic cell present, it's got a better chance of taking up any EVs that we put in the system. How that translates, I have no idea.
So I think the second half of that question is, you're doing regulated EV uptake or random EV uptake to achieve some physiological output. So physiological output in the recipient cell, right? So not necessarily just clearing the EVs out of circulation, but you want to achieve a signaling output in the recipient cell. So in that regard,
No, they do
I would
react.
argue,
Yeah,
what?
they do react. Yeah.
Yes.
Yes,
right?
there is an effect. Yeah.
Okay, let me to somehow, that is actually the next part that I have somehow tried to put it here. You know, I am Iranian, and in our literature, we have one gentleman, Molana, and he has a story. He put an elephant in a dark room and then put a number of people in that dark room and said that what you see, some of the people started to touch the elephant leg. They say, okay, you see, there is some pillars that is like this and that. Some did go to touch the ivory and they said, oh, it is like this and that. And so, and then till everyone said some tail, some the other parts or the ears of the elephant. And then at the end, when the light was shed into the room, they say, oh, great. This is the elephant that we see there. I feel we are a bit also in that situation at the moment. And one of the reasons that we are in that situation, maybe we need to at first define what do we mean by a specificity when we say that the EVs, are they specific or not? And that is the reason somehow I brought this here. Is EV a specificity in us? Are we, because I think we all agree if EVs want to affect a cell, they should at first bind to the cell or should touch it. Do you agree with this? This is it. Then if EVs, any kind of EV can bind, is that the specificity? Does the EV getting internalized in the recipient cells? Is that where we define that they are specific or not? Is the EVs that can somehow deliver the cargo into the cell, is that where we have to say they are specific or not? Or when they inside that cell, as our friend there was saying, they would have the ability to read that cargo, they would be also specific. Or when, as Greg was saying, when the change come into the cell, physiological changes, things, and so that is going to be a specific. And can I also say, there are data like Subhashini data that shows even just probably as fast as binding is enough to somehow exert the effect of the EV. We don't need even the internalization of the cargo. Now, can I change the question and say, what do you think is the effect or the function of the EVs that we see? At which of these levels are specific and at which levels are unspecific? In my own the experience, what I see, it looks to me EVs bind to any kind of cells. That's the most unspecific part of EV interaction. I don't know. But please do comment. This is maybe round 3.
So I don't want to, before I move to round 3, I think it's important. So I wanted to address Julie's question, right? Yeah. So which was thinking that specificity has to be instructed by the recipient somehow, right? So the recipient cell will then say whether or not this event has to be specific. But I don't think that in itself is sufficient. I think in terms of molecular cognate recognition, you need to have the receptor that's from the EV, and then the recipient cell has to have the matching signal for the event to actually happen, right? But
you know, more sorry. Just to go, to go a bit beyond receptor ligand couple, this ligand from the EV could be the surface of lipids or this, you know, more than.
Yeah, compatible interaction, compatible molecular interaction, basically. Yeah, yes. I can live with that. Yeah. So now going to round 3. So I think the binding, personally, I think the binding, I mean, the initial interaction, I want to say that's random. And it is what happens afterwards, whether you bind with sufficient affinity because you have this sort of recognition, then that can trigger internalization and things downstream. So that is for processes that are regulated, right? Because remember, I'm in the school that both of those can happen depending on physiological context.
May I add something like, I just want to have an opinion. Then if I say EVs are not specific, but the cargo molecule inside the EVs may be specific, like EVs can bind unspecifically, but the cargo molecule inside the EV may do the specific work. What do you think about that?
The internalization or the delivery or the reading of it or so is specific, but the binding is not specific. Yeah. Suhashini?
Do you believe that there's a difference in the binding affinity between different EV and cell type pairs? There are some reports
That some of them would bind tighter, some of them would bind less tight, and that would be the specificity of them. Okay, one.
Is the technique sophisticated enough?
Okay, I am the moderator. I have to give the permission that anyone speaks. Don't forget that. The lady in blue would get that. Yeah.
Thank you.
Yeah.
Luckily, I'm not
Chanaka,
red.
that was for you. You keep that to yourself till I say to whom you will give it. Yeah, please go ahead.
No, about the interaction with the membrane of the target cells, I can tell that we saw also different kind of adhesion. I mean, we have vesicles that just is like I showed you, and many others that looks like I call them balloons. So they have like nanotubes or something resembling, and then they tether there, and they usually do not move over the surface. So, but this is just an observation. To study this would be really, I mean, pin in the neck for some PhD student who could do some statistics on this. So the interactions could be different, the adhesion could be different. And also about specificity, I would like to point out also the compartmentalization of the interaction we are talking about. So for me, it's easy to think about the CNS. So this is quite a closed system. So we do have microglial vesicles and all other glial vesicles, and also neuronal vesicles, which are very few in comparison to the glial cells vesicles. So, and there we also observed some internalization by the neurons in co-culture. So we have the transverse system, classical transverse system, and we had glial cells on the top and then neurons on the bottom. And we saw that there was also modulation in some markers. So it looked like messages are transferred also in the co-cultures. And of course, there you have all the soluble factors, but we compared at the same time isolated vesicles and we incubate the cells with those, and the results were quite similar. So bona fide, there's also in culture some internalization. Of course, the ratio and the efficiency of the the transfer of signals. Let's not call it internalization, since of course you can also have signaling through something which is loaded on the surface of the vesicles. You don't always need to have an internalization or fusion or transfer of things, of course. So to me, it could be, half and half. So I mean, specificity is also regarding to the compartment you're looking at. Of course, the bloodstream vesicles, they can reach many more tissues, but first the endothelial cells, and then they have to cross the tissues. So, and then you can also have, as Albert said, some kind of discard, so trash materials that you want to get rid of through urines. So yeah, I would not be that cut clear. It is yes or not. We, in my opinion, we still are beyond the point of knowledge in which we can handle this easily. But there are evidence in literature that there are how you want to call specific interactions, so targeted interactions. You can have by chance contact with, for example, astrocytes, but you'll never see movement of vesicles on astrocytes. Why? I don't know. So we will never observe. We never observed. So possibly you really need those wires to get the diffusion of the signals also to vesicles.
Thank you very much. Okay, who are the ones that wants to give opinion? But no, at first the ones that didn't give. Sorry, if you would go that haven't given any.
Thank you.
Sorry.
So the recent presentation, they just say that EVs are helping in myocardial protection. So if it's non-specific, then how would you induce myocardial protection if you are just sending messages by chance? Would we have myocardial protection? I think it's targeted, perhaps. And so maybe the sender is targeting specific receptors, but is sending so many signals so that by chance, then the specific target cell gets the message.
Then because we give them a function in
the cargo, yes.
I think then they should be specific, but yeah, okay, that is she. Sergey, please. Yeah.
Also maybe comment to your first statement about that EV to have effect must to touch the cell. Actually, you could imagine this situation easily that in EV you have some decoy receptors, and they can just take out signals. For example, in case of morphogens, no, it's easily possible that you have, for example, Frizzled receptor on the surface of EV, and it's decoy the wind, so it doesn't touch at all the cell. And such a way, it modulates the signal. Yeah.
Okay. Okay. And this is another one that it doesn't even to bind; it can touch and go. Everything. Touch this. Okay. We have GetNet there. At first, those that didn't get any chance to say anything, and then we would go to the next.
Okay, yeah, I have just a simple opinion. For example, we have donor cell, like EV is derived from donor cell, and we are hoping it will bind to the recipient cell. But really, we don't have anything even with the proper understanding how the binding work from the donor to the recipient. In other words, the real mechanism, I think, and we just speculate, I think, the mechanism. This EV comes bind with receptor mediated kiss and run, like really it's not really clearly done heavily. And another thing also, do we have even enough presence of receptor site on the recipient cell, which matches to our donor EV? So that need to be clarified. In my opinion, if this EVs are, biogenesis pathway is totally different. We have endosome-derived one and from the microvesicles. That means we have different size group. Different size groups means different cargo. That means all these EVs have been, it's like a message packed in different sizes, but different, some might be the same content, mixed and totally different. So I will, in my opinion, this is my opinion. It's like both. It will be specific and non-specific. And that's all.
Thank you very much. Okay, we have one from the round 5. We have one from round. Okay, wait. Is there any other one that is still in this round has not given? Don't really. Okay.
So I think I wanted to provide some answers to the previous question. So there is evidence, for example, in Drosophila, when wind is produced, right? So it's lipidated and it's on these vesicles, right? And you actually can signal from vesicles, right? From the extracellular vesicle itself that engages the receptor. So there is evidence for that. Second, in breast cancers, right? So it's been shown that you have one of those CDs, 47, somebody, the name changes, whatever. But they identify a cognate binding partner on the recipient cell to facilitate EV uptake. So there is evidence for that too.
Yeah. I don't want it to become a one-to-one person, but okay, GetNet and the gentleman there.
Actually, even in today's presentation, somebody said it's 30 minutes, the reaction and triggering will happen in 30 minutes, some 30 seconds, some 4 hours. Like, how do you know, like, your EV is really active? But because the half-life of EV is really quite limited. And I don't think so. Everything what we see is only because of EVs. There are so many players. Yeah.
Okay. And we have the half-life also coming to the system. Yeah, go ahead, please.
I'm gonna backtrack a little bit because I don't want to sit on my own at dinner. Regards to the specificity again of the uptake, your annexing work actually shows that it was specific because, I mean, this is something that's lacking from nearly all studies. If we show a specific way of blocking the uptake, then it is specific. I'll argue a little bit that it's a scavenger receptor that obviously it's blocking the PS, is that right? The annexin? Yeah, so it's blocking the scavenger receptors, so it could still be a phagocytosis kind of immune clearance thing, but I concede it's specific.
Yeah. Okay. One thing I think for sure, and I'm thinking that we are reaching to the end of the time. It's nearly at 17:35. We have another maybe 10, 15 minutes. Can I make the conclusion that it's very, very hard at all to define if it is still as specific or not specific? But myself, I think it is a very, very important question to somehow look at and somehow be able, especially if we are going towards whether diagnostic or therapeutics and so. One thing, I don't know, maybe I don't look so much in the literature. I don't see EV community so much concerned about this or trying to bring this up as a issue that we have to look at. Or is this my problem, or I am not looking at it very hard enough, or is it not a topic? Everyone is happy to put the EVs to deliver cargo for pharmaceutical companies into the cell. That is definitely, and you know, and then as a result of this, it comes also when we are thinking what we are doing is physiological or not physiological, because then it would become, we start to make models that sometimes I'm just thinking, if EVs are specific and they're doing this, it doesn't work that you have to load the EV with billions of EVs to do what they have to do or so. I don't know, but am I clear what I'm trying to say? Or yes, okay. Do you think that I should be worried or not? Yeah, Matusha, yeah, please.
I think that's, you know, about the consensus, about the specificity. Again, it's, and you know, to me, the main question that need to be answered is, one, what is the main function of EVs? And so if somebody can, you know, answer that question, that physiologically relevant question, what's the champion function of EVs? So then you can really try to treat the specificity and go deeper into the mechanism, because what you know what you are looking at. So I think before, you know, trying to see the specificity, but it's connected, is what's the main function of EVs? And that, what I think that is a big thing that's, you know, missing for the field. We have many, you know, function. They can do many things, but we don't know what's the main function. As we know for other molecules that do intercellular communication, if you take an hormone, you know exactly what the hormone is going to do, right? And what's the main function. We don't have that level, even at the, you know, very broad and high level of, you know, physiologically function.
Okay, thank you very much, Greg. Lucia, is there any other one? Oh, we have one newcomer. Sorry. Go ahead.
Hi, so I'm the only one, I guess, from a chemistry background. So I would like to go with whatever he said, like with the context and the application, like we should look to what the end goal is, because in my work, according to what I know, like it's all from the literature I've done. I haven't done anything by myself. So it's all the literature I've read. So I think in my case, I need all these to be there because I'm working with drug delivery systems. So I need the binding to happen, internalization to happen. The cargo should be delivered. It should be read and it should make changes in the cell. So it's like what I've read is like, there are two types of, like, we'll go with the autologous exosomes and heterologous exosomes, like which is from the cancer cells. Like, so I'm talking about pancreatic cancer cell. So there is autologous, which is from the cancer cell, and heterologous, which is not from the cancer cell. But both do go into the site, like to the pancreatic cancer site. Both are uptaken. I don't know how to say that, but both do go into the site. But the autologous ones have more tropism towards the cancer cells. So it shows a potential tropism. And the cargo, which is, in my case, it's a gemcitabine, it's just a drug. So it doesn't have anything to do with targeting. It's just a molecule sitting inside. So the tropism is caused by the exosome. So it's, in my case, it's based on the origin of the exosome. So it's like, depends on the context and the application. I think it changes. And also sometimes, I don't know, I've read like we can even make modifications on the exosomes, like bioengineering stuff. I'm not sure what they're talking about, but still they make modifications on it to target specific sites, even in the cancer cells itself. So for me, I need all of these things depending upon the context.
Thank you very much. Then trophy set. So, any other comment? I saw, is it round 6 or is it still round 5? Yeah, well, no, up to you. Yeah. Okay, we are going to still round 5. Yes, go ahead, DR.
In my opinion, we can just decide about the specificity of target cell about when this exosome have the change or can change in the cell or not, because all cell maybe take the exosome, but some of them can be engulfed by lysosome, or some have no specific cargo to the cell. But if something happened to specific cell, in this case, we can say that exosome have a specific effect on specific cell. For example, if we take the exosome, we isolated exosome from cancer cell and we treated with 3 kinds of macrophage, M1, M0, M1, M2. And we find that in each condition, they have specific effect. So in this case, we can say that, or we can define the specificity of exosome by the change in the cell. So.
Okay, then the function is the most important issue for you. Albert. And we have 2 minutes left. Yeah,
yeah, just following to what I just said before, and I would like to perhaps modify a little bit of what I said, because I think most of us, when we think about a donor cell that releases extracellular vesicles, and these vesicles are internalized by an acceptor cell, I think we think that there's only one specific type of extracellular vesicle generated by the donor cell, but the donor cell could be generating many different types of exosomes within the same endolysosome, and many different types of microvesicles generated from the plasma membrane. That even makes it even more complicated. From all this heterogeneity from one single cell, some of these vesicles could go specifically to some target cells, but some of those other vesicles could also be distributed, biodistributed in a non-specific manner. So I think the problem is very complex.
Very complex.
Yeah.
Yeah, okay.
Hey
On
guys,
that account.
can anyone hear me? I'm not God, I'm Claire. Can you guys hear
Oh,
me?
Claire,
Hi.
maybe we can see you also. Let
Oh
us to
yeah,
share your
sure,
picture.
sure. I
Wait,
wanted to put my 2 cents in. I think I really agree with pretty much everything you guys are saying. And I think that a lot of it is true, even though it seems counterintuitive. So I think it is true that there is specificity and that there is not. So I think a lot of the phagocytic cells might use these scavenging receptors to take up EVs and as a way to sort of like clear the damage. So for example, these mitochondria that are released, the adipocytes are really releasing them because they're damaged mitochondria. The adipocyte doesn't want them, it can't take care of them, and the macrophages are degrading them. So I think when the macrophages can't take care of it, or that the EVs escape the macrophages, that's when they can actually get into circulation and hit other target cells. So I think the macrophages and other phagocytic cells can be non-specific, but that also regulates the function of EVs systemically, right? Because it gives the EVs the opportunity or prevents them from binding other cells. And then I would just say one more comment. So like we have a genetic system where we can fluorescently label all adipocyte-derived EVs, look by flow cytometry of all the different cells in the tissue and see where these EVs are going, where they're internalized. And there are definitely very specific cells that they hit. Of course, macrophages are up there, but endothelial cells are also just as much taking up these EVs as macrophages. And that is like the perfect competition experiment, right? Because it's in the tissue and the EVs are just being taken up naturally by whatever is taking them up. So it's not distributed equally between cells. Some cells you see no signal whatsoever. Even larger populations of immune cells, you see no signal from adipocyte EVs. And then we also used Greg's beautiful system of uptake and offloading the nanoluciferase HSP70, and we can show that endothelial cell EVs are taken up selectively in adipocytes, and they are offloaded by like double or triple the amount of efficiency than if they're taken up by a different cell type in the tissue. So I think it's both true. I think there's some non-specific uptake and by potentially more phagocytic cells, and there are specific uptake as well. And then one really final point, the EV corona, I think is really important. So adipocyte EVs have adiponectin on the outside of them. And so that is a potential mechanism for targeting because there are adiponectin receptors on other cells within the organism as well that could help target these EVs to those cells. So those are my thoughts after listening.
Thank you very much. I didn't expect it that you were still there. Sorry. yeah, and the picture because we don't see. But okay, because we are reaching nearly to the end of the time. Me and Subhashini, we wanted to write a review about this, and poor Subhashini, maybe 2 years, 3 years, she wrote the things and she sent it to me, and I gave comments, and then we sent it once out to the Jeff. And it come back with actually very, very good review, but they were rejecting it, of course. But it came back with very good reviews, and still it is there. And when I look at Subhashini, I think, should we go again? And partially, it was very frustrating to put everything in one review paper and somehow I can see it here even. All of you are working in this field, but still, okay. But I think the common sense at the end is, or the safest one is to say that it looks to be maybe sensitive, maybe specific, or maybe not specific. None of us can go and somehow bring it. But I personally, I think it is a very, very important question to answer and to be looked at. And I also agree with Greg in the sense that maybe answering these questions requires answering the other kind of questions, that what is the purpose of this? Hence, one of the things I would do, and this session is recorded, please do send an email to me if you feel interested. And I want to somehow gather a kind of group that we may be writing a little article, maybe commentary, or writing a kind of review. I don't know. I have to somehow think how we can do it. If you are interested to contribute, that we would have something to say about are EVs specific or not, and quite a lot of you that are here are people that have published quite a lot in the EV field, then it would bring more somehow weight to that paper. And also, one of the papers that have changed my view about the science and what I do, and even in my career had been really, really affecting me, is a paper that the title of it is Why a Biologist Cannot Fix a Radio. Just go and look at it. It is written by a Russian gentleman that he works in the United States. Amazingly, somehow explains to you what we lack as biologists to be able to fix a radio. It is very eye-opening. I hope this kind of, and that brings me to the point that, okay, if you work on such kind of questions that are looking very philosophical, it can actually change the way that you look at the way that you are doing science, or you want to somehow solve the problems that you get. Okay, I don't want to speak more anything. Then I think by this, I'm going to close this session. I want to thank everyone that was today here. And if I am right, yes, let's have a round of applause for all our speakers that came in. Very good job. Also those that were online, and thank you very much for being still there and listening to us. I think the next part is going to be the dinner that is going to be at 7 o'clock. We have 1.5 hours to reach it. Do everyone knows where the dinner is going to be held? Yeah, we can be sure that you would reach there. It is going to be in UT Cafe, and we would have